Good morning, respected delegates, dignitaries, researchers, and one and all present here. We now begin day 3 of IPSCON 2025 with one of the most prestigious academic highlights of this conference, that is Dr. S.B. Pandey Oration. Dr. S.B. Pandey Oration is a distinguished academic honor instituted to celebrate the legacy of Dr. S.B. Pandey, an eminent pharmacologist whose pioneering contributions have significantly shaped the growth and direction of Indian pharmacology. This oration recognizes excellence in scientific thought, innovative research, and lifelong dedication to the discipline, and ultimately reflects current frontiers in research and therapeutics. The oration upholds Dr. Pandey's legacy by promoting scientific exchange and inspiring future generation of pharmacologists. To guide this prestigious session, I have the honor of inviting our session chairs, Dr. Pawan Kishan and Professor Nusrat Shafiq, to kindly take the dais and formally open the proceedings. Before we begin, I would like to briefly highlight the accomplishments of our esteemed chairpersons. We have Dr. Pawan Kishan, Professor and former Head of Department of Pharmaceutical Sciences and Drug Research at Punjab University, Patiala, an academician of high repute. We are honored to have Professor Nusrat Shafiq, Professor in Clinical Pharmacology Unit, Department of Pharmacology, PGIMR, Chandigarh. She serves as the member secretary of the Institute Ethics Committee and has played a pivotal role in establishing systems for serious adverse events reporting. She is currently a member of Institute of Ethical Committee, AIMS, Jodhpur. We invite our distinguished chairs to now guide the session by formally introducing our speaker, Dr. Anuradha Majumdar. May I invite Dr. Anuradha Majumdar on the stage?
Thank you. It's my pleasure to invite Dr. Anuradha Majumdar along with my co-chair here. Dr. Anuradha Majumdar is B.Pharm, followed by M.Pharm and PhD in Pharmacology. She is an associate professor at the Bombay College of Pharmacy, Mumbai. She has been teaching and conducting research since 1999 and serves as chairperson of the CCSEA-approved Institute Animal Ethics Committee. She's former Dean, Faculty of Science and Technology, University of Mumbai, and has held several key positions, including member of the University Senate and Academic Council. Her teaching and research interests span physiology, pharmacology, toxicology, pharmacovigilance, and clinical pharmacy. And with these words, I invite her to take this esteemed oration. And the topic of the oration, I think, would be introduced by my co-chair. Semaglutide saga, to fix it or not to fix it. So let me do it myself. So Dr. Anuradha is going to talk about one of the burning issues in current practice, research, everything. She's going to speak on semaglutide saga, to fix it or not to fix it. And without much ado, we think we can start with the oration. Over to you, Dr. Anuradha. Thank you.
Thank you, ma'am. Can I have the first slide? A very good morning to all. It's indeed my privilege, pleasure, and I'm humbled to be delivering the oration lecture in memory and in honoring Dr. S.B. Pandey. When I was trying to figure out what I would like to talk about, I decided not to speak on my research area. I decided to speak about a very hot and contemporary topic, which is the GLP-1 agonists, which are sweeping the market. So the talk has been titled To Flex or Not to Flex, as you understand, in relationship to the muscle and the semaglutide saga. So this presentation is dedicated to and is a tribute to late Dr. S.B. Pandey. So on 14th November this month, while we were celebrating Children's Day, it was also the occasion of the World Diabetes Day. And Times of India had a very important article which stated that India's high diabetes burden is quietly breaking the nation's heart, which was from renowned cardiologist Dr. Ramakant Panda, who practices in Asian Heart Hospital in Mumbai and is renowned all over India. They say ICMR India B reveals that India's adult diabetic population rose from 32 million in the year 2000 to currently it's pegged at 90 million adults by 2024, which is showcasing a 10.5% prevalence. Recent studies reveal that roughly 1 in 9 Indian adults have diabetes, and most are undiagnosed. Majority of complications are silent but serious. Diabetic heart disease is under recognized consequence. Chronic hyperglycemia stiffens the heart and raises the risk of heart failure with preserved ejection fraction and other cardiovascular events. So coming to the promise of the GLP-1 agonists in type 2 diabetes and obesity. So GLP-1 and GIP receptor, we are aware of the pleiotropic role of the integrins, which are gut hormones and which act on myriad of organs and systems. So the GLP-1 and GIP receptor agonists enhance insulin secretion, suppress appetite, so it gives a feeling of satiation, and improves energy metabolism. Now, we started off, the market came with liraglutide, then came semaglutide. Semaglutide and liraglutide, both identified as GLP-1 agonists. Later came the dual agonists, which now is creating a lot of buzz in India, and that is tirzepatide, which is a dual agonist which acts on both GLP-1 as well as GIP. It is both dual agonists. And now is also development of retatrutide, which is under development, which is a triple agonist. So it acts on GLP-1, GIP, as well as on glucagon receptors to activate multiple integrin pathways, producing greater glucose control and weight loss than the single GLP-1 agonist like what we identify as semaglutide or Ozempic. So you can see it has, as I said, myriad of action. Some of the actions in CNS and the periphery are direct, some are indirect, both for associated with the GLP agonism as well as the GIP agonism. So this is another reflection of the same. You can see the pleiotropic action. So over and above the pancreatic action, which is the one required for diabetes control and release of insulin, there is also impact on liver, prevents gluconeogenesis, action on gastric emptying and slowing it down, cardioprotective action, which is again, last month we saw some news on that from US FDA, and also action on brain. It improves memory, improves the cognitive function, which is something which we look forward to in the elderly. Coming to the GLP-1 agonist drug, semaglutide. The semaglutide, these are amino acid sequences, polypeptides, 31 amino acid chain with 18 fatty acid attached to it, and it has been promoted for type 2 diabetes. And now the current scenario, last month, US FDA approved it for even to prevent and protect against major adverse cardiovascular events, that is MACE. It has gained a lot of prominence as a weight management in obesity treatment. We have seen Karan Johar, who was known, the producer director, and now he looks absolutely thin and skinny, right? And the buzz in the news was that he is into Ozempic and semaglutide, right? So it is creating a lot of, people want to lose weight without really working on their diet and exercising, right? They think it's a miracle med by which they can lose weight. And so Ozempic, these are brands from Novo Nordisk. Ozempic is the drug which has been marketed for type 2 diabetes, Wegovy for chronic weight management, and Ribelsis. Both of these are subcutaneous injections, while Ribelsis has shown the innovative product is an oral formulation. Next, tirzepatide, Mounjaro, which is again, it has just been launched in March this year in India, and everybody is talking about Mounjaro. So Mounjaro is a 39 amino acid polypeptide. Both of them have long half-lives, both the semaglutide and tirzepatide. It is a dual incretin pathway activating drug, GLP-1 and GIP agonism, and has multitude of effect, a synergistic effect which gives greater glucose control, enhanced weight loss, possibly lean mass preservation, marketed under the brand name. Now, when these companies, this is by Eli Lilly, companies have marketed almost the doses are similar or slightly marginally higher for weight, the obesity control, but they have marketed under different brand names. So here Mounjaro is marketed for type 2 diabetes and Zepbound has been marketed for chronic weight management. Yesterday, Reuters had a news which stated that Eli Lilly, which is the innovator for Mounjaro, tirzepatide, has reached the trillion US dollar group, which was till now only attained by tech companies, the high-end tech companies which we know about. So this is the only pharma company to have the market value of more than 1 trillion, and this was the news yesterday. So you can understand why. Liraglutide, which was developed before semaglutide, has lesser efficacy. This is also a GLP-1 agonist, and it comes from, this is also from Novo Nordisk. It comes with the brand Victoza and Saxenda. Victoza again for type 2 diabetes, and Saxenda is for chronic weight management. This is a shorter half-life compared to tirzepatide and semaglutide. Now coming to, pitching these three important molecules which are there in the market against each other. So semaglutide versus liraglutide versus tirzepatide. So as I said earlier, semaglutide, two brands, Ozempic, Wegovy, semaglutide oral tablets, Rybelsus, which shows, absorption from the stomach. It is a caprylate based technology which helps in absorption, although it's so difficult to have polypeptides being absorbed without getting denatured in the gut. Tirzepatide comes with the brand names Mounjaro, Zepbound, liraglutide, Victoza and Saxenda. This is the only one in pink which I've highlighted, which is a oral preparation of semaglutide. Now this slide is heavy, but I have taken a lot of effort to, summarize the market status and what's happening in this area, because 2025 has been a paradigm change when it now comes to diabetes and obesity control, especially in India. The global West has seen these drugs 2 years back, 3 years back, but we have got the approval this year. So coming to innovator company, as I said, that Novo Nordisk has developed semaglutide, Rybelsus, which is the oral version, is also from Novo Nordisk, obviously. Liraglutide, earlier version of GLP agonist, again from Novo Nordisk. Tirzepatide from Eli Lilly, which is creating a lot of attention because of its dual nature of agonism. I have already told about the brands. Now when I'm saying FDA approval, I'm talking about Drugs Controller General's Office approval. In India, when did they get approval? Rybelsus got in 22, Victoza 2010, Saxenda 2023, marketed by Novo Nordisk, and semaglutide got approval this year in 2025, both Ozempic and Wegovy from Novo Nordisk, while tirzepatide, only Mounjaro, because as I told you, the doses are almost similar. It's only Mounjaro which has been approved by the Indian government, and it is approved for both type 2 diabetes as well as chronic weight control. Huge global market, and the Indian market is heating up, right? There's an incremental, exponential rise in the sales and prescription of these drugs. This is the projected compounded annual growth rate. Now coming to the patent expiry, because the patent cliffs play a very important role in how generic versions are going to come out into the market and what kind of collaborations will happen with the innovator company and the Indian companies. So expiry window of semaglutide next year, 2026, so many companies in the run. It will stagger to 2031 because of the, the oral formulations. So the improvement with respect to the route of administrations, there are other secondary patents. So the patent expiry goes up to 2031. 80% of Indian companies are vying to enter the semaglutide market. Reddy, Cipla, Sun, name it, they are there. They are trying to develop their semaglutide. Now 505(b)(2) submissions are also there with some changes. in the route, in the formulation type. ANDA plans are underway because 2026, everybody is trying to be the day one generic to be launched in Indian market. Then the route, as I said, is subcutaneous. Liraglutide, earlier drug, already it has its, patent expired between 2024 and 2026. Secondary patents are also getting expiring next year. Companies like Biocon, Glenmark, they've come out with biosimilar versions. So biosimilars are either in the stage of launching or going to be launched. You will find liraglutide formulations which are biosimilars in the Indian market, costing much lesser than what the innovators' products are. Then coming to Rybelsus. Rybelsus also oral. People are, that is an extended patent which goes up to 2035. The drug company always, the innovator, the sponsor company tries to extend their monopoly rights over the product. So there are Indian companies who are trying to file 505(b)(2) in that for oral formulation of semaglutide as well, Macleoids being one of them. So you see 505(b)(2) with enhanced bioavailability and oral tablets daily. That will become more patient compliant and friendly because subcutaneous, still there are blocks for patient to take subcutaneous formulations. Now coming to Mounjaro. As I said, India has approved Mounjaro, right? There are companies like Biocon and Dr. Reddy's which are trying to partner, right? And already Cipla has partnered with Eli Lilly. I will come to that. These are all news between and happenings and updation between this year September, October and this month November. So till now tirzepatide no 505(b)(2), very well patent protected, and as we are aware that it is a subcutaneous formulation. So this slide sort of says that all Indian companies since the last 1 or 2 years and this year are running towards these 3 molecules so that they can also get a chunk of the pie, that is the market associated with diabetes. We saw how diabetes has, the prevalence is so high, and also obesity. And so who is under the focus? Semaglutide, tirzepatide and liraglutide, the 3 molecules. Although the main protagonist of this presentation is semaglutide, I would also pitch it with the other heroes in the fray, that is Mounjaro, tirzepatide and also a bit on liraglutide. So coming to the patent cliff which awaits, right, the molecule expiry timeline. So you can see the 100 billion plus patent cliff navigating GLP-1 expiry. Liraglutide, the first descent, okay, it is this next year almost all the patents would expire. So as I said, biosimilars are in the market. Then semaglutide, the main event next year, all companies are gearing up for it and we have to brace for it, right? Primary patent expiry 2026, but crucial method of use with respect to oral formulation still under patent and protected till the next decade. Tirzepatide, the next summit, and I'm sure, as I told you, Eli Lilly hitting the $1 trillion core group of exclusive companies, that just shows what the market is going to pan out to be, right? So tirzepatide is the next summit. The expiry of the patent is 2036 to 2039. So it has become so popular in the West. The other day I read that even in Walmart, the Walmart pharmacy is giving out like, retail Zepbound, and Zepbound is for obesity. So a person, a citizen of US can go and get Zepbound from the Walmart pharmacies. Earlier it was much more stringent through the other, the government associated, pharmacies and not from Walmart. So now this you can see already fake versions and counterfeit versions are there in the market and US FDA is warning about it. That just shows how it has hit. Now last month there were a lot of headlines, news headlines, and those were, for example, Times of India, fat loss drug Mounjaro, now India's #2 pharma brand. You can see here Economic Times, obesity lab, India becomes ground zero for weight loss, right? Eli Lilly's obesity drug, India sales hits a sweet spot. Now sweet spot not literally, not metaphorically, but with respect to the business, right? So it was launched in March this year, and in September it is second in line compared to Augmentin of GSK, which we know is an anti-infective combination. So Mounjaro sale only in the month of September was close to 80 crores, right? And the high unit price of Mounjaro between 14,000 to 27,000 per patient per month is a major driver of its value despite lesser volumes. Today, DCGI says we should look into biosimilars and biologics. Why? Because compared to the volume-based business, the government is looking that you go for value-based business, right? So just see the strategy of big pharma international companies. Eli Lilly has already tied up with Cipla, and it's going to make a tirzepatide brand called as Urpic. This is a very, very recent news in the same dose range, and the price is aligned with that of Mounjaro, right? But here the brand will be Urpic. Why? Eli Lilly's ground force is much lesser compared to Cipla's ground force and logistic, networks across India. So Eli Lilly is leveraging on Cipla's strong distribution across India. So the Lilly-Cipla collaboration underscores how weight loss therapies are becoming major strategic drivers in the Indian pharma landscape, bringing global innovation and local reach together. Earlier, innovation companies would restrict their own brands. This is a strategic partnership, which is win-win for both Eli Lilly as well as Cipla. And although Cipla is not the innovator, it is going to release its own brand along with Lilly into the Indian markets, and we expect it by next year, 2026. Another important development happened last month. US FDA has approved Rybelsus, okay, for, it has expanded its indication beyond type 2 diabetes to reducing risk of heart attack and stroke by lowering inflammation in people with or at risk of cardiovascular disease. So that means this has got an extended indication, right? And there were two trials conducted, very famous trials. The SOUL trial, which proved oral semaglutide reduced the major adverse cardiovascular event by 14%. The SELECT trial, which showed that injectable semaglutide reduced the major cardiac MACE by 20%. See Rybelsus. Why Rybelsus is better? Because it is an oral formulation. So compliance is better. It will be more accepted by the populace. So the Food and Drug Administration has approved oral FDAs, that is semaglutide, and now we have semaglutide, which is not only prescribed for type 2 diabetes, but also to prevent cardiovascular and abate cardiovascular risk, including in those who have not previously had a cardiovascular event. And Novo Nordisk has built a robust patent portfolio, as I told you, with respect to its gut absorption using caprylate, polymers and other delivery systems. Now, semaglutide landscape in India. The key player I told you, 80%, India is emerging as a semaglutide manufacturing as well as export hub aligned with the global metabolic therapy demand. So the innovator companies may be headquartered in the Western world, but they are planning to manufacture their products even in India, and maybe also have, as I told you about Cipla, have separate brands meant for only the Indian market. So it's growing, and the Indian anti-obesity drug market is growing. You can see the peak is rising. Now, semaglutide patent dispute between Novo Nordisk and Dr. Reddy's. This was also, coming up this year. So you can see it's in the Delhi High Court, and Novo Nordisk had filed a lawsuit against Dr. Reddy's Laboratories but you can see the Indian government's take. So Indian government, has a stance which was also there with several anti-cancer drugs, right? For example, Glivec, we have seen it in the past. So the patent evergreening and export exemptions, which comes under the BOLAR provisions, Indian government wants these drugs at reasonable prices, affordable prices, and more accessible. So Indian government is very pro the Indian companies coming out with their versions, and that will apply even in semaglutide, which is coming out in 2026, the generic versions. Now, GLP-1 agonists like semaglutide, or dual agonists like tirzepatide, or the triagonists which are in the developmental phase, will sweep the obesity and type 2 diabetes market. So things will change. The landscape is going to change in the future. So should we be vigilant? Absolutely yes. Why should we be vigilant about it? So if you see the package label, the labels of these products, right, the boxed warnings for both cases, Ozempic and Mounjaro, is thyroid C tumors, but these are more found during the preclinical studies in rodents, right? Not very rare or not encountered in humans as yet. There are common side effects associated with GI tract, other warnings. So while both semaglutide and tirzepatide offer significant metabolic benefits, their prescribing information emphasizes vigilant monitoring, especially for gastrointestinal, thyroid, and volume status risks. Hemodynamic kind of imbalances. So another subpart of my talk is to flex or not to flex. Flex the muscle or not to flex the muscle. The dichotomy of evidence on semaglutide. There are contrary evidences. One very important evidence is that this was a retrospective study, 2 cohorts. The cohort where, the elderly diabetic patients were exposed to semaglutide, that is Ozempic, for 24 months, that is 2 years. There was presence of sarcopenia up to 27.7%, which is a significant, percentage. So we should look into, when the Indian citizens are going to expose to the drug, we should have very robust pharmacovigilance because millions would be taking these drugs. Already lakhs must be already taking the drug. We need to follow up and have a pharmacovigilance process in place. Molecular basis for semaglutide acting on thyroid changes because it is acting on the cyclic AMP, PK signaling pathway, and it's known to act on the TSH suppression, and it can cause C cell proliferation, the thyroid cells, which are known to release calcitonin. Now coming to muscle, semaglutide improves glucose uptake in skeletal muscle by upregulating GLUT4 activity, and weight loss can cause relative lean mass reduction, although functional muscle strength is usually preserved. It's acting via the AMPK pathway, and it is having a catabolic phase during rapid weight loss. So this is the impact. Further, it has been seen that GLP-1 agonists, they cause effective weight loss, but also 25 to 39%. They are causing fat-free mass reduction, which is worrisome, right? Over 36 to 72 week period. The magnitude of total weight loss rather than the direct drug effect likely driven by its muscle loss. By comparison, non-pharmacological diets cause only 10 to 30% of FFM loss. Age-related muscle loss, lean body mass loss, is hardly 0.8% per year. So GLP-1 induced loss is several-fold higher. Now here also you can see a dichotomy, a paradox in the evidences which are coming forth. Some are preclinical evidences, some are clinical evidences. Quickly, semaglutide reduces cardiomyocyte size and cardiac mass in lean and obese mice. GLP receptor agonists, attenuates chronic liver disease-induced skeletal muscle atrophy in diabetic mice. Oral semaglutide loss, induces loss of body fat mass without affecting muscle mass in patients. It's a clinical study. This is something which I was talking about. Chronic semaglutide treatment in rats leads to daily excessive concentration-dependent sucrose intake. A case report of semaglutide-induced sarcopenia causes fatigue in older adults, right? So a lot of dichotomy in the evidences which is coming forth. So the negative evidences, emerging safety concerns, sarcopenia, and cardiac atrophy. These are papers which are associated with preclinical on rats and on heart muscle in mice and human cells. So here you can see that it suggests a systemic muscle remodeling effect of semaglutide, remodeling, restructuring, what we call as re-engineering, influencing both the skeletal and cardiac muscle system. This report, which is a clinical report, it's a case study which says a 74-year-old male, 2 years treatment, semaglutide has accelerated sarcopenia and emphasizes the need for baseline muscle evaluation and rehabilitation support. In the earlier slide also I mentioned that there needs to be protein supplementation, weight training, and those kind of things to build the muscle. Positive cardiac outcomes, semaglutide in heart failure with preserved ejection fraction with obesity.
Sorry to interrupt you, Dr. Anuradha.
5 minutes?
Okay, please.
Yeah, I can see 5 minutes. I'll wrap up in 5 minutes. Semaglutide in patients with heart failure with preserved, I said this is a beneficial action, which is a clinical study which has been depicted in a paper. This is the same study showcasing the results, right? So this is a very positive result showing, and that also probably resonates with the US FDA approval for its protection for cardiovascular events, right? It is preserving lean mass is what another study says. Okay, this is also on patients. It achieved fat specific weight loss, sparing skeletal muscle and prevents sarcopenia, right? So again, a contrary, but these are all clinical studies. The others were more towards preclinical and clinical in the elderly or sporadic case studies. So this is a summary of the positives and the negatives. So here you can see the positive preclinical study, it attenuates liver disease which is induced by muscle atrophy, and it is good. Mechanistic insight is via the SIRT1 PGC1 alpha pathway, so it must be acting on the mitochondria, right? Clinical trials, it causes loss of lean, significant fat mass reduction without loss of lean muscle mass. While the negative cautionary findings is prolonged therapy in older adults associated with accelerated sarcopenia and reduced functional capacity, report of fatigue, this is clinical in older patients, fatigue, weakness, muscle loss improving after dose reductions and resistance exercises. So how do I summarize the semaglutide saga? Dual nature of semaglutide, so it demonstrates fat selective weight loss, metabolic improvements, and there are also evidences to show risk of sarcopenia is reduced, functional decline, the evidence is that risk of sarcopenia is enhanced and functional decline in older or long-term users. So there are preclinical and control trial evidences, real world data which highlights fatigue and weakness. So it's very important to understand that a balanced use requires careful monitoring, proper nutrition, resistance exercises to maximize the benefits while minimizing the muscle loss. To flex or not to flex, impact on muscle morphology and physiology, and there is a need for more evidence when it's in the real world settings in India, which starts from next year, and even the the innovator products are already in the market. This is the future coming soon. Orforglipron, this is also from Lilly. You can see here the Pfizer products have not continued. This is in phase 3 and successfully demonstrated efficacy with respect to oral semaglutide. And this is the future, right? These are not polypeptides. These are small molecules. So it is representing a major shift in peptide to small molecule innovation, enabling easier manufacturing, lower costs, and broader scalability for global obesity and diabetes treatment. So things, there's a lot of research going on and innovation in this sector, and we'll soon see non-peptidic small molecule products in the market in the future once it is approved by US FDA and our DCGI. So these are some of the references for the benefits and the risks. I would like to acknowledge some people who were part of my journey almost for 27 years. So I extend my heartfelt gratitude to the esteemed Indian Pharmacological Society office bearers and the jury of Dr. S.B. Pandey Oration Award. Thank you very much. I'm humbled, and it's like it has been such an honor and treat for me to come here and present in such a honorable esteemed setting and august presence of you all members. I would like to thank my guides. Dr. C.T. Chopre, I did my master's from him. He was my first guide who made a dramatic change in my approach to research from Nagpur. Dr. Emin Saraf, who has been my master's guide. Dr. Professor Kulkarni, he is there. He has influenced me in all phases when I was in Nagpur, when I was in Mumbai, in all phases. Thank you very much, sir, for all your support and mentoring. I would like to thank Professor Anil Kumar for inviting me here. My mentors, seniors, colleagues who constantly they supported, their wisdom, encouragement has shaped my growth as a researcher. I also acknowledge my students. They have been the main strength. They have been the reason that we always feel young and happy in our ecosystem of the campuses, and the support they provide for everything, including fine-tuning the presentation. I thank all my students in the past and the current, and whose curiosity, dedication, and enthusiasm continue to inspire me even today. Very important, I was born at a time when I have seen and witnessed the quiet and the content of the 1980s and 1990s. And after 2000, I saw Google, I saw the, the boom. And thank you very much for having that, this as well. The achievement is a reflection of my collective, guidance of people. Thank you very much. Thank you. This is in one of the events in my lab. Some of my research students and some of the faculty are also there. Thank you very much.
Thank you, Dr. Anuradha, for your wonderful presentation, throwing the insight into the potential of semaglutides and their trivial issue regarding to their efficacy and safety with respect to side effects, whatever they are to be encountered in the coming time, but also showing the potential market for the drug, as there is a lot of epidemic, especially with respect to India, the epidemic of obesity and diabetes. So it's a wonderful presentation. Thank you once again. So as it's a oration, I think rather than having questions, we can have some suggestions and comments if required, if acceptable.
Yeah, we can have a quick comment maybe.
Please, sir.
So the evidences which has come in, sir, is in 2 years of regular intake of the drug. So probably they will have to, because if you go to the Uppsala Monitoring Center meta-analysis and this, nothing is there. I had to literally dig for the information because these are high value products in the market. So UMC, I tried to get some data. COMS, I tried to get some data. So probably when it comes to India, like Madam is from PGI Chandigarh, probably, and from other hospitals, they need to have some kind of focus on GLP-1 and the dual agonists in Indian population.
Because there are some reports when you stop taking the drugs, again you obesity will come back.
Yeah.
And that's one issue. Other issue is about the cost effectiveness. And recently even Donald Trump has allowed giving $1,000 to those who are buying these drugs as a safety money to buy these drugs. They are very expensive.
Yes, very expensive. But liraglutide, once we have the generic version, sir, I think that cost will be much, much reasonable. But whether it is Mounjaro or Ozempic, the costs are formidable. 2026, semaglutide also, I think the costs are going to come down.
Thank you, sir. Thank you, Dr. Anuradha, for such an erudite presentation. And thank you, sir, for bringing up that very, very important comment, because I would like to inform the gathering here that we have evidence from the clinical trials which have already been conducted that the moment you stop the drug, the weight starts creeping up. And if you see the number needed to treat and number needed to harm, I think the balance would shift towards it's very interesting that you pointed out that the drug is being launched rather than approved. That's how it has become so market-driven. So it's very important that we understand all the developments in the market area also, which is so beautifully brought out. But having said that, I think it's very important to keep in mind all the important points that sir raised in the end. And with this, I think we can conclude this session. And thank you very much.
Thank you.
Thank you, ma'am. I request our session chairs to please felicitate our orator with a token of appreciation. Thank you. Our next oration, the Dr. Lalita Kameshwaran oration, holds a special place in the history of Indian pharmacology. Instituted to commemorate the visionary leadership and profound academic contributions of Dr. Lalita, this distinguished honor recognizes scientists whose work has driven transformative progress in pharmacology, therapeutics, and biomedical innovation. Upholding Dr. Kameshwaran's legacy, this oration occupies an esteemed place in Indian pharmacological academia, promoting rigorous scientific developments and fostering a culture of scholarly excellence. Chairing this commemorative oration are two eminent personalities who exemplify academic leadership and mentorship. First, we have Professor Babul Kumar Bezbura, President, Indian Pharmacological Society, and I don't think he needs an introduction at this moment. Sir has graciously chaired one or two sessions before as well, and now joining him today in chairing this session is Dr. Yogesh Kulkarni, who is Professor and Associate Dean at School of Pharmacy and Technology Management, NMIMS University, Mumbai. His work spans pharmacognosy, pharmacology, natural products, and herbal drug research with a strong focus on translational drug discovery. Dr. Kulkarni has contributed substantially to curriculum development and academic administration and continues to be a guiding force in pharmaceutical sciences across India. I request my esteemed chairs to formally preside over the session and introduce our speaker, Professor Mohammad Ishaq Gir. I request Professor Gir to kindly come on the stage.
Very good morning. Dr. Mohammad Ishaq Gir, who is a professor, Department of Pharmaceutical Sciences and Directorate of Lifelong Learning, University of Kashmir, Srinagar, Jammu and Kashmir. Professor Gir Mohammad Ishaq is Professor of Pharmacology, Department of Pharmaceutical Sciences, University of Kashmir, with over 23 years of academic and research experience. He also serves as Directorate of Lifelong Learning, and has held key administrative positions including Director, Center for Career Planning and Counseling. A former drug control officer, he earned his M.Pharm from RGUHS Karnataka and PhD from the University of Kashmir. With over 50 research papers, 170 invited talks, and multiple best paper awards, he contributes extensively to pharmacovigilance, pharmacy practice, research governance, and policy implementation in healthcare and higher education. I request Dr. Gir, please start.
Thank you very much. A very good morning to all of you. First of all, I would like to thank the organizers for giving me this opportunity to present this oration, Dr. Lalita Kameshwaram oration, which is a matter of great privilege and honor indeed. And equally more honorable that I'm speaking in front of legendary professors, pioneers in pharmacology like Professor Kulkarni, Professor Goel and others, and Professor Diwan. So actually my research interests focus around pharmacovigilance, clinical pharmacology, drug utilization evaluation. But instead of talking about my own areas of research and my journey, I thought it will be more prudent to talk about the theme of this particular conference, that is empowering young minds to transform pharmacology for Viksit Bharat 2047. So as already has been emphasized by Professor Y.K. Gupta yesterday, that we need to be futuristic, we need to be contemporary, and we need to be relevant, and we need to look forwards, and we need to gain knowledge about new technologies emerging. So I thought I'll be talking about the transformation, how we could transform pharmacology into 4.0. Nowadays there is a lot of buzz about industry 4.0, there is healthcare 4.0. So what about pharmacology 4.0? How the pharmacology could emerge and evolve with the passage of time, And with the changing technologies, nowadays AI and ML is taking over the world in its stride. So I thought, let me discuss and see in front of all the pioneers and students and scholars what best we can do in pharmacology and how we could contribute towards Viksit Bharat 2047. So to begin with, when we talk about transforming young minds, I don't see any other leader as towering a personality as Swami Vivekananda ji, who said, take up one idea, make that one idea your life. Think of it, dream of it, live on that idea. Let the brain, muscles, nerves, every part of your body be full of that idea. And this is the way to success. So he said, my faith is in the younger generation, the modern generation. They will work out the whole problem like lions. So I thought I'll just present an idea which struck my mind. That I didn't see any other speakers talking about digital twins. And I believe this may be the first time I may be speaking in this on this forum about digital twins. So I thought, let me give that idea to the society and let us see how it will emerge and evolve and how it'll be able to transform the paradigm of pharmacology. And then our Honorable Prime Minister, Shri Narendra Modi ji, also has said that in order to achieve the dream of our Viksit Bharat, we are transitioning from Azadi Ka Amrit Mahotsav after completing 75 years of independence 3 years back. We are now in the Azadi Ka Amrit Kal, wherein we have to do Vimarsh. And in that Vimarsh, he said, this is the period in the history of India when the country is going to take a quantum leap. For India, this is the time, right time. Today, the aim of your goals, your resolutions should be only one, developed India. And idea starts with an I, just like India begins with an I, development efforts begin from self. So we need to introspect, look inwards, and see what best we can do to contribute towards this goal of Viksit Bharat, wherein we need to impart skill-oriented education, innovative research and development, innovation, incubation, startup, and entrepreneurship development in pharmaceutical sciences. So we need to think on these lines in order to develop our pharmacology, develop our pharmaceutical sciences, which could in turn contribute towards the goal of Viksit Bharat. And so the idea which I am presenting today is a 360-degree transformation, paradigm shift in the pharmacology. Already we are undertaking a lot of in silico testing. We are doing target and lead identification, their validation, their optimization in silico. Then we are doing molecular docking, we are doing pharmacophore modeling, molecular modeling, and so much more through computer-aided drug design. So already that much has been gained. We are doing it at a considerable level in our country. But what have you thought about when will we be able to transform the preclinical studies entirely into digital format and also the clinical trials completely into digital format? That is when we will be able to achieve, India has been the leader in the pharmaceutical sciences, the pharmacy of the South, pharmacy of the world. We have provided the lead in COVID-19. We have supplied vaccines to the whole world. We have supplied generics to the whole world. But now, in order to maintain that lead in future, we need to present a new innovative model, an out-of-box solution in pharmacology also, so that we will become leaders in CROs, in clinical research. Already in our country, there are more than 300 CROs, and this market of CROs is 2.5 billion. But we have to reach to $10 billion in clinical research. How that will become possible? How India could be the leader in clinical research? That will become possible only when we completely digitally transform our preclinical studies as well as the clinical trials, as well as the in vitro and in vivo. See, as of now, when you complete your lead identification target optimization, you have to go to the in vivo and in vitro studies on animal cell lines, etc. But when we will be able to do it in a completely digital mode, that is a big question. How that can be achieved and how that will become possible. That will become possible through big data analytics, through use of digital twins in clinical trials, as well as in preclinical. So that is the idea of Pharmacology 4.0, which is emerging as a conceptual framework that represents the next generation of pharmacological science, which integrates advanced technologies like big data analytics, system-level understanding to revolutionize how drugs are discovered, developed, and used. and that parallels with Industry 4.0 and Healthcare 4.0 paradigms, and is a data-driven, AI-enabled, system-integrated approach to pharmacological research, education, and practice, which leverages multi-omics, digital twins, real-world data, and automation for personalized and predictive medicine. Now, when we say pharmacology 4.0, then there must also be pharmacology 1.0, 2.0, 3.0. We haven't heard of it earlier. It hasn't been discussed elsewhere. So how do we segmentize it? How do we compartmentalize it? Like if we look at it, that pharmacology 4.0, pharmacology 1.0, we can trace it. There is no strict classification available anywhere, but I could think of it on these lines. So there may be agreements or disagreements on this, that pharmacology 1 was the era which was descriptive era, which was full of trial and error. Pre-19th century to early 20th century. There was empirical and observation-based drug use guided by trial and error. Focus was on natural products and traditional medicine, but there were no mechanistic understanding of drug action, herbalism, toxicology, and crude drug studies. Then came the pharmacology 2.0, which was a mechanistic and molecular era, from mid-20th century to late 20th century, which showed the emergence of receptor theory, biochemical mechanisms, development of synthetic drugs, understanding dose-response relationship. And then came the pharmacology 3.0, the translational and precision medicine era, the Omics era, early 21st century, which witnessed integration of genomics, proteomics, metabolomics, transcriptomics, and a shift towards personalized and precision medicine. And now we are in the pharmacology 4.0, which is intelligent, interconnected, predictive, human-centric, and autonomous pharmacology. And from 2025 onwards, we can make progress in this direction by leveraging integration of AI, machine learning, digital twins, big data analytics, real-world data collection, human-centric design, IoT, etc. These 21st century technology. Now, why should we do that? Are we not happy and satisfied with the current things which we have at hand? Preclinical studies on animals, clinical trials on humans, giving us real-life data. Why should we think in these directions? Is it really important? Yes, it is. If we go by the words spoken by the Nobel laureate of physics of 2024, who is also known as the godfather of artificial intelligence, Geoffrey Hinton. He has said schools and universities will become obsolete in another 10 years. We won't need them. These are the words which he has quoted in an interview to CBS TV channel in 60 Minutes program, which you can Google out and search for yourself. And no, there may be disagreements. This is debatable. Many people say the technology will never replace the teachers. And I also believe on it to some extent, but when the words are coming from Nobel laureates, the Nobel laureate in chemistry, Demis Hassabis, who is a UK-based computer scientist, Nobel laureate in 2024, he also predicts that there will be an end to all disease within 10 years. There will be an end to all, because of these technologies. And even Bill Gates has said that AI will replace doctors and teachers within 10 years as we enter the era of free intelligence. See, this time we are in the era of generative artificial intelligence, but then we will be switching over to artificial general intelligence. And then will come the stage of artificial super intelligence. So we are already in that queue. We are already marching in that direction. Nothing can stop it. Whether we like it or not, whether we adopt it or not, it is going to happen. Now that we know that it is going to happen and nothing can stop it, why not align with it? Why not synchronize with it? And why not prepare ourselves for it? So that is the whole idea. That is, we need these systems pharmacology approach, systems biology approach, quantitative systems pharmacology approach. We need to integrate all these technologies so that we can develop a biosignature of the human being, which will enable us to develop the digital twins. That is integration and application of omics. We know that genomics, we do the gene mapping. It gives rise, DNA gives rise to RNA. Full RNA picture comes out through transcriptomics. The picture of all the proteins in the body is profiled through proteomics. Then the whole mapping of all the metabolites is done through metabolomics. When we apply it to the drug discovery and design, we call it pharmacometabolomics. And then there is influence of environment. You know, we don't only look at the genotype, we also have to look at the phenotype. So the environmental influences on the genotype, the exposomics, exposures, environmental exposures, and the epigenetics together influence the genotype. And then all of these together, integromics. So our students need to learn these technologies. They need to know about these technologies, how they are going to affect and impact the new drug design and development. But that is not all. Only omics technologies is not all. We also need to learn and integrate the informatics-based technologies, because this will help us know the human body completely. See, so far we have been discussing characterization of natural products in plants, how to isolate them, how to purify them, how to characterize them. Now is the time to characterize the human being. Now is the time to characterize the various components, the elements, the molecules inside the body of a human being, their interaction, their networking, their combination. And that will become possible through these technologies. That is genoinformatics, which deals with genetic variation and mutation data, bioinformatics, target discovery, pathway analysis, and biomarkers, immunoinformatics, the immune response modeling and epitopes, chemoinformatics, molecular design, screening, QSAR, toxicoinformatics, the toxicity prediction and safety profiling, pharmacoinformatics, the ADMET, PKPD, and drug interaction. So when we know all of these about a particular human being, one person, we know his full toxicity profile, drug metabolic profile, we know his genetic profile, this immune profile, it will become easy for us to develop a digital twin of that person. And then when we combine it with the quantum systems pharmacology, quantitative systems pharmacology, we'll be in a position to have a real-life prediction for high-fidelity clinical twins, which can generate real-time data about that particular individual and which can be used for clinical trials. Now, so far as the preclinical trials are concerned, already this new approach methodologies, NAMs, the in silico techniques are being used in preclinical pipelines. But now they will be replaced. The physiologically based PKPD models and QSP models will replace early animal PK studies. The AI predictive toxicology models will replace animal toxicity screening. The organ-on-chip technology, digital simulation will replace organ toxicity studies. The omics plus machine learning will predict efficacy and biomarker discovery. And virtual control arms will reduce patient enrollment. So we'll have virtual control arms, means the control group will be entirely made up of digital twins. There may not be use of any human beings at all. Digital twins which are not necessarily the counterparts of physical human beings. They are, there are two types of digital twins. One is individual digital twin, which is a counterpart, a copy of a physical being, which needs a copy, real life physical being, but population twins, which is generated from the data, population-based data, and which do not need any physical counterparts. So systems biology approach, as I was mentioning, a thorough characterization of organism concerning its molecular components, interactions, and how these interactions contribute to cellular function. This approach will be utilized for quantitative systems pharmacology, which is the application of systems biology principles to the field of pharmacology and seeks to understand how drugs affect the human body as a single complex biological system. So this will check the networks of interactions, which may include chemical protein, protein-protein, genetic, signaling, and physiological interactions. And this systems pharmacology will use bioinformatics and statistics techniques to integrate and interpret these networks. So that is what will lead us to Pharmacology 4.0, which will be a quantum leap in drug development research, a historical inflection point in drug sciences. Integration of all these techniques moves pharmacology, will be moving from empirical experimentation to predictive simulation. And this will bridge molecular mechanisms with data-driven precision techniques. So I'll just jump to because of the paucity of time, I have to finish all the slides now. Looking at the digital twins, what exactly are digital twins? Digital twins are virtually replicas of physical devices that IT professionals and data scientists can use to run simulations before actual devices are built and deployed. A digital twin is a digital representation of a physical object or a system. In essence, it is a computer program that takes real-world data about a physical object and produces output predictions or simulations of how that physical object or system will be affected by those inputs. how actually this digital twin concept has emerged. It has been used way back in 1960 by NASA. And they have, in order to simulate the spacecraft environment, and in order to be able to debug the flight issues in real time, they came up with this digital twin concept of the, when these astronauts go to the outer space. So they build those digital twins of the same environment on the Earth and tested every aspect of it. And the concept was successfully utilized during Apollo 13 mission when the spacecraft suffered a malfunction and the NASA team had to simulate the conditions aboard Apollo 13. So early 2000s, this was discussed, then mid to 2010, 2017 to 19, 20 to 20, and 23 to present. There have been series of changes and series of breakthroughs in this digital twin technology. I'll not go into the details of it, but the benefits of digital technology are that it will provide us personalized medicine, predictive analysis, research and trials, improved patient engagement, healthcare efficiency. In the sense that, there will be personalization will become possible by tailoring of treatment plans to individual physiology and genetics, integrating of data from electronic health records, genomics and wearables, and will enable real-time adjustments based on patient response. Then predictive diagnosis will detect early signs of disease progression, will identify risk factors using historical real-time data, will support preventive care and timely interventions. It will be cost-effective. There are a lot of concerns about cost of digital twins, but this will be cost-effective delivery because it will reduce unnecessary procedures and hospitalizations. It will optimize resource allocation. It will improve efficiency in clinical workflows and will simulate treatment outcomes. Then accelerated drug innovation, accelerated drug design and development will also become possible. So it will have cost and time efficiency. It will improve patient safety. It will enhance data collection, analytics, and insights. And there will be a lot of flexibility in scenario testing. Now, can there be digital twins of only human beings? No. Digital twins are possible for each organ, for each cell, for each tissue. And at tissue level, cellular level, organ level, or the human level, and even for the whole hospital, there could be a digital twin for the whole system. So there are patient-specific digital twins which are individualized at personalized level, which are virtual replica of an individual. Then there are organ-level digital twins, which are virtual models of, you have heard about lung on the chip. So organ on the chip technology, specific digital twins of heart, liver, kidney, etc. Then we can also have digital twins of tissues. So we can have modeling of the disease, modeling of the organs, modeling of the whole human being through these digital twins. That will become possible. A person can know from his own digital twin whether this drug will be effective or not, what kind of adverse reaction based on the data which it has generated from lakhs of studies. Machine learning through databases available already, it will generate data and give him that this drug could, it knows the biology, it knows the physiology as well as the genomics of the person. It will tell him this drug could show this side effect in you, or this may or may not be effective. So, of course, initially it may be costly, it may not be affordable, it may not be accessible, but with the passage of time, as the technology will evolve, the issues related to affordability, accessibility will be resolved. And then there are some terms which are very frequently used with respect to the digital twins. For example, the digital twin fidelity, which largely depends on the real-time data generation, real-world information. The digital twin instance, which describes the physical twin during entire lifespan. When a person is connected to the digital twin throughout his lifespan, and in real-time basis, it is generating and accumulating data from the person. And DT aggregate is the aggregation of some of these DT instances and aggregates, while as DT bank, digital twin bank, is an organized repository of instances as well as aggregates, and a digital twin thread, on the other hand, is a temporal data pipeline right from the birth of a person to the death of a person, which tracks the relation of all the elements over a period of time. So there may be static twins, which are mirror twins, which may not be able to generate real-time data, but there are also functional twins. Functional twins generate real-time data over a period of time. They are constantly linked through biosensors, IoT-based sensors, and through other networks, which will generate real-time data. And there are shadow twins, which are digital twins for medical devices, for biomarker and design, drug discovery, biomanufacturing. and then intelligent twins, which is the most evolved version of the DT, which is a self-adaptive twin with a degree of artificial intelligence. So the difference between individual twin and population digital twin, individual digital twin is connected. It is a mirror twin which mirrors one real person in life. So it needs a physical counterpart, needs real-time sensing, and is used for personalized medicine. On the other hand, the population digital twins do not need physical counterparts. They are not real, these real human beings are not required. They will be built from databases through machine learning and models, and they'll be used for drug development simulations. And how population twins will be constructed? They will be constructed through big biological databases, genomics-based data, physiological PK/PD-based and QSP-based data, AI-generated variability data, and which will be validated using historical clinical outcomes. So, in order to have the digital twins of animals, the laboratory animals, we need to collect biological, physiological data of the animal. Then we need to build a physiologically based PK/PD and QSP mechanistic model. We need to add disease-specific modules. We need to integrate PK/PD. We need to train AI to replicate experimental variability. We need to add virtual sensors, validate using real lab animal data, and then we need to use for simulations, toxicity. So for that, we will have to apply these neural networks based on models. I'm not a computer wizard. I'm not an IT expert. I'm not even an expert in digital twins. But we need to learn and know more about these technologies so that we are in a position to leverage them. Now, what are the challenges? There are concerns related to these digital twins. And there may be definitely new technology. Every new technology has certain challenges. The first and foremost challenge will be that of data privacy and security. When we implement these cybersecurity measures, data encryption and patient consent procedures will need to be incorporated. So data standardization and integration is another challenge. We need to develop universal health data standards and systems to integrate and interpret various databases. The quality of data, see, when we test on actual human beings, the quality of data is good. But when we test on digital twins, what will be the quality of data? Will it be equally good as that on humans? That is a big challenge. For that, we need to conduct regular audits, maintain data quality checks, and use reliable data collection sources. Then there are computational challenges. Every institute, every country, every setup may not be having all the infrastructure. They need to invest in high-capacity servers. They need to invest in cloud computing, edge computing, leverage AI and ML learning. And then there are regulatory concerns. No, they need to be approved by the FDA. So then only we can apply them for clinical trials. So we need to collaborate with regulatory bodies for guidelines and standards. We need to conduct pilot projects and clinical trials. And lastly, there is the challenge of adoption and trust. Can we trust those digital twins? Can we trust these technologies? Privacy, the confidentiality, the quality of the data generated, all these are the issues related to trust. There are a few more challenges and limitations. I will not go into further details of this. But then there are ethical considerations. Ethical considerations while using digital twins, like patient consent and autonomy. So when a patient's digital twin is available, anybody can do anything with that digital twin. So there are issues related to infringement of privacy, autonomy, and patient must understand how their data will be used. And then there are algorithmic biases and fairness. So bias of the algorithm has to be eliminated. The data privacy and security has to be ensured, and fairness audits have to be conducted. These digital twins can rely on sensitive healthcare data. So I will skip some of the slides. Then there are the implications on academia. What are the implications on the universities? The academia institution like this one, what do they need to do for preparing for this? Universities must define pharmacology education and research ecosystem to align with digital transformation, first of all. Then they need to integrate AI, data science, and omics into curricula. They need to establish interdisciplinary centers for computational pharmacology. They need to invest in digital twin laboratories and simulation-based learning. They need to train faculty in AI ethics, in data governance, and system modeling. And then Pharmacology 4 will also democratize research by linking universities globally through open data clouds and collaborative platforms. So these are some of the strategic implications for academics. And then we can slowly, even there will be Pharmacology 5.0. What will be there in Pharmacology 5.0? There will be cognitive AI. There will be empathy algorithms. There will be neurosymbolic intelligence. That is when the machine not only collects or predicts, but also understands. What do you mean by understand? When the machine balances efficacy with empathy. So that is the next level, which of course the computers or artificial intelligence has not achieved as yet. But the vision of the pharmacological civilization, so Vision 2047, coming back to Viksit Bharat, if we want to see, we have ample time. We are not going to do it overnight, in a say, or 1 month, or 1 year, or 10 years. We have time till 2047. So this time we may be jittery, we may be apprehensive about these technologies, we may not be able to digest it, but we have time till 2047 when we can leverage it and we can accumulate, adopt it. So these are some of the DT health research centers around the world. It is not that anything is not being done. Already many companies are working on it, but these are some of the instances of the companies, the community, the emphasis and the aim. They are already working on the digital twins in healthcare. A lot of solutions have already emerged. And the lung, the heart, the spine, they are used in Alzheimer's disease. They are used in breast lesions for research purpose. Then oropharyngeal cancer, type 2, diabetes, mental health conditions, and so many. So these are the number of papers which have been published on digital twins. As you can see, it started somewhere in 2017, and this time, 2023, we have 321 publications. So these are the applications of these digital twins in a whole gamut of areas, not only clinical trials, but also personalized medicine, surgical planning, device design, and a lot more. So as I already mentioned, there are more than 300 CROs, and the 2.5 billion is the current market.
Dr. Gill.
Yeah, I'm concluding, sir. Just a couple of slides more. I'll finish within my time. So projected is $8 to $10 billion. And this is the, if we want to tap this market, already Government of India is providing opportunities like PRIP. The PRIP has a funding of around 5,000 crores. So Government of India is aware of the things which we need to be done, up to 100 crores per project under PRIP. So if there is a funding of 5,000 crores available, which is our government is making, why can't we think on these terms? Yesterday we had a presentation by Dr. Shiv Prakash. He said there is an opportunity for starting a startup in this AI ML. So many people, youngsters who are empowered and who are motivated enough, they can present and make their startups on these lines. They can start from the basic level. They don't need to go too high on this paradigm. But once we start thinking and moving in this direction, then only we'll make some progress. That is the takeaway. I thank you all for your patient audience, and let us take a pledge to further Atmanirbhar Bharat and Viksit Bharat by the year 2047. Thank you very much.
Thank you, Dr. Gill, for finishing in time. I'll not ask you any question. Just one question will be thrown to Kulkarni sir, because you have come across all the, from pharmacology 1.0 to pharmacology 4.0. So what are the differences you are observing, and what will be the future of pharmacology?
No, I have 2-3 general observations to make on the, exhaustive presentation by Dr. Geyer. Number one, too much material on the slide to follow it up was very difficult, and you are also running through your text material there. So it is always better to have little material on the slide, and we can explain this one. The other one is you are talking about Viksit Bharat 1947 as envisioned by our Honorable Prime Minister. In order to become Viksit Bharat, India has to increase its gross educational
enrollment ratio,
gross enrollment ratio from, it is now about 10 or 12, it has to go to 50.
50, yes.
And it needs a lot of other requirements. So I don't know, you asked me what would be the future of pharmacology. The pharmacology will always depend on DRC, dose response relationship. Whatever use AI or anything else, this is the crux of the discipline. Whether it is advanced therapy or old therapy, he was talking about 19th century, 20th century, 21st century. All these centuries, pharmacology is developed on dose response curve. That will remain for all times to come.
Thank you, sir. To sum up your presentation, so Pharmacology 4.0 for Vikshit Bharat, which blends workforce modernization, digital data ecosystem, then regulatory reform, advanced manufacturing, and strong public health stewardship. And that should be implemented phase-wise, and multi-stakeholder governance and measurable KPIs, key performance indicators. India can build a resilient, innovative, and equitable medicine ecosystem that improves outcomes and secures health sovereignty. Thank you, Dr. Gill,
Thank you,
for your
sir.
excellent presentation.
Thank
God
you
bless.
very much.
We now invite our session chairs to perform the honor of presenting a token of appreciation to our speaker, Dr. Gir. The success of this session rests on the shoulders of our reverend session chairs. A big round of applause. Now, I request IPS advisors Dr. Diwan and Dr. Shiv Kumar to honor our chairpersons with a sincere token of our gratitude. So our next session for day is Symposia on Bridging Discovery and Clinical Application in Regenerative Science. This session highlights cutting-edge advancements in regenerative medicine, from laboratory innovation to translation into clinical practice. And to lead this prestigious session, I have the distinct honor of inviting our session chairs, Dr. Anuradha Majumdar and Dr. Manoj K. Aswar. So I request them to kindly come on the dais and officially commence the proceedings. And to recognize their eminence, I would like to take a moment to highlight the notable achievements of our esteemed chairperson. And since Dr. Anuradha Majumdar needs no introduction, as she has been the orator of our previous session, we have
Dr. Manoj Kashinath Aswar, Professor of Pharmacology at Marathawad Mitra Mandalas College of Pharmacy, Pune. With over 20 years of teaching experience, he specializes in pharmacology, neuropharmacology, and preclinical research. He has published 30 papers, presented 35 conference papers, guided 18 MPharm students, and authored the textbook Pharmacology 3rd, Nirali Prakashan 2019. Recipient of the Professor P.C. Dandia Award, Dr. Aswar's work bridges academic pharmacology with translational research. He is deeply committed to mentoring students and fostering innovation, contributing significantly to the advancement of pharmacological education and research. So, with these words, I welcome our session chairs, and I request them to introduce the speakers of this symposia.
I'm honored to welcome our first speaker, Professor Buddhadev Dawn, MD. He's the chairman of the Department of Internal Medicine and chief of the Division of Cardiovascular Medicine at the Kerkorian School of Medicine at the University of Nevada, Las Vegas. Prior to UNLV, Dr. Dawn served as the director of the Division of Cardiovascular Diseases at the University of Kansas Medical Center, where he was the Maureen Marvin Dunn Professor of Cardiology, founding director of the Cardiovascular Research Institute, and the founding director of the Midwest Stem Cell Therapy Center, and vice chairman for research in the Department of Medicine. He received his medical degree from Calcutta Medical College, India, and underwent cardiology training at the same institution. In the USA, he completed residency in medicine at the University of Missouri, Columbia, and completed fellowship in cardiology at the University of Louisville. He is a practicing clinical cardiologist and physician scientist with research interests focused primarily on heart repair with adult stem cells. He has authored over 200 articles and book chapters. He serves on editorial boards of several prominent cardiovascular journals, national scientific committees, and grant review panels. He also played a key role in a successful phase 1 clinical trial using the Wharton's jelly-derived MSCs in patients with graft versus host disease. His clinical interest includes echocardiography and atrial fibrillation. Over to you, Dr. Dawn.
Good morning. Thank you, Dr. Majumdar, and the co-chair, Dr. Aswar. So thank you for inviting me to this wonderful city of Chandigarh and the Punjab University, my first time here. I was in, the last time I was in Chandigarh was in 1991, a very long time ago, and I think the city has changed a lot since then. So it's really a pleasure to be here and be able to participate in this meeting. So I thought my talk time was 30 minutes, but I'll try to go over faster. And I do have, this is not a conflict, but I am a co-founder of a company that is mostly for neurological ALS treatment. It's a stem cell that I'm not going to discuss those data. So there are two types of stem cells, embryonic stem cells and adult stem cells. So these cells were described in the 90s, and these can be obtained from humans. However, these are very hard to differentiate into different specific lineages, and these are truly pluripotent cells. So if even one cell is pluripotent in the injected that is going into human heart, then the patient will get a tumor. So there has not been much advance to the human clinical trials of embryonic stem cells. There has been a lot of hype, but no real advance. Adult stem cells, on the other hand, are the cells that are found in many different organs, including the skin, the hair follicles, in the GI tract, and they can be harvested from different human tissues. Almost any tissue that has been looked into has stem cells, including the cardiac myocardium. It has cardiac stem cells. So these cells can be harvested easier than embryonic stem cells, and Therefore, about 50 different types of adult stem cells have been tested in the lab, and some have advanced to the clinical trials. Even though we work with, my laboratory has worked with stem cells for more than 20 years, I'm not going to show basic science data for this talk because this is focused on how to advance basic science findings to the clinic. So you can see a list of names here, but there are many, many other types of cells that have been tested. So it's very hard to bring basic lab discovery, in particular when it comes to stem cells, because it is extremely difficult, which I'll show a little bit later. So if you look at the timeline, in 1992, that was the first paper which showed the harvest of satellite cells from dogs, and those cells were expanded in culture and implanted in the dog hearts after a cryo injury. Cryo injury is not the best model to test if the cells would work in patients with heart attacks. However, that's what was done. In about 10 years, in 2002, Bodo Strauer from Germany, he tested these cells from the patients with acute myocardial infarction. You may know there are two main types of heart problems for which stem cell therapy is being tested. One is called acute myocardial infarction. When a heart artery is blocked, then the patient gets a heart attack, chest pain and all that. And when that heart attack patient, after a few months or years, the heart begins to fail. So a failure, that's called ischemic heart failure, and then there are non-ischemic heart failure. So treatment of myocardial infarction and heart failure are different. So very quickly in 2003, Dr. Perrin from Texas Heart, he tested these cells in patients with ischemic heart failure. And that was followed by multiple studies. In 2004, there was the first randomized controlled trials with mesenchymal stem cells. In 2009, allogeneic cells were tested. And allogeneic cells are different from autologous cells. Autologous cells come from the same patient. Allogeneic cells come from other patients or normal humans. There are differences, but to use allogeneic cells is more advantageous because then you can keep the product, which is called off-the-shelf treatment. So the patient comes with heart attack, you already have the product, and you infuse that. But if you are going to use autologous cells, then those cells need to be harvested from the same patient, expanded, so it is more complicated and less feasible. So allogeneic cells were successful, and that was in 2009. Very quickly in 2010, a mesenchymal stem cell product was approved in Korea for treatment of acute myocardial infarction. 2011, we saw the trial from cardiac stem cells from Dr. Boli from Louisville. And then StemPucell was tentatively approved in 2016 in India. And people in the audience may know more about this because this was in India, and it was a major advance for critical limb ischemia. In 22, there was a first patch implantation with iPSC-derived cells. These are induced pluripotent stem cells. And 2023, we had a large trial which enrolled 537 patients, and I'll show the results a little bit later. And then 2024, the exosomes were tested in humans. So how effective is cell therapy? So this question has plagued the field since its inception. And for this particular field, for some reason, everybody is interested in the mechanism, not the outcome. So in 2007, 20 years ago, we did a meta-analysis which showed out of 999 patients from 18 trials, the ejection fraction, which is the number that we use, which means when the heart pumps every time in systole, that's the fraction of blood that goes out from the left ventricle. And that's a measure which is tied to mortality, so we look at that. And there are other numbers that improved as well, which are cardiac parameters. So in 20 years, the field has not improved as expected. There are many reasons for that. The primary ones are the cells are not retained in the heart very effectively. That's a big reason. If you inject 1 million cells, only 1% or less will be retained in the heart. The treatment effects, at least by numbers, are 2, 3, 4%. And people may argue that this is not very effective treatment. There could be tumors as well, which is rare. And clinical trials, to enroll one patient in a clinical trial, it costs about $50,000. So to do the trials with large number of patients with a p-value of less than 0.05, it takes a huge number of patients, and many companies are even unable to continue with their trials or quit. So that's another reason. But on the other hand, equally important are the undue expectations. Sometimes patients and investigators expect 10, 20, 30% improvement, which is not possible. Even when you look at the very successful drugs like Entresto, which is a combination of sacubitril and valsartan, even that product only improved survival by about 4%. So it is not fair to expect that stem cells will produce a huge benefit for the patients. And there are differences in opinions, and then the regulations are not very clear, even from FDA. If you call the FDA cell therapy, you may get different answers different times. So there are many challenges because it is not a straightforward option. It's not like you're giving aspirin and that's causing such and such effects. So there are variabilities in the donor. So it depends on who the cell is coming from. If it is the patient himself or herself, what kind of cell it is. Then of course the manufacturing variables are very complex, and that actually cells change. If you plate mesenchymal stem cells in culture, the phenotypes will change as the culture evolves. So typically in the basics laboratory, we use cells from passage 2 to 5 because that's when we have a stable phenotype, so we can have reproducible results. So it's very important to optimize the cell manufacturing parameters. Then it depends on how you're giving the cell. If you're giving it intravenously, you're giving it through the endocardial route, and that can impact the outcomes tremendously. And finally, the recipients. Not everybody is we know that we talk about personalized medicine. That is probably very important for stem cell treatment because depending on the recipient, the outcomes may be different. So the outcomes of cell therapy in the last 25 years have been highly variable. That's one big reason why translation to the clinic has not been very successful. Now this summarizes the different approaches or types of cells that have been used. So something we call first generation cell therapy used more simple cells like bone marrow mononuclear cells, mesenchymal stem cells. Then came the more sophisticated cells such as iPSCs, cardiac stem cells. Then those were changed out because it was realized that injecting the cellular DNA is not really important or necessary. So if you use the secretome or the link RNAs or miRNAs from stem cells, that could in itself produce the benefits. And then more newer studies are using a combination of biomaterials such as hydrogels and cardiac patches and putting cells in them. That way cellular retention is better. And hopefully the results will improve with these newer approaches. Now I will go over 2 or 3 trials that are newer. So this was the big trial that enrolled 565 patients with mesenchymal precursor cells, and 537 patient data were analyzed. And this was a double-blind randomized controlled trial. Cells were injected transendocardially, and placebo patients received standard of care, and the subject study patients received the mesenchymal precursor cells. These are STRO1 positive cells that were described in early 2000s. So for this cell type to come to reasonable clinical use and trial, it took 20+ years. So what's more important, they published the recent data. Here we show on the left top, this shows if patients, control patients with higher hsCRP more than 2, and this is less than 2, there is a difference. The patients with high hsCRP has significantly worse clinical outcome. And when you compare high hsCRP patients, control and treated with cells, there is a significant benefit. That difference is similar when you look at patients with high IL-6. Interleukin-6 is a marker of inflammation. And so what these graphs show, that even if you are in the control group and you have high interleukin-6 or CRP at baseline, then your outcomes are going to be worse. And if you treat with cells intramyocardially, then the benefits are more pronounced in patients who have greater inflammation in the background. This is true for outcomes as well. This is myocardial infarction and stroke during 5-year follow-up. And as you can see here, patients with higher inflammation at background and cell-treated patients benefit definitely in a high ischemic patients in a high inflammation setting. And that is, the same is true here as well. The reduction is 40 to 60% in the outcome analysis. This is another trial which is using a different type of cell. So this is cardiac heart failure. And here the patients are selected at baseline. So that is done by, this is a double-blind randomized control, total of 200 million autologous cells. So they do a potency assay at the beginning. So the patient comes before the cells are injected, the bone marrow is harvested, and cells are checked, patient cells are checked for some markers. And if they qualify, only then that patient is enrolled. So the qualification depends on if they have a high CD34 positive cell in their bone marrow. And they have published data from 10 patients, and there was improvement in 6-minute walk distance, and also they had an improvement of New York Heart Association class at 24 months. So these are important outcome parameters for these cell therapy treatment for heart patients. So they are now doing a trial where they are looking at the benefits of bone marrow derived cells in chronic myocardial ischemia. Patients with a high angina burden will be enrolled, and this trial is actually recruiting patients at this time. There are other types of trials that are going on in which umbilical cord derived mesenchymal stem cells are being used. So umbilical cord, human umbilical cord has supporting cells. Mesenchymal stem cells are present in all the organs. These are supporting cells. They are in the perivascular tissue, they are in the bone marrow, they are in most organs. So the ones that are in the umbilical cord, we have also found in our results that they are more immune modulatory in nature. So they can suppress inflammation. So these cells are being used in this trial which is giving it intravenously. So to give a cell therapy through the heart is much more difficult and complex. So these trials hopefully will have benefit to show with an easier route of delivery. This is a trial in which highly purified ventricular muscle cells are being used as a spheroid. They are developed in the culture as a spheroid, and then they are going to enroll 10 patients. These trials are still ongoing. So they are enrolling 5 patients with a lower dose of 50 million, and another 5 patients will be enrolled in the 150 million cell group. So their safety data have shown that 50 million cells were tolerated well by the patients. So the next stage is going on with 150 million cells. This is yet another type of approach in which a cardiomyocyte patch is developed from induced pluripotent cell-derived cardiomyocytes. These patches are developed in vitro. These are about 3.5 centimeter patches, and they are sutured onto the myocardium. So they open, when the chest is open, the myocardium is exposed pericardium is cut, and the patch is sutured there with proline suture, which is absorbable. And so the patch stays there so that it's not displaced. And the images in the lower panel are showing the red area means vascularity, basically. And so that has improved at 1 year term in patients who received these types of patches. So there was improvement in cellular myocardial contractility as well. So another type of approach involves the use of secretomes. So some of you may know these are called exosomes or extracellular vesicles. There are a lot of data in the basic animal models in which the use of secretome improves the different cardiac parameters and whatnot. So there is a trial going on. This is the first of its kind in Europe, and where these secretome or the exosomes are being injected intravenously. Patients are getting these injected 3 times, 3 weeks apart. And the first patient has showed promising results insofar as this was tolerated well, and there was some improvement in heart function and New York heart classification. There are a number of studies that are currently going on with advanced technologies and products. These include induced pluripotent stem cell, patches, exosomes. These data are from clinicaltrials.gov, and some of these studies are happening in Asia as well as in the Americas. So what's the outcome that we are most interested in? So of course, you want the patient to live longer. I think that's the most important outcome. So we did another meta-analysis in 13 years ago, and we showed that cell therapy improves survival. So this has been confirmed. I don't update this slide, but in a number of meta-analyses, it has been shown that if you give cells to a patient with heart disease, cell therapy, the survival is improved by 30 to 70%. We do not know the reason, and there are many labs that are working on that. However, the fact remains that cell therapy improves survival. So we did a smaller meta-analysis recently in mesenchymal stem cell area, and
So you will have to sum up soon.
Sure. Yeah. We'll go over this very quickly. It takes a long time, 12 to 15 years. Like I said, the trials that are going on now, those cells were described 20 years ago. So the trial that we are doing in GVHD, I won't go over that, but that also we started in 2013, so it's 12 years. So these are the products that have been approved in different countries. That was the first one. This is in India. And also, this was first approved in US. After all these years, this was approved in 2024, and that was for allogenic bone marrow mesenchymal stem cells. So in conclusion, we can say safely that cell therapy modestly improves heart structure and function in patients with ischemic heart disease. It also improves survival. Newer approaches, like I showed a few examples, may improve these outcome benefits further. And many different products, I did not show the full list. There are probably about 12 products, cell therapy products, that have been approved in different countries for various indications. I'm indebted to these individuals who our lab collaborates with, and they also generated our data that I showed. This is our school. Thank you for your attention.
Thank you, Dr. Buddhadev Don, for the insightful insights on, the development of adult stem cells in cardiac conditions. So we have time for one question.
great question. So intra-arterial, you probably mean intracardiac. No, intravenous. Okay. So if you inject any kind of biological material intravenously, most of that gets filtered out in the liver and in the lungs and also in the spleen. So eventually there have been pig studies from 2005 or so in which they showed the actual number of cells that reach the myocardium varies anywhere between 1% to 9% at the most. So we think that the benefits are not coming from the cells that are going to the target organ. The benefits are coming from the secretome that are also in the blood.
So accordingly, the effect of exosomes probably will be more effective than the use of the regenerative stem cells.
Yeah, that's what the field is expecting, because now we have hundreds of preclinical studies that have shown that all the contents I showed in a slide briefly, it includes about 850 microRNAs. It includes different nucleic acids and whatnot, receptors. So they are the real effectors of cell therapy. And so if you give them directly, the benefits will be more pronounced, and there will be no side effect that may come from the cellular DNA.
Thank you, sir. I think the off-target migration of the adult stem cells can be a major issue in therapeutic scenarios. So probably the secretomes would be a better, this with lesser risk associated with it. In the future, probably those studies would be more robust and might lead to more clinical success.
Yes, thank you. Off-target effects of cell therapy are not much, though. There are very few adverse effects that have been reported. There was one case report of intracardiac calcification with mesenchymal stem cells, but I am not aware of too many off-target cardiac cell therapy side effects. However, the point remains that if we inject the concentrate that has all the goodies, that may do the job much better. Yes.
Thank you, Dr. Don. Thank you very much for your talk.
Yes.
The second speaker for today's symposia, Professor Nusrat Shafiq, Madam. Professor Nusrat Shafiq, MD and DM in Clinical Pharmacology, is a distinguished professor in the Clinical Pharmacology Unit of the Department of Pharmacology at PGI. She served as a member secretary of the Institute Ethics Committee for Clinical Trials and Bioequivalence Study, and has been instrumental in operationalizing systems for serious adverse events, reporting and compensation as per the Indian regulations. Her research is highly focused on antimicrobial stewardship, rational use of drugs, phase 1 and dose optimization clinical trials, particularly in the context of multi-drug resistance infections such as TB. She is also deeply engaged in capacity building in research ethics and good clinical practices, authoring multiple publications and books in the field. I request Madam to please continue with the session. Thank you.
Thank you, sir. Thank you, sir, for that kind introduction. At the outset, I would like to thank the organizers for giving me an opportunity to share an experience of nearly 10 years in phase 1 studies, which are typically called the translational studies. Our experience has not been, I would say, so to say, very vast, but it has been very enriching. And I hope I'll be able to just do justice to the same in the next 15 minutes or so of my presentation. I'll be talking about my talk would largely focus on the processes and systems that we have developed over the years, which have gone on to helping in capacity strengthening. And I talk presently also as a principal investigator for ICMR network for phase 1 studies. Having said that, let me start my presentation by a little bit of introduction to this valley of death. It's a very ominous kind of terminology which has been used, But if I just present to you this picture, these two data, you will appreciate that it is, why is it called value of death? When we move from preclinical studies, drug discovery, and we start working in humans to see the safety and pharmacokinetics aspect, and now even some indicators for efficacy through the biomarker evaluation, we realize that a lot of molecules are lost. They have to be killed early. And the proportion way back in 2013 when these papers came up was for every 250 molecule which goes to the next stage, nearly one would be lost at this stage. So it's a huge, huge loss, and that's why it's earned its name as value of death. There are several causes, and these causes delve on the realms of side effect profiles, inconsistent pharmacokinetic profile, and not so much on an efficacy, but if it's in certain situations, even that. So these causes have to be looked into, and an important understanding is to understand this discovery and development disconnect. And that's one reason why we have started emphasizing why those involved in the discovery aspect should work right from day one, I would say day zero, together with those who are going to be involved in the development process. Because over the period of time, that is one learning we have drawn. And this learning has translated into very fruitful interaction with institute like yours, where a lot of background work is carried. When we work with you right from this stage, we have seen that we are able to bring some of these discoveries, early phase developments into translatable products. I would like to just touch upon some of the things that have been developed. Such developments do not happen overnight. A lot of work goes on over a period of time spanning decades. One good example was BRICS initiative by Government of India, which was several decades ago, and which brought industry and academia together to bring products into the market. Now it has taken a new avatar, I would say, of this is the ICMR MedTech Mitra, wherein there is a lot of emphasis for helping early discoverers and people who are working in the basic sciences to take the products to a developmental stage. We all know that we have recently had a launch of one of our antibiotics which was totally discovered and worked upon in the later phases in India, and one of the Indian companies brought it forth for multi-drug resistant infections. But importantly, a lot of players came together to facilitate this, and it is this understanding which is very important. Also, what was realized that the phase 1 studies were not conducted in India. So this was something, and this is happening for many of the products which were being worked upon in the early phases within India. People were going elsewhere, and that's something which sounded talk about having a good network of phase 1 studies. But I would also not fail to mention this important initiative, which was a consortium which developed, and they came together and wrote down the first draft of Chandigarh cluster of research institutes, knowledge cluster. It was acronymed as CRIC, and we were part of it. I would like to share with the audience the first draft of it. My director asked me to write it down, and I'm so happy to see that it has actually transpired into something which is bringing out results now. The point that I want to emphasize is decades and a pot boiler of initiatives and village of diverse experts are needed to bring out this systematic change. So with this, I would like to just touch upon this unit that we developed, that was developed in 2006 at PGI Chandigarh. It was called the Clinical Pharmacology Unit. It was developed with the support from CSIR, and it had certain goals. An important goal was also to create a cadre of trained personnel. We are a teaching institute as well, so we believe in creating this cadre of trained personnel who, when go into the industry or go to the academic or healthcare setups, really bring out these, develop and expand this kind of know-how and systems which can help in translating the products. And very recently, taking clue from all the things that were happening in the country, wherein people were going out for doing phase 1 studies, ICMR took, at the behest of the Ministry of Family Health and Welfare, this very important initiative of creating a network of phase 1 trials. At present, there are just 4 centers, but we hope to have this expanded in the future, and I'm fortunate to be part of one of these, which are located in the institute, which is just across the road. This is just a pictorial representation of the organogram, how we function. It is mainly led by clinical pharmacology unit, and but it is a very collaborative functioning unit. And we have a team which is cross-disciplinary in nature. We have some staff which comes through ICMR, but we work along with a lot of people, and some of the consultants who I don't see in the audience from Punjab University are part of some translations that we are undertaking in this unit. See, it is not just about capacity building, is not about just physical structures. The processes have to be in place, and the team there has to be very trained. And therefore, we develop these standard operating procedures, and we work along on the same. People on the ground are provided training. Here you can see that people being trained by the anesthesia team on basic life support, because when we do any evaluation for the first time in human, we are prepared. We have to be prepared for any kind of eventuality. Not just this thing, the entire ecosystem has to be in place. So our ethics committee with systems for oversight is a must, and we are very happy to appreciate that we have such a system existing. Mechanisms for compliance with serious adverse event reporting, management, and compensation as per regulations also have to be in place, because this is a situation where anything and everything can happen anytime. Even with a very deep understanding of preclinical data, the safety profile, still there are unexpected events. We all know about T. Genero study where many unexpected events occur. We have intensive care facility right there. A basic facility is within the unit, and the team. This is the most important. The backbone of the thing, of the unit and its functioning, is the team, which is comprising of not just clinical pharmacologists, but intensivists and internists who are part of the working team. In order to have capacity building, it's important to have hands-on experience. And this hands-on experience has started with when CDRI, a group from CDRI who had done extensive work on this trioxane peroxide, an anti-malarial compound, which they were trying to develop, and they approached us and said that we want to evaluate it in healthy human volunteers. Together with them, we co-designed the single ascending dose study. We published it. It was found to be relatively safe. We went up to 6 dose escalation. Subsequently, we carried out a pharmacokinetic study in 16 more healthy volunteers, and we were pretty satisfied with the results. For some reason, the product never went on to getting translated because there were logistic issues and those other things, but all those who were part of this study got hands-on experience, and the unit got strengthened. So one important thing was crosstalk among organizations and industry is very important. IPCA was one of the small companies at that time, was our industry partner, which gave us the GMP developed product. When India was grappling with the situation of MDR TB, and we were looking for molecules which could help us, one of our colleagues who had worked extensively with colleagues in JALMA and had repurposed an existing molecule in animals and had done some work initially, we really zeroed on to this. But what was missing was that we did not know how would it fare well in human. Does it give a good pharmacokinetic profile and safety profile? And we proceeded with this study, and we found it to be very reasonable. Ongoing works are going with this. Interestingly, this compound has also shown its efficacy for other pathogens, and this work is continuing. Like, we have had some successes, but that's not to say that all of it is always a very fruitful end. One of the compounds which was painstakingly worked upon with another colleague and a team and a pharmaceutical company. They approached us for evaluating. This was a sustained release nano formulation of a first-line anti-TB drugs, and it had a promise of increasing compliance. But what was not known was how does it fare when given to humans. Unfortunately, we could move only with the single doses evaluation. We could not go to the next dose because of formulation issues. In the next dose, we would need to give nearly 12 capsules to each individual. So that was one of the limitations, and these are the challenges that we have to deal with when we do our phase 1 studies. While the team is still working on the formulation for the time being, we could at least show that safety-wise, we were not having much of an issue, because one of the excipients there was PLGA, and nobody had really evaluated it when it is given orally. The safety of it was not known. So we were able to determine the same. These were some of the examples that we have done from the academic centers. But you know, in order to be in the drug development area, we cannot live alone. We have to partner with the pharmaceutical industry. And not just that, we have to partner with other people who are working. So this is an example of collaboration and co-development that I would like to say, because that is also one of the important aspects of system building. Here, one of our colleagues in anesthesia had developed an oxygenation device which had certain improved features which were there from the existing device. They had done all the preclinical evaluation, and then they wanted to do its evaluation in healthy human volunteer. It required intra-arterial cannulation and evaluation of blood gases. They did not know how to proceed. We said, we'll do it the GCP way, and we'll produce the data which you can be very confident about. Come join hand with us. And they were very readily did so, and we carried out a single ascending dose study for this device and evaluated. I'm not presenting here the data for the blood gases parameter, but the observed and predicted parameters are matching very closely. So that's another important way to develop things. Collaboration is the key, teams matter. So sorry, I was talking about the collaboration with the industry. It is not only the academic centers which come to us. Now, increasingly through ICMR's collaboration, the industry has come ahead and started partnering with this. And I would just like to mention one of the Indian small industries which has gone into drug discovery and development in anti-cancer drug is carrying out their first in human evaluation in cancer patients for a small molecule which they had worked extensively. The preclinical models showed anti-tumor immune activation potential for this particular molecule, and they used the 3+3 design. I'll not delve into the details because that will be out of the scope of this presentation. This study is ongoing. Right now, we are at the 5th cohort for this study, and it is progressing pretty well. And during the course of the study, we had a very interesting finding, which perhaps will feed into subsequent phases of the trial. So this is something that I wanted to let you know about, how when you develop an ecosystem, you have to move out of your silos and work with all kinds of partners. This same group is working on another molecule with us, and again, the target is non-Hodgkin's lymphoma or Hodgkin's lymphoma. Here, I would like some of you to understand for whom this would be a little unfamiliar topic, that here we try it out on patients who have failed all therapies, because you can't possibly deprive somebody of an existing treatment if they're suffering from such a condition. So again, here we have had some interesting findings, but because of confidentiality issues, I can't really discuss the same in details here. But another important forte, and for which India is very proud of, is the vaccine development. And I would like to share with you that the Covishield vaccine, when it was evaluated in our unit. Here came the importance of oversight also. The ethics committee had an on-site monitoring done for that to see the compliance with the protocol. We have graduated to the first in human study for another molecule, for another vaccine candidate, I won't say molecule, which is a pneumococcal conjugate vaccine which has been developed by Serum Institute of India. The study has already initiated. What we do is we have a cohort of healthy volunteers, because it is very important when we are doing such study that these cohorts are well maintained, they are well evaluated, they are well observed, their rights and are well taken care of. Because unless and until we take care of these things, next time they will not come forward and subject themselves to the risks. So this study is ongoing. Already the recruitment is in force. At the bottom of all this is regular trainings. We provide regular trainings in GCP, GLP, basic life support, which I already alluded to, consent process and medical examination. A lot of time, hands-on training is given. So our motto is like every DM candidate who passes out should be trained on one phase 1 study. If it is missed for some reason, we ensure that they get this experience in another format. A lot of people have come together for enhancing this training and capacity building. We conducted a workshop last year, and the theme for this workshop was regulatory and ethical aspects of phase 1 studies. Interestingly, a lot of regulators came down, even from US FDA. The industry partners worked with us in co-designing this workshop, and it was very well appreciated. A sister workshop has recently been concluded at KEM, and we all joined hands again in strengthening and capacity building in this area. I had mentioned about standard operating procedures, and here ICMR team and a J&J team had come down to our department, and they worked with us for enriching our SOPs, making them more crisp, increasing the scope of SOPs. Subsequently, ICMR under this network conducted a workshop, and Dr. Nilanjan Saha had carried it out for us, and it was a learning experience of the sort. So the point that I want to make is that it takes years of efforts to bring it to a level where it starts bringing the results. It's important to have some templates readied here for which are locally contextualized, and some of the templates, which are not very many, we have created are in the form of these two publications that we made. Important one is the one wherein we developed a tool for assessment of comprehension of informed consent, because that was always a concern that participants, healthy volunteers, when they're subjecting themselves to risk, how do they comprehend it? We developed and showed that there are ways to address such issue. So I'd like to just conclude by saying that from bench to bedside transition requires several role players. It is from the people from basic sciences, people from kinetics expert, people who have expertise in clinical pharmacology, and the subject domain experts. Capacity building and strengthening at multiple levels. It should occur at multiple levels, and it should involve multiple stakeholders. Units with expertise in conducting such studies are the need of the hour. And it's important that the ICMR's initiative has started showing its results already. And we need a cadre of trained individuals for addressing the discovery development disconnect that I alluded to in the beginning. I'd like to conclude by saying that learning by doing, sharing, setting examples, systems grow holistically. Thank you very much.
Right on time, ma'am.
I'll be happy to take any questions.
Yeah, we can take a question. Yeah. 1 or 2 questions. Any questions for Dr. Nusrat?
Yes, sir.
Thank you, madam, for the excellent presentation. Recently, we had this workshop at KEM Hospital, as you mentioned, in Mumbai, and Madam Shivsagar team and Dr. Nitya Gupta's team was here. So I am from Hetero, and we look forward the initiative what ICMR has taken, setting up this phase 1 facilities across India at renowned institutions like KEM Hospital. I am from KEM, so we always feel proud about that. But at the same time, we at industry, we always look at the speed of, getting things done, getting the recruitments done. So that is the one thing I would like to request all the ICMR centers to, take up that speed and complete the phase 1 clinical trial, the single ascending dose and multiple ascending dose, because as you know, the drug discovery, it's a time-bound project and every day is important. So if we can have some kind of initiatives for getting, expedite recruitment in phase 1. Thank you.
Thank you, and I really appreciate your concern, but while speed is also our concern, we cannot it cannot come at the cost of safety at any point of time. So having said that, I would like to say that the model that is being used is a multicentric model. For example, for the 2 studies that I showed which come from the industry, 2 centers are involved. So that addresses recruitment speed to an extent. For vaccine studies, we have a very reachable cohort which we can reach out through our community medicine department. In fact, they are the ones who are leading that product. So we have these mechanisms and we have a cohort of healthy volunteers whom we reach out. I think your concern is appreciated, but the bottom line remains not at the cost of safety. Thank you.
Thank you, Dr. Nusrat Shafiq. It was indeed wonderful you narrating the trajectory of, being part of this phase 1 facility at PGI and the kind of support from the government, and you all have evaluated not only, investigational molecules from the government like CDRI, but even the industries, vaccines, and also device. So I think Indian innovators and companies are looking for, centers for having phase 1 evaluation done. And as you said, it is very critical. I congratulate you, your entire team, and we are so happy and proud to know that the happenings at PGI and now came to know about KEM as well. Thank you very much for your wonderful talk. Thank you.
I'd like to say the brainchild behind all this is sitting amongst us. Dr. Nilima Shirsagar is sitting there.
It is so wonderful to have Dr. Nilima Shirsagar in this session with us. Ma'am, you'll have to please wait. After this session, we will have the felicitation. Volunteers, I have seen Dr. Don leaving this venue. Can you just call him back so that, we can felicitate him after the 3rd lecture?
The third speaker for today's symposia, Professor Parth Roy, sir. Professor Parth Roy is a senior professor in the Department of Biosciences and Bioengineering at IIT Roorkee, with deep expertise in animal biotechnology, molecular endocrinology, stem cell biology, cancer therapeutic research, and tissue engineering. He earned his PhD from Vishwa Bharati Vidyapith, Bharati University, and has led numerous projects delving into hormone mechanism, cell biology, and regenerative medicines. Professor Roy has published widely in a peer-reviewed journals, supervised graduate students, and contributed significantly to both basic and translational research in biomedical engineering. I request, sir, to please continue with the session. Thank you.
Okay. Good morning, everybody. So, first of all, my sincere thanks to the organizing committee, Professor Anil Kumar, Professor Sangeeta, Professor Sandeep, for inviting me to this talk, presentation, and giving us an opportunity to present some of our ongoing work. Since I come from a technical institute where our mandate is to do some translational research to take it out to the commercialization. So today I will tell you a story about the development of a scaffold, nanofibers based scaffold for the tissue engineering purpose. So my collaborators, before I start, I need to acknowledge my collaborators. Professor Deblupa Lahiri, she is basically a material scientist, and I take care of the biology part. And Dr. Shobhi Ghosh is our PhD student whose work I am going to present today, who did detailed work, and now he is a postdoc in US in Mayo Clinic. So if you see the neural system here, so we have two types of, for the students are here, I just want to give a brief description about it. We have central nervous system and we have the peripheral nervous system. And central nervous system is that what is brain and the spinal cord, And when you see the peripheral nervous system, it consists of the peripheral part of the neurons and all these things which are spreading out. And when you see in terms of damage that happens in the neuronal system, it is this peripheral nervous system is more prone to damage due to accidents and other disastrous issues. So there is a need of some regenerative process that can be used for the cure and management of those damages that are caused in the neurons or in the nerves. So with this, when I see the nervous system problems, so if you have peripheral nervous system, neuropathies, injuries are there. We have spinal cord injuries, complete or incomplete, traumatic brain injuries there, loss of cognitive functions, loss of motor control, and alteration of behavior. And we have neurodegenerative disorders, which we have heard about in earlier 2 days about these two diseases, so which I am not going to talk, but I will talk about the traumatic brain injuries and other these systematic injuries in my talk. So when I see this one, this is the cartoon, you can see this is the nerve injuries, and this regeneration process is extremely difficult. Nerves do not come and joins together. And you have to understand that therapy of the nerve is very difficult because nerve needs not only mechanical support, but needs an orientation. If the orientation of the nerve is not properly done, the nerves will not function properly. So what we are trying to do here is, so generally the therapies are autograft or allograft cell therapies, or non-surgical means medicine, lifestyle improvement, etc. And treatments focus primarily on limiting damage and slowing the degeneration process. So alternative approach, what we are thinking about is basically neuroscience-based approach, using the engineering approach, and then combining it together with neural tissue engineering. So this is our base, and with this base, we have been trying to work on our laboratories. So if you see what happens during the nerve injuries, so when you have nerve injuries, this is suppose the nerve injury is there, a gap is created. Now this is a proximal stump and this is distal stump, so it has to be put together so that the nerve gets regenerated. So when it happens, so there are several approaches. You can add growth factors in this region. You can have supporting cells, as you heard just now, Professor Don was speaking about in the cardiomyocytes. Same thing can be applied in the neuronal systems as well. And then we have the 3D scaffolds here, multi-channel lumens over there. Nowadays, people are coming up with hydrogels, which are filled with growth factors, other factors. They can be used, or internal materials also sometimes implanted for the regeneration process. But what we are talking about is the aligned nanofibers or nanomaterials that can be aligned and that can be implanted as a scaffold in this damaged area, which will actually help in the regeneration process. So now when I see tissue engineering applications, neural tissue engineering, neural constructs, it should have mechanical properties, it should have electrical properties, as I was telling you. Nerves needs an electrical impulse, it needs an electrical stimulation, it needs an orientation. So electrical property is required. Topographical cues means where it is located, that is also critical. Biodegradation properties, once it's implanted, it should degrade after its job is done. That is what is required. Biocompatibility and control release of bioactive agents. These all parameters are required for development of a good quality of scaffolds. And so what is happening at this point? There are some scaffolds available, but the problem with them is that most of them are either not biocompatible, or sometimes they provide the mechanical support, but they do not provide the electrical impulse supports, or sometimes they provide electrical supports, but they do not provide the mechanical support. So we need to address something which will take care of everything. And this is how the hydrogel concept works. So if you have the hydrogels loaded with your biomaterials, whatever you want to implant, and then you go into the brains, and then they stimulates and the neuronal cell gets regenerated and all. And sometimes people are talking about scaffolds loaded with stem cells injected from the brains, and then they start functioning, and they can, this scaffold provides support, and these cells migrates, and then the regeneration happens. So when you talk about this, so what I just was talking about, most of the problem that we have, polymeric materials that we have these days, they have problem of mechanical stability and electrical topographical issues. Some of the synthetic natural polymers are there, but they are not very stable one. And conductive polymers what are available, they have problem of less biocompatibility and not degradable one. So with this, when you looked about, there are multi-wound carbon nanotubes and graphene nanoplates are there, which are very good options, but they are very difficult to implant, and when they do their functions are not very well governed. So by turning this limited hard substance into which are difficult to implant in the in vivo system. So all these pose a serious issues, and so we wanted to address these all together in a single package. That means having the polymeric materials and then implant them and then reinforce them with these materials so that we get a complete material out of that. So this is how it looks forward. So this is the nerve injuries. You can use stimuli, you can use cells, you can use biomaterials. And what our goal was this one, we have a polymeric materials, and these are loaded with biopolymers, which are loaded with nanofillers, a reinforced one, which will give mechanical properties. And these polymers could be natural or synthetic one. And we used here carbon nanotubes for this purpose. So with this, we wanted to first optimize the different carbon nanofillers and reinforcement inside them, and then induce the embryonic and mesenchymal stem cells for the differentiation on those materials, and then we can synthesize the tissue-near scaffolds. So this is our goal. What we did, in summary I am talking about, so because the time may be less when I finish it off. So what we did the this bioengineered scaffold materials, which were solution casted materials were there, where we took this PCl, polycarbonate as a polymeric material. Within them, we loaded them with multi-walled carbon nanotubes or graphene nanopart plates, reinforced materials into them. And this actually provided them mechanical strength and electrical properties. And we saw their neural regeneration capacities. And then this was with the graphene nanoplates, same thing we did. And then what we did to check that if the electrical impulses on the cells growing on the scaffolds induces them better functions, we did some electrical stimulations on those same materials, and cells were electrically stimulated. So we got these multi-walled carbon nanotubes and graphene nanoplates together. So this was also tested. And then what we did, we made the electrospinning method, because this is another method where you can use different combinations of materials for nanomaterial formations. And then to this we added some medicinally important plant materials into that, that is from the bacopa into these materials, and we made a modified materials combining all this approach what we did together, a new materials which was better suited for that new regeneration process. So as the first part of it, we made a multi-walled carbon nanotubes that was reinforced into the PCl or chitosan collagen polymeric scaffolds, and also we made another one where we used the graphene nanoparticles for this purpose. And so this is the first one. When we made the multi-walled carbon nanotubes that was produced, and then what we did, there are two options. You can align the multi-walled carbon nanotubes electrical stimulations, it was random, or aligned options are there. So you can see here, this is the multi-walled carbon nanotubes are aligned within the polymeric materials. So this was aligned one, otherwise they are in a random process. We used both of them. I wanted to see which was better option for our use. And this diagram actually shows the general characterization of any scaffolds when you make, they need to be characterized with this. So this is our material, and we wanted to wrap it around the damaged neurons. So this is the random carbon nanotubes and aligned one, which you cannot differentiate between them. But when you look at them at the same level analysis, you can see here, these dots indicates that all these aligns, these multi-walled carbon nanotubes are aligned over here. And when you check mechanical properties, so it is very strong as compared to others one. Electrical properties are very, very strong in this material, and biodegradable kinetics was also very good in this material. That means it was biodegradable, conductivity property was there, mechanically strong properties are there. Next, was it really cellular compatible? So we tested them, cell viability was increased, that means allowing the cells to grow. And when you check for the neuronal cells, you can see these are neuronal cells, and you can see these very nicely formed neurons with the neuroid growths. And when you check the number of neuroid-bearing neurons, so the neuroid growth was very, very high in these cells, and the neuroid length was also increased. That means it was enhancing the neural tissue formation or nerve regeneration process. So with this, when we completed this study and we published this study on a journal where we established that multi-walled carbon nanotubes can be used as an materials or reinforced material for the polymeric material. Next, we made the second material of ours, that is graphene nanoparticles plate, graphene nanoplates loaded with chitosan polymer scaffold. That means it was reinforced not with multi-walled carbon nanotube, but it was with graphene. And we got another material from here, and you can see this was the same thing what you see here. So this is the aligned one. So when the aligned material, this is the plates that we made, and this is the aligned ones, and this is the random one. And when you checked at them, so the same thing, mechanical properties, electrical properties are checked. As you can see, the mechanical properties are very strong. Electrical properties are also very well done, and random values were done. Next, when you tested for the biological properties of these, so they showed that clear biological properties of these materials. So you can see the cell growth was very, very strong on those scaffolds. Cell proliferation was high, and also the cells were growing very heavily. No cell death was observed on these materials. So they showed our material was very, very strong and highly biocompatible in our use. Next thing, what is important, that cells are, neurons are growing on them. Are these neurons really functional or not? So we have to test that, and when you check for the neural outgrowths, as you can see, these neuroid growths are there, or when the new cells are growing on the scaffolds as compared to the control cells or the cells growing on only on the cytosol. So you can see the neural cell population was increased, cell survival was increased, also the number of neuroid was also increased. And when you check for the expression of CCAT, that is choline acetyltransferase, and NF160, they are both high expressed, and they are the indicators of differentiated neuronal cells. So with this, that means our cells are differentiating on the surface. And next, we published this one in Biomaterials Advance, and then we moved towards the next part, where we wanted to give electrical stimulation on the cells sitting on the biomaterials or the scaffolds. So we wanted to see, does this material that you have made differentiates the neuronal cells or not? So we used H222 cells, which are hippocampal cells, undifferentiated, and we used mesenchymal stem cells. And what is interestingly what we did, this is the scaffolds, cells were seated on them, and then we given electrical stimulation at 20 minutes per 20 millivolts, 100 millivolts per centimeter for 5 days, and the cells got aligned on the biomaterials. So are these cells better than these cells or not? That's what we wanted to see. And you can see here very interesting thing what happens. As you all know, that when the cell differentiates, they stop proliferating. Say they just dies or they do the function. A proliferating cell cannot differentiate, and that's what showed over here. What we see here, the cells were initially, when this stimulation was given, the cells were proliferating very fast. But when the cells are given electrical stimulations, you see they are not proliferating. Then if they are not proliferating, what they are doing? Are they dying? So you can see with a scratch assay, the cells which were given electrical stimulations, they do not migrate. That means the proliferation is not there. So this means the cells were actually differentiating. So this is what we showed, and these are some of the markers which showed that cells are really differentiating. And as you can see, the differentiation markers, MAP2 is a marker of differentiation of the neurons. They were shown over here, and the cells differentiated. So, and these are the markers, MAP2 and tubulin are the markers for differentiated neurons, and they are upregulated in these cells. And with the electrical stimulation, their response was very, very high. That means you see the cells, given electrical stimulation, cell differentiates better than what you see. So, and then same thing we did for the other scaffold that we see here, where we seeded the cells with mesenchymal stem cells, and you observed the same thing at what you saw. You can see the neuroid growths are extended in response to electrical stimulation when we used the graphene plates in our scaffold materials, and the cells also did well. So nestin, which are markers for the differentiation, they are all PI3K, AKT are upregulated in these cells, which shows that neurons are really doing functions properly. So we published in Translational Biological Macromolecules. This was accepted, and we went on to next phase of study. So my collaborator, Professor Devrupa Lahiri, is basically a material scientist. For her, the natural products does not make any sense. On the contrary, I am a natural product person who works on natural products. So I wanted to induce some of the biomolecules into this particular scaffold and use it for our applications. So I wanted to see, can we load the Brahmi, as you know, Bacopa, as an into the scaffold for its enhanced application process. So she said that you have to validate this thing in your model system, then I can allow you to use it in my materials. So I had to establish myself that, okay, this Brahmi or extract or the Bacopa has a potential to improve the neural cell differentiation and its function. And these are the basic data which shows that how we established that how they can do the function. As you can see here, in response to this Brahmi, the cell's behaviorability was enhanced because oxidative ROS generation was reduced, and BAX was reduced, and BCL2 was enhanced. That means the cells were proliferating, and cells were seem to be very, very healthy in these conditions. So we continued establishing these facts that this Brahmi was really good for the neuronal cells. And as you can see, it was all the time improving the cellular conditions. So if you give H2O2, ROS generation was there, but my drug or this extract of Brahmi was showing the effect, and it was removing this ROS generation, and it was through the expression of GST1 catalyst, 2 different ROS markers in the cells. So this showed, and again when you showed by immunofluorescence analysis, the catalase and GST1 was highly expressed in the cells, that was allowing the cells to survive in these stressful conditions. So Bacopa or Brahmi was inducing the cells to proliferate better. Next, we wanted to see, as you all know, KIAP and NRF2 pathway is very important for the oxidative stress. And what we found was that this NRF2 level was going up in the cells while the KIAP1 was going down. And this KIAP is very notorious material molecule, does not allow this NRF2 to migrate to the nucleus. As a result, the cell undergoes a stressful condition. So what we did when we did a bioinformatic analysis to show that what was happening with KIAP and NRF2, and we found that Brahmi was actually binding to the KIAP on site and inhibiting the binding of NRF2. As a result, NRF2 was migrated to the nucleus and causing the oxidative stress stimulation, recovery from the oxidative stress related stimulations. And so, when you check for this one, as I told you, the nestin, beta tubulin, MAP2 markers for the neural differentiation. So, they are all upregulated in these cells in response to Brahmi and MAP2 and all. But what is interesting, as I told you earlier, cyclin D1 is a cell proliferating agent. Its level was downregulated, and NCAM is a neural differentiation marker, was upregulated. And as you can see here, NCAM level was very, very high, and these are neurites outgrowths, and neurons look very healthy and happy in this condition. Cyclin D level was low because cells are not dividing, but they are differentiating. So that's why we could show that this was really very healthy, keeping the cells very healthy. Now, Dr. Debrupa Lahiri allowed me, and we published in Journal of Pharmacology, to show that neurons can grow very well in response to these Bacopa or Brahmi. And we showed a mechanism of action for this. So now Dr. Debrupa Lahiri allowed me to use it in heart scaffold, and we developed a what we did develop earlier. Now along with that, we added the Bacopa monadi. That means the extract of this Bacopa. We got a new material, and this was again tested for its biophysical properties. So this is the Bacopa containing nanofibers that we developed, and then its stability was shown. It was biodegradation was very efficient, and this is the cell viability of these cells are very healthily, happily going on them. So, and this is how we looked at it when I looked at it. So, this is the neurons growing on these scaffolds. You can see this is the same image of the scaffolds and the cells growing on the aligned scaffolds. And when I see the cyclin D level in these cells, so they are down regulated in these cells expression, that means they are differentiating. And when tested now, what we did is that the sciatic nerve injury in this mouse and implanted this scaffold into it. And what you see here, this is the left leg of the animals, which was damaged on day 30 and day 60. It was damaged, but the animals where this scaffold was loaded into them or wrapped into it, so they comes back. These are footprints of the mouse, and also they come back as good as the right one. So their animals are moving normally. This is the gastrocnemius muscles, which were damaged in the animals, which were damaged neurons. But once this wrapped with these particular materials, so they have come back. This is the left one, what I am showing. This is the staining of these gastrocnemius muscles, nerves of muscles over here. They are all collagen lost, but with response to the wrapping with this one, so it was back. So it was not only doing the neuronal function, it was also inducing the function of muscles also. So it was helping in the neuromuscular junction formation. So, and then finally we check the sciatic nerves. So you can see the negative control nerves are all damaged, but when the nerves are wrapped with our scaffold, it regenerated back. This is sciatic nerves which we damaged in the animals and wrapped it our materials along this one. So these are neurons, and this is the negative control of this myelination. Myelination all gone, but in response to our this scaffold, the myelination has come back. And what is most important is that yesterday we were hearing that some of the researcher was talking about that vasculature of the neural system is very, very critical, because sometimes neuron regenerates, but vasculature is not there. And as you can see, this red arrow indicates the vasculature has come back in the neural system. Neuron has regenerated with the vasculature structures coming back. So with this, we showed the markers NF60, 160, and 100 are differentiated neural markers. They are all upregulated, and they could be seen in these animals. So this was actually shown. So we published this in recently, this year only we published in small, and we could show that first time that a reinforced material can be used for the purpose of having biological materials from the plant source as well as from them.
Dr. Partho, you will have to wrap up.
So this is my current group of research. So these are my Deepmala and Momita, my postdocs. These are others, my PhD students working on different areas. And this is the work I talked about was Dr. Shobik, who worked. These are my collaborators, and these are my different funding agencies. So this is the state of Uttarakhand where I come from, and it is the country of God's. It says that God's own country. You can see the rivers, and this is Rishikesh, which is the, welcome to my state if you have any opportunities to come here. Thank you.
Thank you, Dr. Partha. It was a very exhaustive talk. We have time for one question. Yes. Yes, sir, Dr. Nilima. Can somebody give her the mic, madam? Huh.
Dr. Partha, that was a very fascinating talk, and I think it must have taken several experiments for you to be able to present this work. I don't know how many years this work it is.
Long time.
Several work. I just have one question. When we are, translating this into, let's say tomorrow you are ready with all of that into human studies. Two questions which I have. One is, the concentration of the product that you have used, whether it is bacoside or whether it is another chemical, they're quite high. 20 microgram per ml or 200 microgram, that's a pretty high concentration. Normally if we consider that this is a product which has been used for many years and therefore it is safe and all that. But given orally, the concentration that you will get in the body will be very low. I don't know, maybe some kinetic studies will be available. So I'm just wondering, it's very high. So what do you say about that? And that's one. And at that high concentration, what will be the local cell toxicity? I'm sure you would have looked at these. I thought you might be able to tell us a bit more on that.
Yeah, thank you, ma'am, for such a nice question. We have checked it for local. See what happens, whatever we load, only a part of it is released out, if not fully. So whatever we get, it should not be toxic to the cells. So that way we have already tested it out. And we have tested, what I reported here is for the mouse. We have tested the same thing on the dogs as a higher model. Same thing happened, cytokine injury, and we have tested this. Toxicity was still not noted. And now we are looking for a clinical trial. We are collaborating with some industries to take it further.
Any more question?
Dr. Roy, already you presented so much efficacy, safety, and toxicity as well, I believe. So, have you approached through a CRO or industry to develop it as a phytopharmaceutical or as a novel drug delivery system of this bacoside?
No, it is basically it comes under not exactly drugs, it comes under device. So we tried to talk to DGCR about the pharmacological things. So it comes under device, it cannot be a drug. So what we are saying that because it will be implanted into as a wrapping material on the injured nerves. So its mode of action will not be exactly targeted drug delivery because it will not deliver drugs. We are working as regeneration. So we are trying to see that neurons get aligned and then they form a particular structure. It will be completely acellular materials. We are not loading any cells also there. It's just a material which will induce the nerve regeneration.
Thank you.
Thank you, Dr. Partha. Indeed, it was an exhaustive talk, and we pharmacologists, we know how difficult it is to, bioengineer muscle tissue or neuronal tissue compared to, let's say, epithelial connective tissue matrices. So indeed, a commendable research must be going on for several years, and wishing you the very best for the translational part of it. I request Dr. Roy to stay back on the dais. I would also request Dr. Nusrat and Dr. Buddhadev to please come up to the dais for felicitation. I would request our colleagues to give a loud round of applause for our very commendable speakers, fascinating, different arenas of work. We enjoyed hearing them. Thank you very much.
May I now request session chairs, Dr. Anuradha Majumdar and Dr. Manoj K. Aswar to felicitate our esteemed speakers.
We would request Dr. S.K. Kulkarni, sir, to...
And I also request Professor S.K. Kulkarni to please join.
We would request Professor S.K. Kulkarni to felicitate our speakers. So please come.
Now, I request Professor S.K. Kulkarni to felicitate session chairs, Dr. Majumdar and Dr. Manoj K. Aswar. Thank you, sir. So with this, we conclude Symposium 27. And now we will take a break here for a cup of tea. And we request all the delegates and participants to kindly return back to the hall by 11:20 a.m. for the exciting IPSCON 2025 quiz, which is going to be held just after the tea break. I request all of you to please return back after a quick break because it's already 11:35.
We are starting it. We are starting with the prelim round for the quiz. Come with your partners. Come with your partners. All one auditorium only. Reach back. This is Dr. Nitin Kothari this side. Reach to the hall 1 for prelim round of the quiz. With your partner, Sampriti, they open you the table. Na? You all of you please reach the hall 1 immediately. Okay. So team 4 is informed, team 1 is informed.
this Mr. Yogesh Yadav? Dr. Nitin Godhare? Dr. Nitin Godhare this side? Please call number 1 for the prelim round of the quiz as soon as possible with your partner. Prelim round of quiz is starting. Please reach with your partners.
Kumar? Dr. Hassan here. We are registered for the quiz. So please come immediately to hall 1. We are starting the prelim round. With your partners. Please come. इसको कॉल कर लिया 1 और 3 कॉल हो गया
Okay, 1 and 3.
2 is not picking up.
Okay, then sir, 4 number.
Can you brief me about quiz rounds?
You are participant?
Yes, sir.
Please wait for some.
Sir,
First round will be the written round.
okay. And then second one also?
Second will be in the face-to-face.
Okay. On our mobile phone, question will be displayed or here?
Here, on the screen.
Okay.
Just please sit down here. We will inform you.
Single person can also be.
What's your name?
Komal.
Komal, na? You are in the team, na?
Yes, sir. If someone wants to.
No, please call them. Actually, we have the 2 or 3 person who are single. We can club run together.
Okay.
Who is interested?
It's just that I am wanted to be single.
Please sit down here. We are calling all the person who are the single, na? So they can be clubbed up in a team.
Okay.
So I am trying for the team number 2, sir. So no one is picking the phone. And sir, these team number 4 and 5 has been informed.
Hello. You are Mr. Karthik? You have registered for the quiz, na?
Yes, sir.
What about your partners, Nikita and Gitanjali? They can come? So ask Nikita to come. So she can start. As soon as you finish, you can come. Right. We have to start the prelims in another 5 minutes. Hall 1, the Lord. So Nikita will come. They are tied up in posters. As soon as they finish, they will come.
Okay.
All 2 is contacted. Any team already there?
No, sir. One person has been come here. That girl.
Any of the students have registered for quiz? Are you here? Okay. Can you just tell me your names? Please come here. No, we'll write them down. We'll write them in the book. So please come on stage. I'll just tally the names. No. I'll just verify the names, then they go back. We'll just. No, I have a pen. Meera Kumari, Sampriti, Komal. All 3 of you are there?
Yes, sir.
So team 4 is here.
Okay, sir.
Okay, you sit there. You can sit in the first 2 rows.
Okay, sir.
First 2 rows. Please side by side. Next team. No. First prelim round would be a written round, so you can sit in this room.
Excuse me, sir. I have registered solely.
team up with Kunal. Kunal Rana is also small. So at least you will have two members who will consider you as team number 10. So Chanchal is here. So we will pair her with Kunal. Okay, sit. You can sit and talk.
Which row?
First two rows, any of the first two rows. Okay.
Answer team 1.
Team 1.
Sir, we two people, we need one more.
Now, what's your name?
Sir, Aditya. It's where? Team, oh sorry.
Aditya.
Team
This is
7.
team 7. So Priya, you will
Yes, sir.
mention your name.
No, I sent Supriya.
So you can pair up with him.
No problem.
So team 8 is here. So you can sit.
Sir, we need one more.
In the meantime, if you can get one more, you're welcome. But you can start with your team. Next.
Sir, Roshan.
Roshan.
Manisha.
Roshan, Manisha and Om.
Yes, sir.
So we are here. Team number 1 is here. Okay, thank you. Sit.
Thank you.
Kunal. So Kunal, you are paired with Chanchal. Okay. So you can sit. Where is Chanchal sitting? Find Chanchal and sit with her.
Sir, Rohan.
Rohan. Rohan is which one? Rohan, not here. Rohan is not on our list.
Sure, register with 3. We have a team of 3. There is preparation list.
Okay, give me your names.
Rohan.
Surname?
Brandon. BHA.
BHA.
DAN. GE.
Okay. Who is
Jagdish.
written?
Jagdish.
Jagdish.
Jagdish Chan. CHA.
Jagdish Chan we had, na?
Yeah, actually.
Jagdish Chandan and Vishnu Sai.
Jagdish name is already here.
No, it's Chan.
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 Okay.
type paper. It's basically a race against time. There are 60 questions, +1 for a correct answer, -half for an incorrect answer. So there are negative markings, so be careful. And 30 minutes is allowed. So don't start right now. Let all the teams receive it. So I'm calling out as the names that we have. So first, make it a point that on the designated place, please write down your names. You can keep some space between the teams. So spread out. You can sit there. There's nothing to look at the screen, so you can spread out. हाँ नहीं ठीक है, वो तीनों एक साथ है वैसे टीम है हाँ, sit as a team. So Nitin, first team to first three names I have is Roshan Nagar, Manisha Pandey, Om Devangan. You can give them the then Meera Kumari, Sampriti De, Komal. They are sitting on that side. First, write down your names, please. Next team, Nivas, Jadav and Abhishek. One is in the poster, so they'll join us. Next team is Kaushiki, Niharika and Savita. Next team is Aditya Vikram Singh and Supriya. Next team, Ashu Joshi, Jyoti Sarup and Amay Gupta. Next team is Vishnu Sai Bire, Keshari Kumar Srivastav and Sanjeevan Gupta. Next team is Kunal Rana, Chanchal Mittal and Shivam Verma. Next team is Rohan Vadange, Jagdish Chand, Mansi Trivedi. Okay, no one should keep their mobile phones out. Please keep them in your pockets. If we find you are looking at the mobile phones, you are disqualified. Next is Akshita, Simrandeep Kaur and Payal Thakur. They are there. Nitin, they are there. Akshita, Simrandeep and Payal. Have I left out any team? All the teams covered? So have you written down your names? Okay, so you can start now. It's 11:55, so by 12:25 we should hand over. What's your names? Karthik, Gita, sorry, I left out your name. Just give them. Karthik, Nikita and Gitanjali. So time starts now. And don't forget to write down your names on the designated place. Nitin, एक होता सर को दे दो. Please write down your names. Keep your mobile phones aside. Nitin, Shiv Kumar Saini, Vivek Kumar, they have just arrived, so we can give them one paper.
Some rounds of questions will ask
So we are ready to announce the results. Okay. So we are naming the 5 teams which go into the final round, which will be starting immediately. First team which goes is Meera, Sampriti and Komal. Congratulations. I'll request the audience if they can shift to the second row onwards, please, and allow the 5 finalists. Second team which has made it to the final round is Vishnu Shai Bire, Keshari Kumar Srivastav and Sanjeevan Gupta. Third team which has made it to the final is Jagdish Chand, Rohan Bhadange and Mansi Trivedi. Please come in. Fourth team, Abhishek Akolkar, Nivas Marge and Jogesh Yadav. Please. And the fifth and final team, Chanchal Mittal, Kunal Rana and Shivam Verma. So that I'll be able to identify the teams. नहीं वो डिस्कशन होगा तो मुश्किल हो जाएगा ठीक है team A whichever team is sitting here we'll consider you as team A it'll be team B so another team sit here doesn't matter which team sits another team sits on this side नहीं A B this side you remain. So you mark D. E कहाँ बैठेंगे? A, B, C. Okay. Can you sit a bit in the middle? Can you shift 2 seats to the? So give them 2 mics to this thing. So we'll distribute the rounds. We'll start with the first round. A, B, C, D. Easier. It goes clockwise. Team. So I'll request the teams to please introduce yourselves. The mics will be given. We'll start with Team A. Just tell us your name and institute for the audience.
Good afternoon, everyone. I am Keshari Kumar Srivastav, and I'm a PhD scholar from Naipar Mohali.
Okay. I am Vishnu Sai Bire. I am from Manipal College of Pharmaceutical Sciences. I'm a research scholar.
Okay.
Good afternoon, everyone. My name is Sanjeevan Gupta, Master's Second Year student at Naipar Mohali.
Okay. So we have Naipar and Manipal Institute combinations. Welcome and sit down. Team B, can you quickly introduce yourselves?
Good afternoon, sir. Myself Nivas Harge, and I am a student of UIPS, University Institute of Pharmaceutical Science, Punjab University.
Okay, so home team. So, good support.
Good afternoon, sir. My name is Yogesh Jadhav. I am from myself Yogesh Jadhav, UIPS, Punjab University.
Good.
Hello, sir. Hello, sir. I am Abhishek Akolkar from UIPS department. I am from first year student.
So home team, lot of support from the gallery. You're under pressure. Team C.
Sir, I am Komal, master second year student from Chaudhry Bansilal University.
Okay.
Good afternoon. Myself Sampriti, Masters Pharmacology final year from UIPS.
Okay.
Good afternoon, sir. Myself Meera Kumari. I am from UIPS, Punjab University.
Okay. Home team.
Good afternoon
Team
to
D.
all. Good afternoon to all. Myself Jagdish, and in my team, she is Mansi, and he is Rohan Badange. We all three are from BITS Bilani, Pilani campus.
Okay. Welcome. So BITS Pilani, full team.
Good afternoon all. I am Shivam Verma from Guru Gobind Singh College of Pharmacy, Yamunanagar.
Good afternoon all. I am Chanchal Mittal from Naipar, Kolkata.
Okay.
Good afternoon all. Myself Kunal from Gurukul Kangri University, Haridwar.
Okay, so this is a mixed team. So we'll start with the quiz without further ado. So let me introduce myself. I am Professor Abhijit Hajra of the Department of Pharmacology, Institute of Postgraduate Medical Education and Research, Kolkata. With me is Professor Nitin Kothari. He is from Government Medical College, Dungapur, Rajasthan. And I'll request our scorer and timekeeper to introduce themselves.
So, hello everyone, myself Vansh Kamboj, and I am from UIPS department PU, and I am a bachelor second year student. Thank you.
So, good afternoon everyone. Myself Ritik Sapru, and I am from UIPS, and I am currently in B Farm 4th year. Thank you.
Hello, hello. Okay. So we are starting the final round of the quiz, and this being the IPS platform, we can call it the national quiz. I think we deserve this epithet. And as you know, a quiz is meant to be fun. It's not just hardcore knowledge. It's a lot about GK and what you can recollect, your responses, etc. So we have made this final round audio-visual. There are 5 teams, and at the beginning there will be 8 or 9 rounds, and at the beginning of each round I'm going to explain the rules. The rules will differ a bit from round to round. Some of the questions will be passing and some of them will not be passing. So we'll explain at the beginning of the round. And can you put it on? So we require both halves of your brain, left and right, to address this thing. Okay. So welcome to your hot seats. Get some adrenaline pumping into your system. For a good measure, add some cortisol too. Okay. Let your heart beat faster. And I'll request Nitin to start with the first round.
Thank you, sir. So be ready for the first round.
Just anyone. You're not started.
Code
yet?
blue, code blue.
Code blue, good.
This is also warm up.
So warm up round. Just have some straight questions to set the ball rolling.
So these are the rules for the warm up. Each slide presents one state question, and you will have to answer in this 30 seconds. 10 points for a correct answer and no deduction for incorrect.
So
Question.
Please note 10 points and 5 bonus points. And time allowed for the first team. Whose direct
Is
question 30 seconds.
30
bonus
seconds.
round they must.
Audience are please must not prompt, and violators will be given marching orders.
We'll have an audience question for each round. Okay. I don't know whether they are chocolates, but I'm told you can take an extra helping of dessert at lunch.
Identify this drug.
There are clues on the slide. Team A, your direct question. Seconds, huh?
Hello. Indomethacin.
Incorrect. Passes to team B. Team C. Team D.
T3.
Hmm?
Triiodothyronine.
Because you can see iodine out there. No.
Last.
Team E. No. You will get to participate as an audience now. Okay, passes to the audience. See, it has a polymeric structure and there is iodine. Yes, povidone iodine, polyvinyl pyrrolidone iodine. Okay, so nobody got it? Okay. Team B, your direct question. These two gentlemen received the Nobel Prize last year. So what did they do? Seconds. No, you don't have to. Just say when the time up. Time up. Team C. Team D. You can guess, there's no penalty points. Team E. And that's written down there. What did they do? Team A.
Something related to miRNA.
Not something related to mRNA, for the discovery of microRNA.
MicroRNA, yes.
So it seems that only 2% of our RNA is coding. The rest is all non-coding, and a large fraction of them are microRNAs, and now we are hearing of microRNA therapeutics. So we'll give 5 bonus points to Team A. So Team A opens with 5 bonus points. Team C, your direct question. Look at the peculiar cage-like structure. No, just say when time is up. Okay, just can you name at least one which is specifically targeted by this drug? No. What is this drug? Time? Time up. Okay, question passes to Team D. They have already given a hint. Team E. No, that's incorrect. Team A.
Sir, physostigmine. Physostigmine.
No. Team B. Okay, audience, this is an antidote for a neuromuscular blocker, as they said. And the drug is called Sugammadex. This is a specific antidote for which one? Not all neuromuscular blockers, two. Most importantly, rocuronium, and the other is vecuronium. Okay. Team D. Some famous discoveries in medicine have come from students. And perhaps the most famous student among them is sitting on your left. Team D, can you identify this student? He was in a medical school and he was still studying physiology. Time's up. Team E. Team A. Team B. Team C. For the audience, we'll show another picture. The student with his teacher. 1921. This is Charles Best. And earlier slides showed his friend Clark Noble, and this is Frederick Grant Banting, his professor of physiology, and their team made the discovery of insulin 1921 at the University of Toronto. So, Team E. You see, we don't like the title on this slide. Can you replace it by a more acceptable title? Pharmacological apartheid doesn't sound nice. Time's up. Team A.
The drug only works for whites.
Title. Can you replace the slide title by a better word? Team B.
There is a change in Palme patients. It is prescriptions, I think.
Instead of pharmacological apartheid, which would be a more acceptable term for this kind of variation in drug action? Team C. So what should it be called? Team D. Sorry? Climate, no. Audience? Pharmacogenetics or pharmacogenomics, either of them. So that's a more acceptable term, is the genes which are making the difference. Okay. So we have completed, I think. So one audience question only for the audience. You see a list of medicines. All of them are antibiotics, as you have correctly said. List is not very long. Yes. No, all of them are not. Exactly. Thank you. Congratulations. So this is the WHO aware classification. Access watch. This is the reserve list of antimicrobials. Okay. So we have completed round 1. So round 2 is around the world. Please listen to the rules. So we'll be visiting one location in different parts of the world, which is associated with something of pharmacological and medical importance. You have to do two things. You have to identify what is being shown and then identify what is its significance. If you get both parts of the answer correct, 20 points. If one part, 10 points. So please note, both parts correct, 20, or one part correct, 10. And you have total 60 seconds. Now, if you need a clue, within this 60 seconds, you have to ask for the clue. And I'll show you another slide which has the clue. But in case you make use of the clue, your points would be halved, 10 or 5. Got it? This question will not pass and there's no deduction if you are giving an incorrect answer. So you can take as many guesses as you can like within your 60 seconds. So team A, ready? Okay. So we are visiting the Amazon jungles in Venezuela. And we are looking at a plant. Fleshy leaves. If you want, you can ask for a clue. You want the clue.
Poison. Huh? Arrow poison.
No, what was the entity? Entity was a plant.
Digoxin. हाँ digoxin. Digoxin.
Digoxin? No, you still have some time. You said arrow poison. We'll say it's a dart poison anyway. Time's up. Okay. Audience. Curare. Right. And the entity, the plant? Anyone? It's Chondrodendron tomentosum, the curare plant, which is native to the Amazonian jungles. And from that plant, D-tuberculin was extracted. Natives use that extracts of the plant as on their poison-tipped arrows and darts, and it has made modern surgery possible. So significance is curare or D-tuberculin. Entity is the plant Chondrodendron tomentosum. Team B, ready? Now we are going to the deserts of North America. There are deserts in Texas, parts of Arizona, California, where you get this thing, this monstrous thing.
Sir, clue.
Clue. Here's your clue. What is that thing and what is its significance? Time's up. Audience. Not semaglutide, exenatide, okay? From the saliva of the Gila monster, exandine, and from which the first GLP-1 receptor agonist drug was developed, exenatide. We'll tell you what points to give. So none of the teams have scored yet. Team C, ready to visit another part of the world? You have gone to Madagascar, the largest island in Africa. And you see this plant with purple flowers. In fact, they also grow in India. And sir is saying in this very campus, you can see it. Please, just be a little louder. I can't hear you. Clue. Okay, you have told the significance. And what is this plant? No, second part is done. You just name the plant. Okay, no, they have said it. We'll take it. Catharanthus roseus. Common name is periwinkle plant. We have the Indian variety. This is the Madagascar variety, which actually has relatively high concentrations of vincristine, vinblastine, and is extracted from that. Okay. So we'll give 20 points to team C. Well done. Okay. No. Oh, acha, they took the clue. Sorry. They'll get 10 points, not 20. They made use of the clue. So team C gets 10 points. Next, team D. You're in Boston, heart of the city. And you see a dome. Clue. A dome-shaped structure and a visualization of certain events which occurred there.
Dopamine?
No. Events occurred on October 16, 1846.
Clinical trials.
Hmm?
Clinical trial. Initial.
Audience. No, not first time GA, first time ether anesthesia. So till date, anesthesiologists celebrate October 16th as the World Anesthesia Day or the World Ether Day. This is the ether dome, the OT where ether anesthesia was first administered by William Morton. And this is an enactment, a role play of that event. Okay, and they have preserved it like a museum, the Massachusetts General Hospital. So it goes to team E now. Your question. We have traveled to the famous town of Oxford in England, particular hospital, and you can read out the name of the hospital.
Sir, clue.
Clue. Your clue is a petri dish. Penicillin, okay. What else can you say? What else can you say about this? Who was the hero of the story? Alexander Fleming. So Alexander Fleming, his discovery of penicillin occurred in a St. Mary's Hospital in Oxford. And we all of us know the story. Penicillin was the antibiotic which started the golden age of antimicrobial. So they get 10 points because in a clue. So audience, do we have a question for you? A plant growing like a weed. It's small. You can see it comparison to a coin. No, everything from China is not Artemisinin. You go back. Autonomic pharmacology. No, ephedra. Ephedra. This is Ma Huang, Ephedra sinica, from which ephedrine, the first sympathomimetic, was derived. Okay. So we'll go on to third round. So this is the structure of ephedrine. Path breakers. Okay. You can see Dashrath Majidar, the Gelot district in Bihar. And this was the man, he, with his shovel, he shoveled for 20 years to make a path through this rocky outcrops. His wife died because he couldn't reach medical facility on time because this was obstructing and nobody was doing anything about this. Pleas to the government officials fell on deaf ears, so he took it upon themselves to make a path. So he was a literal path breaker for 20 years. But we have many path breakers in medicine, team A. So each slide will present one accomplished individual, a path breaker in medicine. So a pharmacological event is associated with that individual and the slide title will provide a clue. Okay, so you have 60 seconds to name the person and the achievement. 20 points or 10 points. Okay, this question will pass to the next team. Time is 60 seconds. 20 points, both parts, 10 points, one part. If it passes, 10 and 5. Team A, ready? Everything is written out there. History records this as the only clinical trial done on a ship. Clue? Okay. It's the answer.
Huh? Vitamin C first. Vitamin C first.
Vitamin
No,
C.
I won't give it to you if you say vitamin C. Vitamin C was discovered much later. Person and the event. Team D. Team E. Audience? Yes, James Lind on H. She has also named the ship. Thank you, HMS Salisbury. And you read about how 12 sailors were exposed to different things, including seawater and ginger ale. That's an alcoholic drink, and lime. Okay, and the only 2 sailors who survived were those who were put on lime. And from that day, lime became a standard ration for sailors on long sea voyages, and British Navy men actually used to be called limeys. So Albert and Georgi discovered ascorbic acid as the active principle much later on. So this was question to team A. Team B, your question. There are 2 individuals here. The first chemotherapeutic molecule which they discovered. You can ask for a clue. Clue. If you can't say this, I'll give you minus point. Time's up. Team C. 2 persons. Team D. Team E. क्या हो गया? ठीक. So Louis Goodman and Alfred Gilman already written out there. The one on the left is Goodman and this is Gilman. Okay. The molecule was meclorethamine, the first of the nitrogen mustard class of molecules discovered as an alkylating agent. Before that, nobody believed that drugs could, I wouldn't say cure, but drugs could address cancer. This was the first molecule which made Hodgkin's lymphoma literally dissolve the tumors, but later on they came back. Okay, but that was the beginnings of cancer chemotherapy. Team C. So they get bonus points, 5. This is pathbreaker from Madras Medical College. No, R.N. Chopra was not from Madras Medical College. You still have time. Clue. Yes. You want to see the clue slide? Okay, it's written. So that man is, time's up. Okay. Next team. What did he do? Team D. Why is he a pathbreaker? Else? Okay. Yes, okay. So we'll give that team, it was their direct question. So we'll give them, no, 10 points. They use the clue. 10 points. And we'll give them 5 bonus points. So Subbarao has a distinguished career. He was behind the discovery of adenosine triphosphate. He and his team, okay, in the Lederle laboratories of American Cyanamide Company, they were instrumental in discovering methotrexate and other drugs. Okay, their efforts were behind diethyl carbamazine and tetracyclines also. Okay, so worked as a treatment. Aureomycin, the first tetracycline which was introduced. So whose question is it? Team D. milk has many benefits, direct and indirect.
Louis Pasteur.
No, that's not Louis Pasteur.
Clue.
Clue.
Smallpox vaccine.
So the significance is smallpox vaccine. And who is this doctor?
Jenner. Sorry.
Okay. So Edward Jenner?
Edward Jenner.
Okay. So you know the story of the milkmaid. So when the smallpox epidemic was raging in rural England, Jenner found a milkmaid who was not afraid to go into the houses. And when he asked him, she replied that she was not afraid because she already had cowpox, so she will not get smallpox. So that led Jenner to theorize and introduce the concept of vaccination. So that is Edward Jenner, and we all know the story of the smallpox vaccination. But the vaccine did not come in a day. He had to fight the establishment for almost 20 years before it was accepted in his own country, vaccination. Okay, so we'll give them full 10 points. No, they use the clue. They use the clue. 10 points. Team E. This you should be. So this lady at a much younger age, and when she won the Nobel Prize, 2015.
Clue.
Clue.
Artemisinin, sir.
So artemisinin, discovery of artemisinin from the plant Artemisia annua, Chinese sweet wormwood. And who is the lady? You still have some time? Okay. Team A, B, C, D. Chinese scientists, they're saying. 2UU, madam, 2UU. So we'll distribute 5 points. We'll give 5 points to team E, and they use the clue. And part of the answer they got, and we'll give 5 points to team C. Team C. Now this is for the audience. Which one? You have to be specific. She wrote a book where she told Americans that soon you'll be waking up in spring without the sound of robin, swallows, and other birds that you associate with springtime, and all because of an environmental toxicant, DDT. So this is Rachel Carson, and her book raised a huge hue and cry, and this led in 1957 to the establishment of Environmental Protection Agency US, the first environmental protection agency in the world. So this was a path-breaking effort. She herself was an environmental scientist, okay, Rachel Carson. So we have done 3 rounds. So can we have a roundabout of the scores?
So the net result is A team has 10 points, B has none, C has 25, D has 10, and E has 20.
So team C is leading at this stage, but it is, you don't need to get excited. There are a lot of things. So this is the Hyde round. After Dr. Jekyll and Mr. Hyde, you know drugs can be poisons, and Robert Louis Stevenson's novel. So we'll be addressing the Hyde components of some drugs. Team A. Okay. So it's on a toxicological matter. You have 45 seconds to identify and state the significance. I can provide a clue, 20 and 10 points, no passing in this round. Okay, 45 seconds, right? 20 and 10 points. Team A.
Ricin.
I didn't ask the question. Okay. What's the significance of ricin?
It is often from that seeds, castor that
Comes from?
that castor seed.
Ah. See? They were caught with 4 kg of castor bean mash.
Do you want castor scientific name something? Castor scientific name, are you asking?
Say it.
Ricinus communis.
Ricinus communis. And why? What's the importance of ricin?
Castor poison. I think poison.
What kind of poison? Neurotoxin, cardiovascular toxin, what? Time's up. Okay, they have said ricin and they have said it's poison, we'll give it to them. So ricin is one of the most powerful plant-derived toxin. It comes from the, show the next slide. It comes from the castor bean, and this plant, Ricinus communis, grows wild over India. So I had gone for ISRPTCon in October, where I clicked the picture from the hostel campus of SCB Medical College. I am sure it will be growing in Chandigarh also. Okay, it's found wild all over India. It's called erende in many Indian languages. Okay. From the castor bean, the oil is pressed, and from that bean, the ricin toxin, it's class 1 biological warfare agent. Okay, if you can mix a little of it with a water supply, many people will be poisoned. So we'll give them full credit, 20 points. Well done, Team A. Team B. This is showing the ECG of a patient who has consumed this flowers as a poison. In forensics, it's very commonly taught. If you can identify the abnormality you can see in the ECG is good, but at least identify the plant.
Sir, arrhythmic drug.
No, I'm saying plant.
Plant.
What plant?
No.
You can ask for a clue.
Clue, sir.
Oh, time's up. Okay. This question does not pass. Audience? No. Thivesha, yellow oleander. It's another plant which grows wild, and it is planted by municipal corporations because of its beautiful elongated yellow flowers. It has digoxin-like effect. And what you are seeing on the ECG is a complete heart block. Okay. Not torsades. This is complete heart block. Okay. And that occurs because of these glycosides. Glycosides like molecules, pyruvoside and its allied compounds, which have digoxin-like effects on the heart. Basically H plus K plus ATPase channel poison. Na plus. Yeah. So no points on this question. Team C, your question. It's a North Korean leader is very powerful, Mr. Kim Jong-un, and his estranged half-brother, Kim Jong-nam, was fatally attacked. Fatally attacked means they just went behind this poison and they were told rub his cheeks with baby lotion. And he'll get paid for that. And poor things, they were not aware that the people who paid them were North Korean agents. Clue. Time's up. Audience. Which one? Tabun, sarin, samun, soman, they are gases. This I told you, they were killed by rubbing the cheek. He fell ill in the airport. Doctors tried to revive him but couldn't. So this is called the VX agent. VX, just a venomous agent developed in Russia, probably stockpiled by Russians and other countries. It's regarded as a chemical warfare agent. It's a very, very potent neurotoxin working in the same mechanism as organophosphorus poisoning. Percutaneous absorption is very high. So rubbing the cheeks kill. Team D, your question. This is from Japan. If you want to eat this fish,
Clue,
your
clue.
cook. You want a clue. The clue is already there. Okay, we'll give you the clue.
Sir, polonium. Sir, polonium.
If you just say polonium not accepted.
Polonium.
210. So what does it do? नहीं Time's up. So audience? It's a highly radioactive substance and very small quantities can cause fatal radiation sickness. So this poor Russian ex-spy was living in London and he went to meet his friends in a restaurant and the friends were also Russian spies. So they slipped, it is believed, small quantity of polonium-210 in his tea and he died of radiation sickness. Doctors couldn't diagnose him till the last moment, but by that time he was late and he died in the University College of London in intensive care. So that is the teacup murder. So all 5 teams done. Audience, do you want a question? Okay.
Marks.
Scores? They have said polonium-210, so we'll give them 10. They have not used the term radiation sickness or radiation illness. All you see the fluorinated general anesthetics, enflurane, desflurane, isoflurane, sevoflurane. Which one is destined to be phased out? No. The answer is desflurane. Okay. Because of its environmental toxicity. It is considered as a greenhouse gas. Consumption of 1 bottle in the OT is equivalent to burning 450 kg of coal. And the global warming potential is 3,700 times that of carbon dioxide. So it's been decided that it will be phased out from human use. Desflurane. So next round. So this is about natural things. Okay. Prior to the 19th century, all our remedies, basically all our remedies were dependent on plants. And you can see this was the medicine chest given to David Livingstone when he went to Africa. And this chest was made by him for Burroughs Wellcome and Company, that company which still exists. And all the ingredients, all the medicines, they put him in his first aid medicine kit were plant derived, 1857. So not long ago, we were dependent on natural things. So each slide is on a natural entity. Just have to identify what it is and its significance in 30 seconds, 20 and 10 points. Okay. But time is short, 30. Okay. It will pass. We'll pass it quickly. Team A. I think we'll reverse the round. 4 rounds you have completed. Team E, you start. दे दो क्लू. Say which plant?
Tobacco plant, sir.
Tobacco plant. Botanical name?
No.
Time's up. So will significance already was there in the next.
Is it Nicotinum tabacum?
Nicotiana tabacum. Nicotiana tabacum, but they have identified the tobacco plant. See, it's cultivated, but it's probably the most dangerous plant in the world. Okay. So they will get 10 points. No, we have not passed that question, this thing, because the significance is already said. We are not asking for the botanical name, just the name of the plant. Clue was given?
Clue was given.
So, so they named both the thing, na? Nicotine and the plant. So 10 they are getting,
Without
not
clue is
20.
20.
Without clue is 20. So team D, your question. This is a large fish.
Omega?
You want clue?
Yeah.
Okay. One is named, other is not named.
Protamine sulfate.
Poofa? Poofa?
What fish? So one drug derived from this fish is protamine sulfate, the antidote to heparin. Time's up.
Sir,
Team C. Salmon. Specifically the Norwegian salmon, very large fish. Okay. And the significance, one is protamine, the other? You spray it like this. Okay. No.
Sir, it is antidote. Protamine sulfate is the antidote of.
That's already done. You said.
It is the antidote of heparin.
We have already said that. So they have said protamine. So I think after the clue they can get 5 points, and Team C also gets 5 points. The first drug is a peptide, as you can see, and the clue is it is given intranasally, and it is derived from the salmon. What drug is it? Salmon calcitonin. Okay. In osteoporosis, though it's no longer that much recommended. So these are the two fishy polypeptides. Next question to Team C. This is a little bit difficult. Clue. Time's up. Team B, would you like to add anything?
Is it PCN 4126V?
Sorry? That is the code number of the chemical you are reading out. No. Pass. TB. So alpha conotoxin, what's its significance? Team D. So this is the Pacific cone snail. It's a large snail. Because of its cone shape, it's called a cone snail. It's a source of different types of conotoxins. And from the alpha conotoxin family, we have developed one analgesic drug, ziconotide, which is given intrathecally, so it's not become very popular. But it's a potential source of natural analgesics in future. So significance is conotoxins. So nobody gets any point on this round. Team B, your question. Drug honored by a postage stamp. You don't find postage stamp honoring drugs.
Sir, clue.
So many clues are already there. You still need a clue. Okay. This village is in the Kalimpong district of Bengal, known for its cultivation of something. Time's up. Team A.
Cinchona.
Drug?
Cinnamon.
Cinnamon, cinchona, not the same thing. Drug.
Syncotanic acid.
Sir, quinine.
Quinine, okay. So quinine is obtained from the cinchona bark still. It can be chemically synthesized, but that is complex and won't be cost effective. So it is still derived from as a natural agent from the cinchona bark. And there were so many clues on the slide. There was a structure of quinine. You can see the malaria parasite within the red blood cell and the two pharmacists, Pelletier and Cavendish in France, who had extracted quinine from the cinchona bark. Okay. Next question goes to, they will get 5, and they will also get 5 for telling cinchona as this thing.
Sir, we said
You said Syncona. So 5 will give it to them, and the drug was named by team E. So they get 5. Whose question now is it? Team A. Yeah. So nice fruit. You get this nice looking juice from it. What is it? What's its significance? Clue.
grapefruit juice.
Say it again.
Grapefruit juice.
Grapefruit juice, not grape juice. This
Grapefruit
is
only,
grapefruit.
grapefruit juice.
Grape and grapefruit are separate. These are not grape. This is grapefruit, as you have said. And why is this juice important?
It is CYP3A for inhibitor.
It's a strong CYP3A4 inhibitor, just like you have many other inhibitors and inducers. So earlier it's not used to be a problem, but now if you go to malls, you'll get tetra packs of many juice: lychee juice, mango juice, grapefruit juice. And if you're in the habit of drinking it, you have to be careful. So they get full 20 points. So well done. Sorry, they used a clue, so 10 points. 10 points, team A. Audience, would you like one question? Okay. A mushroom. No, Amanita phyllodes is not colorful. It's drab, brownish. Anyone? This bright red-looking mushroom, very characteristic. This is Amanita muscaria, the original source of muscarine, which opened up the field of autonomic pharmacology. Okay. Next round. Score. Okay.
Tie between A and E, 45-45, B 5, C 30, D 20.
Okay, so team B. D20. Okay, so team B, you need to buck up a little bit. Okay, so pharmacological geography. Each slide will present a map with some medical or pharmacological significance. This question will not pass. 30 seconds, 10 points. Okay, now this one has a negative marking. Okay, minus. So at your own risk. Ready? Team E. Team E, you are ready? Okay. (2025) WHO data. Time's up. Audience? Yes? BCG? No, BCG is worldwide. Anyone? Take a guess. There's no smallpox vaccination now in the world. Cervical cancer. This is the human papilloma virus vaccine. Okay. Not yet part of the India's universal immunization program. So it's not routinely administered in India. So very good. This is the papilloma virus, but nobody gets a point on this. Team D. Ready? So if you guess, you have to be careful. And you can see a golden triangle and a golden crescent. Unfortunately, India is sandwiched between the two. And the ones in red are the source countries. Time's up. Audience? Opium. The opium cultivation areas of the world. Okay. And also the major opium consumers, I would also say. But India is being affected because it is sandwiched between the two. Okay. So, Team C, your question. Not the whole world, only a part of it. No. So we have to give them, unfortunately, -5. Okay. Audience? No. See, only part of the world, Southeast Asia and parts of Oceania. Mosquito-borne disease. Not dengue. Affects the brain. JE. This is the Japanese encephalitis vaccine. Okay. So most of the world is not concerned with it, but we in South and Southeast Asia are involved. Whose question now? So they have, unfortunately, Team C has 5 negative points. Team B. See, a drug on which the world is divided. Some countries it is legal, some it's legal only for medical use. In some countries, like in India, it is illegal. Although it's an Indian drug. Traditional knowledge. Time's up. Okay, audience. Cannabis. Okay. Cannabis from the cannabis sativa plant, which is native to the Indian subcontinent, very useful plant, top to bottom pharmacological importance. Unfortunately, the Indian government doesn't allow it for valid reasons. Okay. Team A, the last question in this round. Clue, there's no clue here. Clue is in the, whatever is written on the slide. Time's up. Audience? Leishmaniasis. Good try, but it doesn't occur in so many countries. Much more common than leishmaniasis? Yes. Who said that? Okay. Congratulations. This is resistance of falciparum malaria to chloroquine. So it's a chloroquine resistance map, updated, not fully updated, but more or less the recent times. You can see there are resistant maps of many things, and that was the chloroquine resistance map. Okay, so one question for the audience. Take a guess. Very high in Mongolia and parts of Africa. Higher in China than in India. No. 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 Sleeping sickness won't be there outside Africa. Communicable only? No. Which is the largest organ in the body, apart from the skin?
Liver.
Liver.
Liver cirrhosis?
Which one?
Hepatitis C virus.
This is hepatitis C map.
C,
This is hepatitis C map. And the treatment of hepatitis C has become complex with genotyping of the virus. Okay? So how much time do we have? We have to close now. So, okay. So, this round is pharmacology in art. Okay. Sometimes artists have drawn something of pharmacological significance. And each slide will present a famous painting with some pharmacological significance. You have to identify both parts in 60 seconds, 20 and 10. Okay, 60 seconds, 20 and 10. No separate. Okay, I'll give clues for everybody. This question will pass. Okay. This is a painting from the Renaissance era. It's a painting by the French painter Jacques-Louis David. And it is actually showing a person committing suicide. Team E. Can you name the painting or its pharmacological significance? There's a clue. It's showing a famous Greek scholar committing suicide. And he has asked his student to give him the cup containing the poison by which he is going to commit suicide, and the student is very regretful and reluctant, but still he is following the teacher's command. 60 seconds.
Eye irritating.
No. Hemlock poisoning. So who is the person? No. Hemlock is the plant. The Greek philosopher? No, Greek philosopher ka naam. Painting is named after him. Name all the Greek philosophers you know. We'll give the teams a chance. Okay. Team A?
Aristotle.
Oh, good try. No, this is not Aristotle. Audience. Socrates. So, he is taking a cup of hemlock. Hemlock contains the poisonous compound aconitin. Conine. It contains the poisonous compound. It's called Conius maculatus, the plant. And the conine is the alkaloid, which has an action like it initially stimulates the parasympathetic system and then stops the release of acetylcholine, causing fatal and flaccid paralysis. So painting is the death of Socrates. No, we are into this round. Let's complete this round. We'll give them half the points. Okay. No, this was 20 and 10, na?
20 and 10.
So they'll get 10 points. 10 points. Team. No, maybe. Sorry, this question was for Team A. So this was their. So this was their. They got it on the pass. So they'll get 5. They'll get 5. Okay, make correct. So Team D, your question.
This is the question. So that was the question.
No. This is the question. This is actually an altarpiece. So what they put in a church. So it's a sculpture which was later painted over, and you can see the details of this painting and who painted it. And this is dedicated to a particular saint. Okay. And he is the patron saint of a disease. Disease which causes necrosis due to peripheral vasoconstriction. Altarpiece is already said. I just want you to identify this person. Is a Christian saint. A disease is named after him, and that disease is of great pharmacological significance. Anyone? Team B? Team A? Okay. Team E? So these are grains of barley, and some of them are infected. Yes. Saint Anthony's fire. Okay. So Saint Anthony was the patron saint of the peasants who used to suffer from ergotism. Ergotism is due to claviceps purpurea infecting and spoiling grains. And if you eat these contaminated grains, you'll get ergot-like compounds which can be powerful peripheral vasoconstrictors and cause associated pain, something like Berger's disease, that the pain that we get. So Saint Anthony's fire, the disease used to be called in medieval times. A bit difficult question this was, I agree. Next question 2. So again in Greek mythology, there are 3 sisters. Okay, just listen to the clue that I'm giving. 3 sisters. They are the 3 fates which determines a person's destiny. One of them spins the thread of life. The other winds the thread of life. And the third one, in the center, fearful looking, cuts the thread of life. Okay. So identify the middle sister in this painting. No. You still have time? Time's up. Oh, don't you? Team B, Team A, Team E, Team D. Clue for everybody, audience. Yes, so this is Atropa belladonna, the plant named after this third sister, Atropos, who cuts the thread of life. So that's a poisonous plant, but we know that atropine can be a life-saving drug as well. And this 3 sisters, the 3 fates, is a recurring theme in classical Greek mythology. You'll find it in paintings, in sculptures, etc. So the 3 sisters, this is Cloto, the word from which we get cloth. So Cloto is spins the thread of life, Lachesis winds the thread of life, and Atropos cuts the thread of life. Okay. Question now to team B. This is a famous painting by a famous artist called Salvador Dali. He's a Spanish painter. One of his most famous paintings, and what is its medical significance? Go to rapid fire.
Sir, chronological disease?
No. You still have, I think, some time left. 30 seconds. This is associated with the painting. Time's up. Team A.
Sir, is there any clue for this?
Already shown.
Sir. So negative marking is there?
No. But you're taking too much time. Team E. Team D. Team C. Good, very good. This painting is called Persistence of Memory. And the way you have drawn this painting, it does not represent real life. And Salvador Dali said that he could. I'm explaining the significance. He could paint it only because he consumed this thing. This is a hallucinogenic cactus, the Mexican peyote cactus Lophophora williamsi, from which you get LSD-like drugs, psilocin, psilocybin. You can also get it from other cacti, but Salvador Dali used to consume this cactus, and he used to go into a high when he painted some of his most famous impressionist paintings. So very good, persistence of memory. So they have a bonus point, 5. No, I didn't give the clue, but now I'm seeing these questions are becoming difficult, so I'm offering clue for everybody. You got a point on that, na? Okay. So last question in this round goes to Team A. Now this is a famous painting by an American artist called Robert Thom. He was asked by Park Davis and Company to paint 50 portraits on milestones in medicine, and one of them is this. This is showing a famous surgeon doing something to a boy patient. So what is this painting?
Plastic surgery?
No. It's of pharmacological significance.
It's a clue.
No clue as such. I've already told you. This is a famous surgeon. 19th century English surgeon. Father of something. Time. Time's up. Team E, any guesses? Team D? Team, yes. Team C?
It's a surgery.
Who is the doctor? Team B? Now for the audience, I'll give this clue. Joseph Lister, the father of antisepsis. And this painting is showing that compound fracture, Lister had splintered and bandaged him, and in those days a compound fracture was almost like a death sentence, but Lister has soaked the bandages with phenol, carbolic acid, and after 8 days when he opened, the wound was healthy. So that led Lister to introduce antisepsis with phenol, carbolic acid spray, and you can see a replica of his instrument. Okay, so Joseph Lister. So we'll just have one question for the audience, and then we'll move to the last round, rapid fire round. Audience question. So this question had gone to Team A, and nobody had said it. It was the audience which has said it, so no points on this. No, this painting, did you say? Previous one you got marks, no? This was this painting. No. Are you referring to the previous question? Persistence of time? Previous to that? Did they say carbolic acid? No. This you are going forward. Team A, but nobody said this. And you didn't answer, na? The birth of antisepsis, the painting is called. What did you say? You didn't say anything. Previous question? They got points for that, na? You said persistence of memory. You got points for that. That was given. Nobody gets a point for this question. So this is easy. This is almost like a repeat question. Audience. What is this painting called? This painting is called The Birth of Ether Anesthesia. It's a painting which is still there, and it is celebrating Morton. You can see his signature, W.T.J. Morton, William Thomas Green Morton, and a version of the vaporizer that he developed for administering ether. So we'll have a quick roundup of scores, and then we'll go to the final rapid fire round. We'll skip them. A and E are tied. And B and C, what are the scores? D20. Okay. Now, this is going to be a rapid fire round. Please note the rules carefully. So, you select the person who will respond. Others in the team can help the person, but I'll accept the answer from only your spokesperson. Right? Okay. In 1 minute, I'll ask you maximum of 10 questions. You get 10 marks for a correct answer, no deduction. Right? We'll go back. We'll start with you only, A. If you are not able to answer a question, then you have to say pass. Unless you say pass, I won't move to the next question. Either answer. If you answer, I'll say whether you are correct or incorrect. They'll keep a record of the correct responses. And if you say pass, then only I will go to the next question. There's no clues. Just direct questions, simple questions. As many questions as you can fit in 1 minute. We will not go back to the unanswered questions. Right? Who will be your spokesperson? Okay. So ready?
Yes.
Let me handle this. Moresky scale is for measuring what?
Pass.
Explain the S in 30S ribosome.
Subunit.
Correct. What is the location of National Institute of Biologicals?
Pass.
Expand the abbreviation HAM.
Pass.
Amyloid plaques and neurofibrillary tangles are the hallmarks of which condition?
Alzheimer's.
Correct. Why is Autoloy famous?
Pass.
Oral bioavailability of metronidazole is?
Pass.
True or false? Nusinersen is indicated in spinal muscular atrophy.
Yes. True.
Correct. So I'll take that. So they could attempt or pass 8 questions, 3 correct responses. So they get 30 points on the rapid fire round. Okay. Team B, who will be your spokesperson? He'll be the spokesperson? Okay. I'll take responses only from you. So your questions coming up. Garfinkel algorithm is for doing what?
Pass.
Explain the 450 in cytochrome P450 enzyme.
Pass.
What is the location of the National Center for Disease Informatics and Research?
Delhi.
Incorrect. Expand the abbreviation LAST.
Pass.
Philadelphia chromosome is the hallmark of?
Pass.
Why is Austin Bradford Hill famous?
Pass.
Time's up. So they could answer or pass 6 questions and they don't have any correct response. Team C, phone down. Okay. But eyes and ears and brain open. Phone closed. Okay. So they have not got any correct responses. Team B. Team C, your questions coming up. Ready? Who will be your spokesperson? Please give them a mic. You have it. Montgomery-Ashburg scale is for rating what?
Pass.
Explain the A in CPDA.
Pass.
What is the location of the National Institute for Research in Bacterial Infections?
Pune.
Incorrect. Expand the abbreviation DDD.
Pass.
Tremor, rigidity, bradykinesia is the classical triad of which?
Parkinson.
Correct. Why is Rudolf Buchem famous?
Father, he first established experimental pharmacology laboratory.
Correct. Accepted. Shadox 1 NCOV19 is popularly known in India as? Okay, you can still answer.
COVID-19 vaccine.
No, incorrect. No, not Covaxin.
Covaxin.
No, it's Covishield. Okay. Anyway, so they could attempt to pass 7 questions. They got 2 correct responses. So 20 points they have scored on the rapid fire round. So team D, your spokesperson? Okay. Your questions coming up. Womack scale is for assessing clinical severity of what?
Pass.
Explain the M in 99M technetium.
Molecular weight.
Incorrect. What is the location of the National Brain Research Center? Incorrect. Expand the abbreviation CAR T cell.
Kinergic antigen receptor T cell therapy.
Okay, accepted. Antibodies against the nicotinic acetylcholine receptor is found in which condition?
Myasthenia gravis.
Yeah. Correct. Why is Carrie Banks Mullis famous?
PCR.
Correct. Henrietta Lacks is immortalized as?
Pass.
Time's up. So they have got 3 correct responses. CAR T cell, they have said myasthenia gravis, and they have said PCR. Okay. So 30 points for team D. Team E. Okay. Your questions coming up. Who is your spokesperson? You are. Ramsay scale is for measuring what?
Pass.
Explain the GY in radiation dosimetry.
Gyration.
Incorrect. What is the location of the National Institute of
Pune.
Correct. Expand the abbreviation DHFR.
Dihydrofolic acid reductase.
Try again.
Dihydrofolate reductase.
Correct. Benz-Jones proteinuria occurs in which hematological condition?
Pass.
Why is Barry Marshall famous?
Pass.
LAL assay is a test for?
Pyrogen testing.
Correct. True or false? Trientine is indicated in Wilson's disease.
Pass.
What is the other name for acetaminophen?
Paracetamol.
Correct. What is the generic drug in Botox?
Botulinum toxin.
Botulinum neurotoxin. Accepted. So 5 correct responses they had. So they score 50 points in the rapid fire round. So while our scorers will tell us the scores, but before that, let us all give a big round of applause to all the participants in the final round as well as the previous round. Okay? So scorers, are you ready? So please tell us that you can come here. Or you have a mic, na? Mic nahi, idhar aa jao. So that's what Sir Stephen Hawking said. We are an advanced breed of monkeys on a minor planet of a very average star. But we can understand the universe. That makes us special. Okay.
So the lowest marks of I'll go chronologically. So team A had marks 75, team B had 5, team C 55, team D 50, and team E 95. So therefore the first team is team E, the second team is team A, and the third is team C.
So fairly closely fought round, and we need to keep this record.
Yeah.
Okay. So the winners are Team E. You can announce your names again.
Sir.
Just tell your names.
Shivam Verma.
Chanchal Mittal.
Kunal Rana.
Okay, so they were a lost team. Only one isolated, not finding partners. So we clubbed them together and see what mixing can do. Okay. And the first runners up, Team A. Team A, just tell us your names again.
Vishnu Sai Bire from Manipal College of Pharmaceutical Sciences.
Keshri Kumar Srivastava from Naipar Mohali.
Naipar Mohali, and he's also from Naipar.
Sanjeevan Gupta from Naipar Mohali.
Naipar Mohali. Okay. And Team C, you are the second runners up. So we have 3 awards: the winner, the first runners up, and second runners up. So Team C, just tell us your names again.
Meera Kumari from UIPS Punjab University.
Okay.
Sampriti Dev from Punjab University.
Komal from Chaudhary Bansilal University, Bhiwadi.
Okay, so 2 from the home institute, and the other one also from Chandigarh. So thank you, everybody. I hope we have enjoyed the quiz. And now they're asking us to break for lunch. Thank you. इसको नाम लिख रहे थे ना, देने होगा.
Sir.
Winning teams, just come here once. Team C, Team A, and Team E once. Just come here once. और नितिन सर को नाम और एक बार बता दो वैसे. We'll have to.
सर आ रहे हैं एक्चुअली.
मास्टर को बोल दो सर सेकंड छुट्टी दे दीजिए.
मैडम यहाँ पे, हाँ यहाँ पे डालना पड़ेगा।
ये आपको
आ गया है,
ठीक है,
वो यहाँ पे आ जाएगा ना, वो बाद में करेंगे।
तो मैडम आएंगे तो
मैडम
A very good afternoon to all of you. I welcome you all back post-lunch to IPSCON 2025. That is the last session for today. So now we are going to start with the keynote talk by Dr. Nilima Sagar ma'am, who's the Professor Emeritus of State Medical Sciences University, Maharashtra. See here. It's okay. आप अभी introduce करवाएंगे मैम को. I would request the session chairs to please come on the dais. I request Professor S.K. Kulkarni sir to please grace the dais as a chair. I would request Dr. Subhadeep Sinha sir to please grace the dais. Thank you so much, sir. I would request you to kindly introduce our keynote speaker to the audience.
What a pleasure I have to chair your session. Please come on. Betty. Good afternoon, everybody. Welcome to the last session of the 55th Annual Conference of Indian Pharmacological Society. We have an eminent pharmacologist and clinical pharmacologist of India, Dr. Nilima Sagar from Mumbai. Nilima ji was Dean of KM Hospital in Mumbai, and she was also, ICMR National Professor in Clinical Pharmacology for many years. And I have known her for almost 30, 40 years. And we had a very close interaction when I spent 4 or 5 years in Mumbai before I moved on to the other place. And Nilima ji is going to talk to us 50 years in pharmacology, lessons to be learned. I think lessons are to be learned even after 50 years. I have a great pleasure in welcoming Dr. Sagar. Stage is yours. You have about 25 minutes, I guess. Take your time.
Am I audible? Thank you very much. Firstly, at the outset, I am deeply grateful to Dr. Professor S.K. Kulkarni, my many years of friend and colleague, and Dr. Subhadeep Sinha, who are chairing this session keynote address. very grateful to the organizers of this wonderful Indian Pharmacological Society's annual conference, Dr. Anil Kumar and his team of people, Dr. Bez Badua and Shiv Prakash, Kala Kumar, Dr. Ramesh and Dr. P.L. Sharma who is here. Extremely grateful for you all to be here. And what I'm going to say today is actually, sir, more intended for students because these are the learnings which I have got actually from all of you here. But I thought that we are at the 55th annual conference and I've had, I think it must have been probably more than 55th, but whatever. And I thought that I will share with you 50 years of my experience in pharmacology. So these are the contents of my talk. I'm going to share some of the, am I audible clearly? Yes. I'm going to share some of the stories of the patients whom we have seen. Then I'll tell some anecdotes of the product development and clinical trials which we have conducted. And then working with various government and international organization experiences there. And the lessons learned. Actually, I'm going to intersperse in each of these the lessons learned, and at the end, I will have a sort of a summary lessons learned. Actually, this is what I did in the last 50 years, and as was famously put in one of the books, this is what I do. I thought that I should say as a message to the students, do what you like, but whatever you do, you should be able to tell your grandmother exactly what you're doing. She should understand what you're doing and she should say, yes, son or daughter, whatever you're doing is right and I think that's the right thing to do. If you can't explain to her what you're doing, go back to the drawing board and see what you're doing. So some short stories of patients. So here was this patient. He was given IV metronidazole for his post-operative antibacterial. This was many years back. That was the treatment given at that time. And a very astute physician, he noticed that the two of his patients had palpitation and dilatation of pupil. Now, when I'm putting it in that context, you can very quickly say that it is anticholinergic. But for him to have realized that this is anticholinergic effect, and he came to us asking, does metronidazole have anticholinergic effect? And we said, not to the best of knowledge that we have. And then he got the full box of metronidazole bottles that he had. And he said, now test these. When he says anticholinergic, it could be just anything. So the best thing that we could do was to put all these contents of each of these bottles into the rabbit eye. And indeed, 2 of the 10 that he brought to us actually produced dilatation of pupil in the rabbit eye. And then we found that the manufacturer of metronidazole bottle was also manufacturing atropine. And he had probably got the labeling wrong. So when we did the chemical testing, we found indeed it contained atropine. So we hats off to that physician who spotted this. So what is the lesson that we learned? To solve a patient's problem, you not only need clinical astuteness like this, but you should have knowledge and skills of pharmacology, clinical pharmacology. You should know the animal testing. You should know chemical testing, pharmacy, manufacturing. Well, we do hope that with the good manufacturing practices, we no longer have such mix-up. But we know we have been reading in the newspaper about the kind of mix-up that can happen. Next patient, an engineering student. He was referred to us. He was getting frequent GI bleeds. They wanted to know if any of the drugs he is taking is causing this frequent GI bleeds. When we asked him what he takes for pain, he says, I know that I should not be taking any anti-inflammatory drugs. I should not be taking aspirin. So out of curiosity, we asked him, what does he take then for his toothache or headache? He said, oh, I have got a wonderful Ayurvedic drug called Vedana Nigraha Ras, and it is very effective. So we were quite interested. So if it's Ayurvedic drug and very effective, let's test it. And we asked for the package. Now here's the twist. This was manufactured by Kanwar or Kovar Pharma Company in Lucknow. no aspirations on Lucknow, just located in Lucknow. And the only good thing about this fellow, the manufacturer, was that he had written on the package here for us what it was, and it was acetyl salicylic acid 500 milligram. Not surprising that he was getting good effect. In spite of him being so well educated and engineering student, he didn't know that acetyl salicylic acid is the same as aspirin. So I thought that was so many years back. Let me see what's happening today. So I hit ChatGPT, and this is what I found on today's 20th November. I looked at the ChatGPT, and it said Vedna nigraris is the trademark of that company. And then look what it says. The exact constituents of this are not given on the Google or anywhere. Lesson learned. Important to take careful history of the patient. Dr. P.L. Sharma, when he was taking my DM exam, he was very careful to see that I am doing the right thing. Patient history, critically important. And of course, you should know the other thing. Third patient, this, I've often quoted this example. Exam going student suffering from epilepsy during exam time. To enhance memory, he was started to take Shankapushpi. And he came to us with loss of seizure control. Of course, when he came to us, we didn't know that he was taking Shankapushpi. And we found that his phenytoin levels were low. So we thought he's non-compliant. But on lot of asking his question and this, he ultimately said that he's taking Shankapushpi. That's when we suspected that there could be an interaction between phenytoin and Shankapushpi. So we tested all the constituents of Shankapushpi into animal models of tonic-clonic seizures. And we found that they indeed did have anti-epileptic effect. However, they had an interaction with phenytoin. Both pharmacodynamic interaction reduced the anti-epileptic effect of the drug, and also these were enzyme inducers and reduced phenytoin levels. What was the lesson learned? You need to know, if you are an allopathic physician or a pharmacologist, you still need to know Ayurvedic formulation, and also of course careful history taking forever remains a good thing. We were working many years back on epidural morphine. There was a publication in Lancet, and we thought we will do testing and see how effective it is a pain relieving. And we had patient of peripheral vascular disease, and they used to get a lot of pain in the inferior extremities, so we gave epidural morphine. And we found that he had a toe ulcer, and actually the toe ulcer started showing actually bleeding. That's when we also did impedance plethysmography, a machine which was developed by BRC, and it was put in KEM for carrying out tests to check whether it can tell us anything about blood flow. And we found that indeed the blood flow in the lower extremity had improved after epidural morphine. So what is the lesson learned? That sometimes in order to have some idea that you may have, you need to be able to provide some proof for it. In this case, we used impedance plethysmography. Today, we have multiple types of equipment, and you should use that to actually show your proof of concept. This machine was later marketed by L&T. I'll next move on to giving you some anecdotes from product development and clinical trials. We just saw this picture in the student quiz that we had, Artemisia annua, and you know that this was developed by the Chinese UU2, and she showed 100% efficacy in extracts in animal models and in clinical trials. But Americans questioned. They said that this may be having CNS toxicity, and therefore they didn't want to accept it. Ultimately, it was left for the Swiss giant Novartis to take it up for further development. And around the same time, we had malaria resurgence in Mumbai. Now, many years there was very little malaria, and as politicians would say, they blamed it to migrants. So they said this is imported malaria got from outside into Mumbai. But actually we realized that it was on account of increase in the mosquito breeding sites. This is what my beautiful Mumbai looked like many years back, and with the high-rise buildings, checking of the overhead water storage tank for mosquito breeding was difficult. And we also realized that there was probably chloroquine resistance. So I'm happy to say then, well, not so happy because we were having so much malaria, but nevertheless, we were part of the multi-country, multi-centric trial on co-artemether and lumefantrine. Artemether and lumefantrine, and we showed that this was better than chloroquine because already there was so much of chloroquine resistance, and this was then accepted as the first line treatment by WHO and world over, as you all very well know. Now this is another offshoot of the clinical trial with malaria and product development of malaria. The commissioner of Mumbai got malaria. And at that time, not many physicians were familiar with malaria treatment, but we were well known for good knowledge and testing facilities for malaria. So we were asked to provide him treatment. He had Vivac, and he took 14-day primaquine, but he still got a relapse. And then we advised 28-day primaquine. Luckily after that, he didn't get any relapse. So that's how he realized the importance of clinical pharmacology, stroke pharmacology in patient care and in public health. And I think that was one of the reasons why our department got recognition a few months after that. So we can little bit boast for having contributed to UU2's Nobel Prize. Some of the interesting thing that we did was with a WHO funding, we carried out a clinical trial in a village setting, Kurujadi and Selukate, where patients with filaria, those of you know, microfilaria diagnosis was with 12 midnight blood collection. So we camped in the villages, and that was quite an experience. We carried out ultrasonography machine from Mumbai there and carried out ultrasonography test for filaria worm, and also of course an antigen test which had just come up. What was interesting is that we learned about the patients or the villagers' fear about groin pain, and which was because of the filarial worm being affected. Many of you have probably heard our story of liposome development, which we did in collaboration with Professor B.K. Bachawat and Gregoriadis and late Dr. S.K. Pandya, who is a neurosurgeon. And we had developed the liposome, which was already developed by B.K. Bachawat, carried out further, making it, as we say, patient, possibly to give it to the patient and patient worthy, and carried out manufacturing, and many of the PhD students who did their PhD with, and they had pharmacy, very good background. So this kind of manufacturing was done, and you can see the bottle. That was how our bottle, when we made it ourselves in Mumbai, in KEM, it looked like that. And carried out from phase 1 onwards clinical trials, and gave the drug also to the patient even in their homes, so much confident we were of its safety. We carried out studies in Patna also, and finally we had the technology transfer, and then Dr. J.N. Verma, a product of Chandigarh, he has actually taken to manufacturing. It took long time. If you think that development takes long time, from completing clinical trials and getting the patent and everything, the further manufacturing and marketing takes even longer time, which we realized as we went along. That's what it looked like. And the good thing was that besides fungal infection, it could be used in leishmaniasis, which, as you know, is the diseases of the poor patient due to sandfly. Post-Kala-Azar dermal leishmaniasis, which is part of the manifestation of visceral leishmaniasis. This liposomal amphotericin, which we had prepared, was also effective in such patients. It is also effective in mucormycosis, the black fungus in COVID-19. And we were therefore quite happy to say that because of this product, there was access to, at a lower cost, I wouldn't say at very low cost, but at a lower cost not only in our country, but the other countries where it is exported. So what are the lessons learned in this? One should prioritize public health problems so that what work you do is available for a larger number of people. Should try and benefit the patient by providing safe and affordable medicine. Now people are talking of devices. And to do this, there is a need to collaborate. As you could see, right from Department of Biochemistry to Neurosurgery to Public Health, we had collaborated with so many departments. Seize the opportunity. As soon as the commissioner wanted treatment, we immediately went forth with it. Working with different organizations. We can say broadly that there are two expertises which as pharmacologists or clinical pharmacologists we can think about. One is product development or development of medicines and clinical trials and so on. The other is use of medicines. So as a national chair with ICMR, I proposed to then DG ICMR, Dr. Balram Bhargav, who many of you would remember his father, Professor B.K. Bhargav, a very well-known senior pharmacologist. So happily because of that, or because it was a good project, he quickly accepted this proposal of Center for Clinical Pharmacology and gave a good budget and had two channels or two wings to it, product development and rational use. So the objective of the product development center was to evaluate the basic science projects which had a potential to develop into clinical products. So these were some of the products which were proposed from basic science people and from departments, various departments all over the country, including the Delhi University. Unfortunately, these products we could not take into clinical trials at all. There were some others which were developing new drug delivery system or pediatric formulation, and that was successful. Actrec developed a pediatric formulation for one of the chemotherapeutic agents, and the Chandigarh University along with the PJMER is developing another product. So the lesson that we learned from this was that basic science researchers need training to understand the drug development process and to carry out the experiments accordingly. It's one thing to carry out experiments to unravel, discover things and mechanisms of action and so on, but you need to understand as to what is the drug development process, what is needed in the drug development process, and carry out focused focus experiments accordingly. While the pharmacology and clinical pharmacology should, you should be interacting so that we are able to tell you as to where is the gap in the current treatment guidelines. And for repurposed drugs, it's important that the dose of the drug is clearly understood when you're doing the repurposing. Anyway, I'm happy to say that out of these product development centers, diagnostic tools have emerged, which one of it was the point-of-care G6PD testing, and the other one was viral diagnostic test. And for both these products, there has been a technology transfer, and they will be marketed soon. Other part of the ICMR project was rational use of medicine, for which there were two parts, prescription research and the other one was prescribing skills training program. So for prescription research, we had 15 centers, and this is the way prescription capture was done and was evaluated for appropriateness. But because of the drawback of this manual work, PrescriSoft, a prescription research software was developed, and this was jointly developed by the project, the Rational Use Medicine Centers and the ICMR's Medical Statistics Institution. It had some very good features such as having the WHO drug dictionary, the ICD disease code, and the national list of essential medicines. And also this could do, besides prescription, analysis by disease and drug, but this is under development. Anyway, with the validation being done and 1,000 prescriptions being entered, this was launched in March by the DG. Regarding the prescribing skills course for medical graduates, these were the institutions that were involved in developing this. I'm not sure whether we have time, but this next video is just to emphasize the importance of communication skill. Here is a resident doctor who is talking to the patient, and at the end of it you will say this is exactly what a doctor should not be talking to the patient. I don't know whether it will run.
नमस्ते डॉक्टर।
नमस्ते मनोज, कैसे हो तुम?
डॉक्टर अभी मुझे ठीक नहीं लग रहा है, चक्कर सा आ रहा है।
कितने दिनों से?
पिछले 1 हफ्ते से डॉक्टर।
तो बीपी वगैरह नापा है कहीं पे?
नहीं डॉक्टर।
ठीक है, मैं तुम्हारा बीपी देख लेती हूँ। तुम्हारा हाथ देना।
Is it audible?
तो मनोज, तुम्हारा बीपी अभी भी काफ़ी ज़्यादा है, मुझे दवाई बढ़ानी पड़ेगी एक, ठीक है? ये दवा मैं आज से बढ़ा रही हूँ, चालू करना, 4 दिन बाद मुझे आके दिखाना, ठीक है?
डॉक्टर ये दवाएं कैसे लेनी है मुझे? सुबह लेनी है, शाम लेनी है?
सुबह शाम दोनों टाइम लेनी है, बाकी का आप उधर मेडिकल में पूछ लेना।
और डॉक्टर यहाँ पे आपने SRD करके लिखा हुआ है, यही माने क्या है?
भाई SRD मतलब सॉल्ट रेस्ट्रिक्टेड डाइट। मतलब तुमको नमक कम खाना है।
तो कितना कम खाना है डॉक्टर? क्या खा सकते हैं और क्या नहीं खा सकते?
वो सब तुम उधर डाइटिशियन को पूछ लेना ना, बाहर मिलेंगे तुमको।
कहाँ पे बाहर मिलेंगे डॉक्टर?
देखो ये सब तुम बाहर सिस्टर को पूछना, वो तुमको सब बताएंगे, डाइटिशियन कहाँ मिलेंगे, फार्मासिस्ट कहाँ मिलेंगे। अभी मुझे ये सारी भीड़ जमा हुई है, वो सब खत्म करनी है नेक्स्ट 1 घंटे में, तो प्लीज आप उधर पूछ लेना, ठीक है?
So many of you would have been on one side of the table or the other side, but this was just to say that when you are wanting to use medicines correctly, you need to communicate it to the patient well. In interest of time, I'm going to skip
आपको
this
एक बात याद रखनी है कि आप ये ORS वगैरह उसको दे रहे हो।
That video showed how to actually communicate. For this course, we had, as you can see, ran it through 3 cycles, and we had more than 15,000 registration. We had a good feedback, and as you can see, there was a pre-test, post-test. The intern didn't seem to improve so much, but the registered medical practitioners gave a excellent, I think they attended much better, and there the post-test results improved so much. And interestingly, that the states which have low health indices, actually, from where we want more registration, actually had very low registration. So that gives us a strategy what we should be doing in future when we want to run this program further. Let me move on to one of the WHO committee that we had. This is miltefosine, a drug which is oral drug used for Kala-Azar, but when its use was extended to 12 weeks, patients started getting ocular side effects. And therefore, WHO set up this multidisciplinary technical group, and based on the data available, it was realized that there is a causal relation possible, and we gave a whole lot of risk minimization measures so that early detection and prevention of eye complications. Some of the regulatory activities in which I have been involved, of course, we developed this handbook, and the fixed dose combination review, which was based on the Supreme Court order, and wanted to evaluate. The important thing was that we had to give a hearing to the manufacturers to see as to why they wanted to continue the manufacture and sale of this fixed dose combination. And then, of course, our committee's report resulted in banning of several drugs.
Madam, time to conclude.
Yeah. So finally, while I've given all the other aspects of lessons to be learned, one most important lesson to be learned is to develop a team, have good mentors, as you can see here, and of course mentors like Professor P.L. Sharma. These are some of my mentors, and most important is all these of team from GS Medical KEM, and you can see Shubdeep who is here already, and you can see Pankaj also very sincerely doing injection into HPLC. Paying attention to detail that others have informed is one important lesson, sir, you will agree with me. But I always like to end up, despite the time that I have completed, just this particular 5 capabilities of a learning process with late APJ Abdul Kalam has said that you should have disciplinary mind, that is have mastery over one discipline. And he said you should be able to achieve that in 10 years time, maybe. Synthesizing mind, integrate ideas from different disciplines, be able to interact. Creative mind, this is particularly important in the era of AI, should be able to uncover and be able to find solution a step ahead of computer. So don't be only dependent on computer. But these are not really sufficiently good enough if you don't have a respectful mind, that is a mature mind, that will appreciate differences between humans, and an ethical mind that is able to work with integrity beyond self-interest. With that, I thank you all for your attention. Thank you very much.
I think the clap should go further. This is once in a lifetime opportunity for me, at least, sitting on the other side of the chair. While I saw ma'am in front of Professor P.L. Sharma, we feel the same, once a student, always a student. And she mentioned 1974 she did MD Pharmacology. I was born that year. Anyway, so the lessons learned are never forgotten, and lessons will always continue to happen. Yeah.
Well, thank you. I enjoyed your talk. It's good to know that a lot is being planned by the government. But the problem is there's no connect with the industry. We treat the industry as a pariah. Why don't we trust them, you see? I know, even in the CSIR, early days when I wrote to them, this is the problem, can you help me with money so that I can have few beds? You know the answer which I received? Please approach the industry. So that barrier has to break. And we learn a lot, you see, from patients. And many a time, when you get the queries, you have to do out of the box thinking, nothing to do with pharmacology. I give you one example. One fine morning, in my office, I get a call from the professor of cardiology that there are 2 patients sitting in front of me, well controlled on alpha-methyl-dopa. They come for a monthly checkup, and I find their anti-inflammatory completely gone. Now what do you advise? So it was in my knowledge at that time that there's a problem with the import of the base material, and this is not available in the market. So at Professor Bidwai asked these patients what tablets are they taking. And fortunately they had some tablets. I have a look at the tablets. Said, is it L-Dopa by any means? So he looked at it, my God, it's L-Dopa. You have the answer to your query. M-Dopa is not available. He said, koi paas de do. Chemist gave L-Dopa. Then another problem, VIP, former Chief Minister of Haryana, Bhajanlal was severely ill in Delhi, admitted to AIMS for 17 days, no diagnosis. He had himself shifted to PGI, and that afternoon I had a phone call from the head of the unit, Professor Mehta, under whose care he was. डॉक्टर शर्मा भजन लाल जी आ गए हैं, मैं क्या मालूम है हेडेक आ गई तेरी. He is there for 17 days. They would have done all the investigations possible. He said, any suggestion? I said, look, as far as my knowledge of differential diagnosis goes, if in spite of all your efforts you are not able to make a diagnosis, two possibilities still remain. One is a mix of psychiatric disease, other is drug-induced toxicity. These two still remain. And here I may tell you my limited experience with them, VIPs, they do a lot of doctor shopping. And many a time they take treatment from more than one system medicine. If you take the drug history, they won't tell you, lest you may feel offended. So my suggestion to you is, take a blood sample, send it for estimation of heavy metals. Idea appealed to him. Next day I get a call, Sharma, you hit the nail on the head. The fellow is loaded with lead.
Thank you, sir.
So
We can continue to have this discussion with Dr. Nilima Kshirsagar at tea time. Thank you very much, madam, for your excellent sharing your observations of 50 years with all of us. On behalf of Indian Pharmacological Society, I thank you very much.
Thank you very much, sir. Good evening.
So we'll now start the process of valedictory session. Just within 2 minutes we'll start.
A very warm good evening to one and all. As we arrive at the concluding session of IPSCON 2025, we stand at the crossroads of conclusion and new beginning. Over the past 2 days, we have learned, exchanged ideas, and built everlasting professional bonds. This moment marks not the end of the conference, but the continuation of a shared journey in pharmacological innovation for a Viksit Bharat. On behalf of University Institute of Pharmaceutical Sciences, Punjab University, Chandigarh, I welcome you all to the valedictory function of 55th Annual Conference of Indian Pharmacological Society. And it is my proud privilege to welcome and invite dignitaries on the dais. Chief guest of the occasion, Professor P.L. Sharma. Guest of honor, Dr. Jyoti Yadav, Chief Scientist and Head of the Recruitment Board for the CSIR. President IPS, Dr. Babul K. Bezabaro. General Secretary IPS, Dr. B. Kalakumar. IPS advisors, Dr. P.B. Diwan and Dr. B.K. Roy. Organizing Chair, Professor Kawaljeet Chopra and Professor Anil Kumar, Organizing Secretary, IPSCON 2025. Okay. I invite IPS advisor, Professor Shiv Prakash, to please come on the dais. I also welcome all the dignitaries of the dais, renowned pharmacologists, esteemed speakers, delegates, guests, and dear students. Thank you all for gracing this occasion. Now, I invite Organizing Chair, Professor Kawaljeet Chopra, to present a welcome address.
Thank you, Dr. Sangeeta. A very good evening, everyone. And as the organizing chair of the 55th Annual Conference of the Indian Pharmacological Society, IPC IPSCON 2025, it's my profound pleasure and an honor to welcome you all to this valedictory function. So first of all, I tender my gratitude to our chief guest of the valedictory function, respected Professor P.L. Sharma ji, our guest of honor, Dr. Jyoti Yadav, and all other esteemed guests and leaders of Indian Pharmacological Society. So the last 5 days of intense scientific discourse held under the theme, Empowering Young Minds to Transform Pharmacology for Viksit Bharat, have been nothing short of spectacular. We kick-started with pre-conference workshop on emerging tools and technologies, alternatives to animal testing, with a focus on hands-on training on various advanced techniques, followed by a meticulously designed scientific platter comprising of esteemed orations, 25 specialized symposia, invited lectures, numerous invited lectures, panel discussions by 143 national and 23 international speakers from academia, industry, and drug regulation department. So I'm very confident that our young researchers, who were the real focus of this conference, they have fostered new friendships, new collaborations, and have noted down definite take-home messages so that we can collectively, as a pharmacology fraternity, realize the vision of Viksit Bharat. Thank you all for making this conference an unparalleled success, and I wish all of you a safe journey back home. Thank you so much.
Thank you, ma'am, for your welcoming words. Now, I invite and request President IPS, Dr. Babul K. Bezabaro, to say a few words.
Respected today's chief guest, P.L. Sharma sir, Jyoti ma'am, my colleagues and chairperson and organizing secretary of 55th IPSCON 2025. What to say? In each and every aspect, I have taken feedback from each and every, most of the members, and nobody said a single sentence against in any aspect. Please give a big hand to this team of UIPS. And who have organized this conference so nicely, and hospitality, food, transportation, everything was so perfect that, and we should thank the young guys who has helped us a lot during transportation, fooding, etc. So for the first time, I have seen this collaboration amongst all the members, and it's a good team. Without a team, organizing a conference will not be successful. Organizing a national conference is not a joke. And ultimately, Dr. Anil really has done a great job. So, I, on behalf of IPS family and all the members of IPS, again thank you, pay thanks to this whole the team, and hopefully everything was a grand success. We are coming to the end of this conference. I have seen the quiz session also. Dr. Hajra and Nitin, very nicely they have conducted good deliberation amongst the speakers, as well as if you go to the theme, it was meant for you people, that is younger people. So Biksit Bharat means our aim is 2047. Will not be there in 2047, but you people will have to come forward to just help the country in progressing the aspect of medicine and health aspect of India. So nothing more to say. Again, hats off to the team, UIPS. Jai Hind.
May I now request Dr. B. Kalakumar, General Secretary IPS, to address the gathering.
A warm, a warm good evening to all of you. The auditorium seems to be full as on the inaugural day. Hope you are quite anxious to know the winners of the various presentations, oral and poster presentations. Maybe that's the reason the hall is full. But as far as my experience goes, in all other valedictory functions, there would be very few people, about 10 to 15. But here the hall is full. Maybe Anil did some magic to see that the chairs, that the auditorium is full, right? One unique thing about this IPS conference is that Anil had gone for live presentation of all the sessions. Generally, it would be you pay, then you participate. Here it was pay or no pay, gather the knowledge of pharmacology. I think this is wonderful, and this is the next organizing secretary, that is West Bengal chapter, would also follow this, and it would be there on YouTube. Even because if you are in hall 1, you miss hall 3, 4 and the main hall. But now, even if you have missed live, you can have the recorded version. You can go through at ease. And because of my illness, I could not attend Shubdeep's uh, session on biosimilars and regulatory requirements. But I have gone through the recorded version. That's good, Anil. Uh, on behalf of IPS, I thank you for uh, uh, this gem that you have presented. Coming to the published papers, for the past 2 years, there was some miscommunication and gap, but this time the organizing secretary, he has taken care to announce prizes for the published papers too. At one point of time, we were in jitters, and we also had plan B. If Chandigarh fails to come out, we thought of going to Belgaum, and plans were afoot to shift the venue from Chandigarh to Belgaum, but still the organizing secretary has done his best and saw that this is done in Chandigarh itself. That's the reason, because of his efforts, we are all here, and I thank Anil for that. With these few words, I thank every one of you.
Thank you so much, sir. We are honored to have amongst us Dr. Jyoti Yadav, Chief Scientist and Head CSIR Recruitment Assessment Board. She handles the recruitment and assessment for CSIR, ensuring transparency and efficiency. Welcome, ma'am. Now, I invite Dr. Jyoti Yadav to share her words of wisdom.
Hi. Good evening. Everybody awake? Post lunch and valedictory. So most of the time, most of the people will be looking at the watch, right? Anyway, respected President of the Indian Pharmacological Society, distinguished office bearers, eminent scientists, honored guests, and my dear friends, young pharmacologists. You are the aspiration of the country. You are not just a student of pharmacology, you are the budding future of the country, a budding future for Viksit Bharat. It's really my privilege to be here, and thanks to Dr. Harlokesh. I was a little reluctant because, as somebody also said, there was a plan or maybe plan B of shifting the conference from here. But he assured me and he said, no, there's a very vibrant young people, young students who are really looking forward for this conference, and there's nothing as such which is being portrayed in the media these days. I'm sure this conference has not just brought together many, many delegates from many different countries, many different speakers, but all the pharmacologists together in this, and you must have had a very, very beautiful time, not just learning science, various aspects of not just pharmacology, but different aspects of science and medicine. I feel pharmacology is a very uniquely placed science, a melting pot, or rather a cusp of medicine and basic sciences. And I find the pharmacologists in my career, as my students, my PhD students, the people who come from the pharma background, have a very deep analytical mindset, and which I think is a very, very unique skill set. And thanks to all the leaders and your teachers who have probably instilled, or it's a science, because I'm a student of chemistry, very similar, but somehow I feel that I always lack that medical input into my chemical knowledge. Today, pharmacology, as a pharmacology student, you also need a little bit more than that. You also need to learn a little bit about AI, the upcoming technologies, the biotechnology, the AI, and the basic and the clinical pharmacology. From my eyes, to my eyes, you people might be having a understanding of the differences. For me, clinical pharmacology and basic pharmacology, or basic sciences, which you probably call, they're absolutely intertwined. And a medical research without a pharmacology base is quite hollow. As ma'am just mentioned a few cases where just because of her pharmacological background, she could analyze and she could treat those patients in a very different way. as a policy maker, rather, which I have become of late, I also feel that pharmacology is also a very strong pillar for policy making. A policy without a good evidence cannot be a good policy ever. And with the pharmacological evidences, we can, if the evidences are gathered very carefully, as somebody mentioned, that's a basis of a very good policy. There are several areas where pharmacology students may be required, are required rather. And as an academic institution, I belong to CSIR and I represent CSIR. CSIR is also looking forward with both hands, very young pharmacologists, because there's a lot of basic science happening in 37 different CSIR laboratories. However, I don't know what the reason is, but the pharma students have not shown that greater affection towards CSIR. We look forward, and I feel that CSIR is a good place, is a good institution where you can hone your skills. I feel, and I strongly feel rather, that the knowledge which you gather would bring you, will get you a job. But your skill set would make you stay there, would make you flourish in that area. So you have to hone your skills, not just what you have studied, that's absolutely fine, but there are many, many more areas where if you can, some of them would be science communication, policy writing, if you know how well basic science can be implemented, that all would give you a much larger, breadth of looking for future recruitment plans. And with that, I would keep short, because you must have had a very long and prolific sessions of science and different areas of new IPs and many new areas you would have explored. What I would want you to take back with you, not just science, but take all your pharmacological, fraternity together. And I see a lot of pharma fraternity here supporting each other, which is actually a very, very positive. So I'm very sure that with this kind of an effort, you would definitely flourish. The whole society is going to flourish, and the students would. So Jai Hind, thank you very much.
Thank you so much, ma'am, for your inspiring words. Now we are moving to the next segment and the most exciting segment of the valedictory function. And for this, I request Dr. Vandita to please take over the mic.
Thank you so much, Dr. Sangeeta. So now we have arrived to the very important segment, and for the most important moment we all have been waiting for, the award ceremony. To start with, the first category of award we have is the IPS prizes for oral presentation, and we have 5 awards in this category. The first one is G. Achari Prize, and it goes to Mr. Satnam Singh, AIMS, New Delhi. The UK Seat Prize is the second category of this award, which goes to Mr. Gauri Chaturvedi, again from AIIMS, New Delhi. Third is the Gufic Prize Work for Endogenous Medicines in India, which goes to Chandan Chauhan from Naipur, Rai Bareli. Next, we have the O.D. Gulhati Prize, which goes to Janvi Soni from Parul University. The next one is Professor Manjeet Singh Prize, which goes to Mr. Pranav Panchbal, AIIMS, New Delhi. Congratulations to all the awardees. The next segment of award is Best Paper Published. We have 4 categories under this. The first is B. Unvas Prize for Best Paper Published, which goes to Dr. Ashok K. Dastusala from Naipur, Rai Bareli, UP. Next is PP Suryakumari Prize, which goes to Dr. Mahinder Bishnoi, Nabi, Mohali, Punjab. Then we have Saroj V.N. Sharma, Best Paper Published, which goes to Dr. Kuldeep Kumar, Punjabi University, Patiala. Next, we have Professor Hardayal Singh Prize, which goes to Dr. Siddharth Mehan, ISF Moga, Punjab. Congratulations to all the awardees of the IPS prizes. Now we have award for the poster presentation. Since we had received a large number of entries, we had to keep the session in the morning and the evening, and total number of prizes which we have is 15. So we have Sakshi Mohet, Ankan and Keshari Kumar to please come on the dais. Sakshi, Ankan and Keshari. Then we have Akshita, Sonam Yadav and Ojas Tiku. Please, may I have all of them on the dais, please? Isha, Harshita, Nidhi, Ambika, Nikita, Ashish Jangi, Srishti, Rohan, and Ramadeep. Congratulations to all the awardees. Now we have another award, the PC Dandiya Best Poster Award. And this award goes to Harshita Singh, Central University, Punjab. Now it is the time to felicitate all the awardees of Young Investigator Award. I request all of them to come please on the dais.
This year, the organizing committee has constituted Young Investigator Awards. So there are a lot of youngs have been promoted, encouraged with the Young Investigator Award. And award carries the medal, certificate and travel subsidy. So all Young Investigator Award are requested to please come fast and take group photographs. Fast.
you will get it later on. You will call before this. Then come.
Congratulations to all of them. A huge round of applause for our young investigators. Now it is a time to announce the PETA India Award, which was initiated this year to recognize the contribution of the researcher working on non-animal model for regulatory and safety testing. I would like to call upon stage Dr. Rohit Bish, scientist, regulatory testing, pharma and medical devices. On the dais, please. The award has been initiated by PETA India, and this year the award goes to Ms. Khushbu Patania, who is at present working as a Commonwealth Fellow, and she's working under the supervision of Dr. Sandeep Pawar and Dr. Sangeeta. May I request the guides to please take her award on her behalf. Dr. Sandeep Pawar is the backbone of this conference, and Dr. Sangeeta Pilkwal Shah, again the joint organizing secretary of this conference, have worked tirelessly for the success of this event. A huge round of applause for both of them.
This award got the all nomination to the PETA. PETA has independently taken the decision and they notify us. Thank you.
Now we are going to announce the award for the quiz. So the first team who has won is team number E, that is Kunal, Chanchal and Shivam.
May I request the Dr. Avijit Hazara to please come on the desk. Avijit Hazara, Dr. Avijit Hazara.
And Dr. Mohit as well. Can we have Dr. Mohit here on the dais, please?
Now this e-quiz is a new feature in IPS, and that carries the award of 21,000 total, divided into 3 parts.
So this quiz award, which is presented on the forum of IPS conference as the National Pharmacology Quiz, this is a contribution from the West Bengal chapter of the Indian Pharmacological Society.
Second prize goes to team A. Vishnu Keshari and Sanjeevam Gupta. Third prize goes to team C. Sampriti, Komal and Meera Kumari. Congratulations to all the teams. Now we have an award for the sponsorship of for the event of IPSCON. So we have the award for Himalaya, and we would wish to call upon stage Dr. Eliza from Himalaya to receive the award, please.
We are just providing the certificate of sponsorship so that they will be encouraged and they will continue to support us. Himalayan representative, please come.
Thank you so much.
This year we are also motivating, encouraging the past general secretary, organizing secretary, and I request Hari Lokesh to please come on the dais, as he has also successfully organized the IPSCON 2024. And this is a trend we are setting, that we are honoring the past organizing secretary in coming event to honor their contribution.
Thank you so much all. May I request all the dignitaries to please take their seats on the dais. So we're deeply honored to have with us Professor P.L. Sharma sir, and I request sir to deliver the valedictory address.
Well, ladies and gentlemen, there's a saying that all good things come to an end. So your annual conference has come to an end. I'm sure you have had a good feast, academic as well as the gustatory. And you must be going back home with your batteries fully charged to devote to research. Now, I spent 57 years of my life in pharmacology. If you ask me one thing which concerns me today, is that the declining interest of the young generation in research. This is very evident in medical colleges, less so in pharmacy colleges, they are much better. If you ask a undergraduate what you are doing, or MD people, oh sir, with these animal restriction, we can't do anything. I don't think there can't be a more lame excuse than that. There is one story which I used to tell all my students, that to do research It needs only a frame of mind and your determination to do so. If you have the motivation and the perseverance, nothing in this world can stop you. And my pet story is this. A South American pharmacologist engaged in psychodramatic drugs research. He got imprisoned on a political charge. He was awarded A class in the jail. All facilities but no work because there is no RI for the political prisoners. There's a lot of time, very difficult to kill. So in the evening, sitting in a corner of his room sipping coffee, he just noted in the wall some cobwebs, spider cobwebs. So in frustration, he damaged all of them, removed them. 2-3 days, he again found those cobwebs. Same design. So it occurred to him that the design of the web is specific for the spider. There are two spiders, different designs. Now here it came an idea into his head, why can't I do research sitting in jail? So he made a proposal and sent it through the jail authorities to the government that he would like to get a balance, a weight box, some glassware, limited chemicals, a photographic camera with black and white film, because that's all what was available. So what he did was, he said, I don't mind if the jail authorities keep all this in their custody, give it to me at time of experiment, and if one of your officers stand at my back and watches what I'm doing. So what he'll do is, sprinkle some chalk powder on the web, give a good contrast and take a black and white photo. Did it 3 times. That is the control. Then he exposed those spiders with a sprayer to a very dilute solution of psychodramatic drug and destroyed the web. 2-3 days later, the webs came back, but the design has gone haywire. Again did 3 recordings, stopped the drug treatment, and then 3 weeks later, the normal designs appeared. The gentlemen tell me what facilities are needed, all simple things. Sitting in the jail, he was able to publish this work in a peer-reviewed top biomedical journal. Simply is determination to work. In India, my advice to the youngsters is get started with whatever you have. Because if you wait for the ideal things to come up before you start, you will never get started. This is Mother India. Everything moves at a snail's pace. And we Indians are very good at jugaad, what we call improvise. Use it. I give you one example. One of my specialties is cardiac arrhythmias. And when I was doing my MD, I read all this. Maran Prinzmetal in USA utilized high-speed cinematography to show that the impulse is arising in the ectopic focus where he has applied aconitine. Even in London, I didn't have that sort of facility, the high-speed cinematography. Film at high speed, view it at slow speeds and see what's happening. So I used a jugaad. Took a curved artery forceps, put a rubber tube on it so that it won't bleed the atria, and applied a clamp on a little away from the tip of the right atrium so that the aconitine treated area is separated from the rest of the atrium. And after crushing the tissue, I removed the artery forceps. On a 4-channel recorder, simple. I could show the aconitine treated area fibrillating and the rest of the atrium and the left atrium, sinus rhythm 150. Simple jugaad, no high-speed cinematography. So, and this paper is published in British Journal of Pharmacology. They will say, what have you done? What is that and what's that? You can do that. Similarly, when I started clinical pharmacology here, I didn't ask for anything because I knew I'm not going to get it. But it's an institute heavily dedicated to patient care. Everything is there for the hospital. In the research department, you have to make do with your research grants. And I needed a bicycle ergometer or treadmill, I didn't have that. So I went back to the master's two-step. That also was not available. So I went to the stores, get a sturdy wooden crate issued in my name, went to the workshops, had with the carpenter, had that master's two-step made. When more money became available, bicycle ergometer came, then treadmill came. Had I waited for the treadmill to come, and those papers, master's two-step, are published in international peer-reviewed journals. They didn't say that why have you used this. So, if you want to start, just start whatever you have. Don't wait for ideal things to come because that's not going to happen. And my other little concern is that number of you, when you pass your MSc degree or MD, don't publish your work. You get a job and become pleasant. What is the use? Why spend time on it? You never know, a situation may come when you have to face a very awkward situation. Again, I'll narrate to you, somebody has not published his PhD work. And why, as a consultant with Ranbaxy, I was interviewing for the director's post. A beautiful CV. He was from Canada. Professor there, he wanted to come back and head the preclinical pharmacology division. And to look at his CV, I knew that it has been written by a professional body. A lot of CROs are available abroad. Will make you CA for fee. And so much praise for his MSc work and his PhD work, which he did in India before he went to Canada. And when I started the interview, he all started with, oh, I did this, I did this. I kept on hearing. I said, may I ask you one question? If this work was so good, your CV says you are proud of it, your list of publications starts from Canada. Why have you not published this work? I said, my interpretation of what you're saying is that your work was not worth publishing. How you got your degree, God knows. But you are not publishing your PhD work, then I don't know what sort of work you did or what quality work you did. So with that, I think I'll stop here because all of you must be very eager to go back home to your working stations. Wish you all the best, comfortable and safe journey back home. Thank you very much.
Thank you so much, sir, for such insightful and inspiring address. I really hope each of us have a take-home message today. Thank you so much, sir. Now is the time to felicitate our guest. May I request all the present on the dais to please honor our guest of honor. Thank you. A huge round of applause for ma'am. May I request all to felicitate our guest of honor, sir, chief guest of the day, Dr. P.L. Sharma, sir. Time to felicitate our President IPS, Dr. Babul. Our General Secretary IPS, Dr. B. Kalakumar. Advisors IPS, Dr. Shiv Prakash sir and Dr. Roy. The organizing chair of IPSCON 2025, Professor Kawaljeet Chopra. A huge round of applause for ma'am. The heart and soul of the conference, our chairperson and the main organizer of the conference, Professor Anil Kumar sir. Thank you so much, all of you. I would now like to invite Dr. Anil Kumar sir to deliver the vote of thanks.
Hello. So the journey of the whole year is now going to complete. This journey was started November 2024 with mixed of emotional experience and lot of people's support. Now the fruits you can see in the last 3 or 5 days, Now this is a time to acknowledge my whole team, particularly starting from the IPS leadership, particularly the president, secretary, executive members, past office bearer, advisors. They were the main guidance, which are the backbone of this conference. So thank you, sir, for constant support. Dr. Babulji, Shiv Kumarji, Kala Kumarji, P.K. Roy, and P.V. Diwanji. I'm also express my heartfelt appreciation to our all international speakers, national speakers, whose expertise, thought-provoking lectures, visionary perspectives, enrich scientific dialogues, broaden our understanding. And I'm also thanks to all the speakers who never missed even a single lecture, and they attended and deliver as per their commitment. Thank you all international and national speakers from my core of the heart. In addition to this, there are long list of poster evaluators who were not in this stage, but they are from the backside. They did their job, and I'm also thanked to all of the reviewers, After that, no event is successful without the support of the institution. So I would like to say thank you, UIPS family. So I would like to request my all UIPS faculty to please come on the dais so that they can know the contribution of our UIPS faculty. Request, please come. ठीक है. So this is the backbone of the institution, who are directly, indirectly supporting the entire event. Dr. Shuni is also a part of my faculty. So you can see the backbone of the institution, who provide the support, guidance, advisor, and long, you can say the motivation. The institution's success depends only the guidance of these eminent seniors, juniors, and this is not possible without involving the guest faculty. So I request my all guest faculty to please come on the dais, and I would like to say thank you from a core of my heart. All the guest faculty, please come. They were the main guiding force who are handling my hall 1, 2, 3, 4. So thank you, UIPS family. Now, in addition to this, I would like to extend another thank for उसके बिगड़ तो मेरी इवेंट पॉसिबल ही नहीं थी. डॉक्टर संदीप पवार, डॉक्टर संगीता, डॉक्टर नीलिमा, so they are my joint organizing secretary, and I would like to say thank you, and I would like to felicitate to them. I request Kojit ma'am, who is the organizing chair, to special felicitate to them. I request the IPS president and advisors to please help and motivate these young people. Dr. Sandeep was one of the guy who taken care of the accommodation, transport, scientific program, abstract book. He was involved in everywhere. Even I said, अगर मुझे कुछ होता है, तो he will be my second most line. Now, Dr. Sangeeta, who taken care of the pre-conference workshop, and I have given the independent charge to her to design the program, call the speaker, taken care of the accommodation, everything. And I request the Dr. Sangeeta team members, please come on the dais. Okay. So moving forward, I would like to thank Dr. Vandita, Dr. Prerna Swarup, who is taking care of the hall 1. Hall 2 is taken care by Dr. Manish, who is taking care of the entire oral presentation. Dr. Ashok Yadav, who has taken care of the poster presentation. Dr. Amita, who has taken care of the hall 3. Dr. Monica, hall 3. Dr. Binta and Dr. hall 4 में, that is Shilpa Bhasin. Okay. Now the more core of my team, my Khados team, कहाँ हैं? Please come. Yeah. Let me come back first. Wait. My core team का Khados team, Dr. Miss Garima, Ritu, Priya, Harvin, Neetu, Minni, Naman, Pankaj, Suraj, please come. These are the core people, 9 हीरे हैं मेरे ये. बेटे सामने आ जाओ. And I request President IPS to please motivate them, encourage them. Yesterday you have given the chocolate, I know that. You have given a special chocolate to them and you encourage and motivate them. जैसे मेरी कोर टीम है ये, जो रात को 2 बजे 4 बजे सब जगह काम किया है इन्होंने. अभी भी ये कोर कंट्रोल रूम में से ऑपरेट कर रहे हैं. थैंक यू कोर टीम, खड़ूस टीम. Now comes to my hospitality team. You know that everybody appreciated the hospitality. And Dr. Gurupal team is there. Dr. Gurupal, where is good Dr. Gurupal? Please come, bring your team. Dr. Gurupal. Dr. Gurupal, please bring your team. Ranjit, Dr. Ranjit कहाँ है? Dr. Ranjit का तो ऐसा है कि सर मैं बता सकता हूँ, अगर वो इतना स्ट्रिक्ट है कि रोटी मुंह से छीन सकता है अगर आपके पास कार्ड नहीं है तो. इतना स्ट्रिक्ट है. समटाइम्स मेरे अपने गेस्ट भी नाराज हो गए कि भाई थोड़ा रिलैक्स करो, ये तो बहुत ज़्यादा टाइट हो गए हैं हमारे साथ. उसके बाद डॉक्टर अशोक हु हैज टेकन केयर ऑफ द पोस्टर सेशन. प्लीज डॉक्टर अशोक, वेयर इज देयर डॉक्टर अशोक, प्लीज कम. डॉक्टर मनीष, प्लीज कम. Poster and oral presentation is taken care by Dr. Dr. Ashok and Dr. Manish. Please come. Your contribution need to be recognized. Please come. In addition to this, okay. Dr. Ashok. आ जाओ सामने आ जाओ. Dr. Manish, please come. They were the team who has given the independent charge and they were calculating everything, poster awards, oral presentation award, oral के submission जाने हैं, intimation होनी है, everything is taken care by independently. And I was not sure whether they will be able to do it or not, but they did so well. आप बता नहीं सकता हूँ मुझे, मेरी event management team ने मुझे धोखा दे दिया, but these were the people who supported me. Thank you, Dr. Manish and Ashok. Now the another one is the cultural team. You know that the day 1 evening show, झकास show यहाँ पर हुआ है जो. You know Dr. नीलिमा कहाँ है? Dr. नीलिमा and team, cultural team. हाँ. She was not well, even then she did arrangement. You know all तरह का इसने किया, भंगड़ा वाला जो performance, you can see it. ओके मेरी टीम खड़ूस टीम बोल रही है कि भाई एक अजय रह गया है और कहाँ गया दूसरा वाला कौन है? पायल. कहाँ है बच्चा, बुलाओ. आ जाओ बेटे, कम फास्ट. नाउ दिस इज टाइम वी नीड टू रिकॉग्नाइज यू बेटे. आपके एफर्ट्स को हम नेगलेक्ट कर ही नहीं सकते हैं. आ जाओ प्लीज. वेट 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 आ जाओ. इन एडिशन टू दिस, ये 5 दिन में जो वॉलेंटियर टीम थी मेरी, यू ऑल अवेयर हाउ taken care of, accommodation, transportation. So I request my all volunteer team, please come. All volunteer team, please come. All students, please come. Volunteer team, please come. All volunteer team, please come. And ये वो students हैं जो अपने सेशनल एग्जाम देने के बाद डे नाइट हमारे साथ हैं. बोल रहा बोला. आ रहा हूँ मैडम, आ रहा हूँ. प्लीज प्लीज कमिंग कमिंग कमिंग. वेट मैडम, वेट 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 वेट. नाउ स्पेशल थैंक्स टू सूरज, नमन, प्रिया, पंकज, नाजिया, हर्ष, श्रुति, अंचित, मानस, कार्तिक, ऋतिक, कृतिका, रोहित, वंश, मुस्कान, निकिता, शुभम, अभिषेक. सर present sir, you need to be, you can see the force behind, ये वो force है UIPS की. Very nice. Sir, we need your support to them so that they will work. No. आगे आ सकते हो बेटे, आगे आ जाओ. You should be on front line बेटे, अब तो already आप trained हो. Thank you once again. Thank you. Now you can see the, when you go out you will see the number of news items, you know, जितनी भी प्रेस लगी है हमारी बाहर, अभी मैंने सबकी प्रिंटिंग लगाई है. हाउ मेनी न्यूज़पेपर कवर्ड आवर न्यूज़ फ्रॉम लास्ट 3 days, एंड उसका श्रेय जाता है पूनम जी मैडम हैं. मैडम पूनम मैडम आओ प्लीज़, आओ प्लीज़ मैम, प्लीज़ मैम. शी इज़ कंटिन्यूसली मेकिंग द प्रेस कॉन्फ्रेंसेज एंड ऑल्सो कंटिन्यूसली ट्रैकिंग द न्यूज़पेपर रिपोर्टर्स. हमारी न्यूज़ लगानी है और 3 दिन तक लगानी है, अलग अलग तरह की लगानी है. सो यू कैन सी द न्यूज़ आइटम्स आउटसाइड मैंने लगाए हैं, तो यू कैन सी इट. एंड प्रेस मीडिया टीम प्रेरणा एंड दिव्या, प्लीज कम. पुनिया जी, अरे हाँ ये तो यहाँ बैठे हैं. प्लीज प्लीज सर, सर आ जाओ. ये डीपीआर के बगैर तो हमारा कुछ हो ही नहीं सकता सर. आ जाओ प्लीज सर, पुनिया सर प्लीज कम. पुनिया जी ने क्या किया, हमारे यहाँ इस स्ट्रेटेजी बनाई कि भाई कैसे हम हर डॉक्टर की न्यूज़ अलग अलग तरह से निकाल सकते हैं. एंड नाउ यू कैन सी इट. डॉक्टर धल्ला की न्यूज़ अलग आ रही है, डॉक्टर देवेंद्र जी की न्यूज़ अलग आ रही है, एंड आई हैव पुट इट ऑल दी कॉपीज आउटसाइड. यू कैन सी इट. ऑल न्यूज़पेपर कवर्ड दी UIPS IPSCON 2025 लास्ट 5 दिन की सारी न्यूज़ आइटम बाहर लगा रखी है मैंने. एंड आई विल शेयर विद यू पर्सनली थ्रू योर मेल. So how our media is covering your impact of your research to the society. That is what is there. Thank you प्रिया जी. Thank you. इसके अलावा ये इंस्टीट्यूशन में हम कोई कॉन्फ्रेंस नहीं कर सकते विदाउट द सपोर्ट ऑफ द यूनिवर्सिटी. सो आई वुड लाइक टू थैंक वाइस चांसलर मैडम, रजिस्ट्रार सर, DOI, Control Examination, FDO, they are all RUSA. RUSA is also one of the great support financially for the success of this conference. In addition to this, there are a lot of sponsors are there, industry partners are there who supported financially for the success of the conference, particularly the AcroLab, ADVT, Abbott, Abogenesis, Clean Pharma, High Tech Formulation, Pharmacoevidence, FarmBios, Experimental Pharmacology Softwares. There's long list is there, but I would like to thank every sponsor, advertisers who supported for this conference. In addition to this, teaching, non-teaching support, but there is another मेरी backbone है, वो है non-teaching support. I request my all non-teaching support office, please come on the dais. My all non-teaching staff, ध्रुव जी कहाँ गए? आ जाओ प्लीज, प्लीज कम. हमारा रजिस्ट्रेशन सिन्हा सर ने संभाला, इंदू मैडम ने हमारी एग्जीबिशन संभाली, और कहाँ गए बाकी? आ जाओ मैम, आ जाओ प्लीज प्लीज. ये टाइम है मैडम, प्लीज कम. सिन्हा सर आ जाओ. अलका मैम प्लीज आ जाओ, 1 डे रजिस्ट्रेशन आपने किया, आ जाओ प्लीज कम. अरे मैम प्लीज आओ ना, ये टाइम है मैडम. इट इज बीइंग रिकॉर्डेड ऑल्सो. आई वुड लाइक टू थैंक यू ऑल्सो मैम. सिन्हा सर प्लीज कम. सर आ जाओ प्लीज. अरे आ जाओ सर. हमारे रजिस्ट्रेशन के इंचार्ज जयमली जी कहाँ गए? इतने सारे हैं, मेरे तो समझ नहीं आ रहा. डॉक्टर जय आ जाओ प्लीज. रजिस्ट्रेशन, अलका मैम 1 डे रजिस्ट्रेशन. हमने एक नया स्कोप शुरू किया सर इस बार कि 1 डे रजिस्ट्रेशन भी करो. जो पे ज़्यादा नहीं कर सकता एटलीस्ट 1 डे में आओ, पेपर प्रेजेंटेशन दो, एब्स्ट्रैक्ट प्रेजेंट करो. आप रजिस्ट्रेशन किट नहीं देंगे बट यू कैन पार्टिसिपेट इन द कॉन्फ्रेंस, so that everybody can participate in the conference. And this was the next new feature added into IPS conference मैंने. So these are my non-teaching staff, who office staff basically, who support me in TA DA clearing करना है, ऑफिस में क्लास को कैसे ऑक्यूपाई रखना है। हॉल जो आपने देखा कि हर कॉन्फ्रेंस में हॉल पैक नहीं होता है। सो दीज वर द फोर्स बिहाइंड द स्टूडेंट को कैसे एंटर करना है, बाहर से लोगों को कैसे अंदर करना है। सो ऑल हॉल वर पैक्ड बिकॉज ऑफ दैम। इन्होंने कई बार खाना भी नहीं खाया, हमने बोला कि बाद में खाना, तो नाराज भी हुए, तो मैंने उनको सॉरी भी बोला बाद में, बट सॉरी अगर मेरे से कोई गलती हुई हो। थैंक यू। इसके अलावा मेरे से गलती से कोई रह गया हो, भूल गया हूँ, तो सॉरी फ्रॉम माई कोर ऑफ माई हार्ट, एंड दिस इज ऑल फ्रॉम माई साइड। थैंक यू वंस अगेन फॉर योर पेशेंट लिसनिंग। थैंक यू। Before this, with immense pride and gratitude, we formally declare the International Pharmacology Conference 2025 IPSCON closed. Next, IPSCON 2026 would be held in Calcutta. You are most invited. So now, before you leave, please please say, we need to close this function by a national anthem. इसके अलावा हमारी एक मैंने फिर बोला कि मेरे से बहुत सारी लॉन्ग लिस्ट है। एक गगनदीप है जो हमारे ऑडियो वीडियो सारा है वो देख रहे हैं। प्लीज गगनदीप आ जाओ बिफोर वी स्टार्ट नेशनल एंथम। He was the person who made all the posters, all the display, all audio video. They are the core team who has helped, Thank you, Gagandeep. इसके अलावा अविनाश जिसका खाना सभी लोगों ने प्रशंसा किया, वो अलग है, पीछे अभी भी काम कर रहा है आपकी चाय के लिए। थैंक यू।
जन गण मन अधिनायक जय हे भारत भाग्य विधाता। पंजाब सिंध गुजरात मराठा द्राविड़ उत्कल बंगा। विंध्य हिमाचल यमुना गंगा उच्चल जलधि तरंगा। तव शुभ नामे जागे तव शुभ आशीष मांगे। गाहे तव जय गाथा जन गण मंगलदायक जय हे भारत भाग्य विधाता। जय हे जय हे जय हे जय 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 हे।
I request you to all please take a cup of tea outside, in the food area. You can have a tea and in the food area.
आइए सर मैम का भी करो।